Hello everyone, welcome to our second day in SoScience. We are here for last day of your event. It was very exciting in first day. I hope in this day you will be excited as well. I'm going to leave the word to Samet. Hello Samet, how are you?
Fine, thanks. And you?
I'm fine too, thank you. I'm going to give the speech to you. Have a nice session.
Okay, thank you. Hello everyone, I am Samet Başkaya. I study Bioengineering Department in GTÜ as a preparation class student.
Hello and welcome to everybody. It is so exciting to say that we are starting the SoScience first section today. I'm İdil Polat. Also, I'm undergraduate student of Gebze Technical University in Department of Molecular Biology and Genetics, and I'm in my first term.
Hello everyone, I'm Ruket Özdemir and I study molecular biology and genetics as a second grade at Gebze Technical University.
I will present you the first section. Our first guest is Dr. Ali Ertuğ. He works at IEEE Helmut Zentrum München. His field of study in entities of cheese engineering and regenerate medicine. Today he will tell you about artificial intelligence-based imaging methods for next generation biomedical solutions. If you have questions, you can write to chat box during chat box during this section. You will be able to ask question at the of the section. And I end my speech here. You can share your screen for views. Teacher.
Teacher, if you're ready to, we are ready. You can share your presentation or we can start.
Okay, do you see my presentation?
Yes.
Do you see the bridge?
Yes.
Good. Okay, shall I start?
Yes.
Yes.
Good. Thanks for your invitation. Guys, it's a pleasure to talk to you. Hope that I can give you a little bit of what we are doing in the last years. So I will talk about some of the published work, but also some of the very recent work. Now, you can put the links to the chat, so you can download these bioarchive papers. Those are our most recent papers that you can have a look after the talk if some parts you missed. So now if we think about all biomedical research, you know, there has been, I guess, several hundreds of thousands of papers and maybe millions of quantifications and graphs related to research of biomedicine. And if all these papers and graphs, they boil down to one, it would be this one. So that would be the average life expectancy of human being. So how biomedical sciences affects that. You can see, so have several hundred, even thousands of years, actually the human being lived average about 35 years. A major reason, of course, for that, people die very early, premature death because of accidents, infections, wars. But now with the, of course, modern medicine, hospitalization, we can tackle a lot of these problems and we started to live almost around average 80. But you see now also in the last decades, looks like this progress is slowing down. We are not potentially able to reach, we won't be able to reach 120, which is our maximum capacity of living, at least recorded in some of the individuals. So a reason, of course, for this slowing down because of the complicated diseases. Those are heart diseases, cancer, neurodegeneration, and others. So we are not able to deal with these diseases similar to infectious diseases where we have an antibiotics or a vaccine. So major problem behind this diseases are complicated. Might be several genetic factors, several environmental factors. So therefore, it is important that we have to accelerate research, biomedical science, that we can investigate multiple factors at the same time. We cannot go anymore one by one, which was typically what has been done so far. Therefore, my purpose in science is to generate technologies and accelerate discoveries of personalized treatment for these complicated diseases. And how are we going to do this? Our approach is to use technologies, those are unbiased, high throughput, that is scalable and targeted. That means we can specifically treat individuals and problematic pathological cells. In the core, we have tissue clearing technologies, and everything else we integrate around, from engineering, AI to nanotechnology. I will introduce you in a short time what is tissue clearing. But first, let's start with precision medicine, personalized treatment. So you might heard about this in the last years. It's also a bit buzzword, getting popular, but may not be clear what does it mean. So precision medicine has a couple of steps. In the initial stage, we have to understand disease heterogeneity, complexity of diseases. And that means if you think about, for example, lung cancer, only, you cannot just say there is one lung cancer. It might be hundreds of different cancer from person to person. Even the lung cancer cells within the person might be very different. So there might be hundreds, even thousands different types of cancer cells within the person. So we really need to understand this complexity, and luckily we have tools to investigate this, from transcriptomics to proteomics to AI-based technologies. But usually what has been done, we take very tiny piece of tissue and then do all analysis, and we ignore the rest of the body. Of course, that can introduce bias. So that means we don't see the other cells that might be problematic, but away from where we sample. So if we understand first the complexity, then we can develop treatments for these cells and pathologies. And that's important. So that means we develop treatments per person throughout the body, every cells. If you think like chemotherapy, which has a lot of side effects because it has no selection, it kills every cell. But we need to bring the treatments to only cells that are problematic. So how our technologies can impact all these steps, if we now start with disease heterogeneity. As an organism, we are an organism of interconnected systems. From head to toe, we are wired with vessels, nerves, and other systems. Therefore, a disease affecting one part of the body will affect the rest. But so far, there are really good technologies to look at the biology at whole body level. If you think about imaging modalities, we have MRI, fMRI kind of modalities where we look at the gross anatomy, but we don't see the cells and molecules. If you want to see cells and molecules, we can do tissue histology, but then the problem with the histology, we are just looking at a tiny piece of tissue. So our technologies combines these two. We can look at the whole biology, that could be whole human organ or even whole human body at cellular level. And we can do that in a time and cost efficient way. So what is tissue clearing? You might be familiar with tissue histology, where we have biological tissue. We take it out, we section, we stain, and look at under the microscope. That's very standard histology. And that has been great for decades, but it's not enough if you want to look at the entire picture, big picture. And also, it is not enough if we want to study the networks of vessels and nerves, which has a 3D meaning. So we cannot look at just fragments and understand or try to understand what is wrong, let's say, in wiring of the nerves in a neuropsychiatric problem disease. So to overcome this, we have been developing so-called optical tissue clearing technologies where we convert biological tissue into transparent structures. You can think this converting milk into water. You can see through and scan through to obtain cellular level information throughout the biological specimens. When I started my lab around 2015, we wanted to bring these technologies from mouse organs to whole mouse bodies, because mouse is the most commonly used animal model to study diseases. So initially we developed UDisco technology where we could render whole mouse bodies transparent and also scan for the first time with light sheet microscopy. Then in 3 years, we developed UDisco technology where we could amplify the signal about 100 times. So if you look at, for example, these are both same mouse transgenic line. On the left, we don't see the nerves very much because there's high autofluorescence, but on the right, after amplification, all these nerves throughout the intact mouse body very visible through intact skin and bones. UDisco technology truly renders the whole mouse transparent. You see it like a glass. If you put it into solution, it becomes totally invisible. And now with the lights coming from side, and we can scan those transparent biological tissues, even whole mouse bodies at cell level. So in neuroscientists, I wanted to study first the nervous system of an adult mouse. You can see here, for example, again from this Taiwan GFP mice, you can see the wiring of the nervous system of entire mouse. So what we did actually in this work, we did a local brain injury. So you can see here at the right somatosensory cortex of the mouse, we did a local brain injury. And then we did, instead of just looking at the brain, after a brain lesion, we look at the whole body and identified, for example, the axons here that are innervating shoulder blades after brain injury. They're highly reduced compared to controls. So this is very simple experiments, but I think it tells a lot. We do a local brain injury and it spreads throughout the body. So that is quite interesting to know, because now that means anything we want to tackle in terms of diseases, we really need to look at the whole picture, not just a local network. As you can imagine, these are hundreds of thousands of optical images, and we have terabytes, even sometimes petabytes of data. We cannot analyze them using typical standard technologies such as IMARIS or ImageJ open sources. They will fail terribly. So we have to use smart technologies, in particular deep learning we have been using. So we develop algorithms to study cancer metastasis and drug targeting in whole mouse body, and also technologies to analyze vasculature down to capillary level, as well as algorithms and mapping technologies to visualize cells in the intact human organs. In RESTUP technology, what we did was to label all the mouse brain vasculature down to capillary level, and afterwards we could visualize them using light sheet microscopy and analyze them deep learning. You can see large vessels as well as in cerebellum and cortex, but if you dive deep into brain, you see all this capillary level information throughout the intact mouse brain. And then we can quantify everything related to vessels, their diameters, branching points, and length, and so on, and register it to Allen Brain Atlas, where now we have vascular information in 1,000 plus brain regions. So that means we can now visualize how the vascular structures are organized in every tiny brain region. I think this is very exciting because we know that vessels or vasculature is a proxy to the diseases. If there's a tiny pathology, if there is a tiny metastasis, for example, cancer or initial stages of Alzheimer's disease, maybe some plaques, those vessels will start to change shape. And using this technology, we can identify these regions of the change in the brain and dive into the molecular mechanisms. Using these technologies, we not only the vessels in the brain, but outside of the brain, we were also able to visualize, because we are not taking, when we studied the brain, we are not taking it out. It remains in the head with skull and meninges. Therefore, we could very well visualize easily meningeal vessels and the immune cells inside. So while we were doing these experiments, what we identify, actually human skull marrow has connections to the some vascular connections to the meningeal surface of the brain, which we name skull meninges connections, SMCs. You can see these individual cells in these channels. So this is from transplant mouse head. Actually, the same we could identify with the human. You can see in post-mortem skull samples with meninges. Again, these channels are there, and there are these immune cells inside. So we now believe that these immune cells mediate these channels mediate immune cell trafficking between skull marrow and the meningeal surface of the brain. Therefore, it is important to understand what is the nature of these cells and what do they do into brain. And we believe they can be leveraged to diagnose and treat brain diseases from a new perspective. So to be able to do this, use skull in a way, first we have to understand how it is different than other bones. Is it like skull marrow, the calvaria bone marrow? Is it just another bone marrow, or is it different than the bones in our legs and arms or other part of the body bones? So, and also we need to understand its relevance in diseases, in particular human diseases. We started investigating this by inducing a stroke lesion in mouse heads, and you can see here with transparency and imaging the neutrophil populations. On the right-hand side, we see this enormous increase of these neutrophil populations compared to the controls on the left. So we do a brain lesion, and there is this increase in neutrophil cells. And when we do transcriptomics, like experiments, look at the different bones, and what we see is that skull has unique cell populations compared to other bones like pelvis, femur, or humerus. And when we quantify again in transcriptomics, so-called ligand receptor pairs, what we observe is that skull has the highest number of diversity. If we move to proteomics and we observe the same heterogeneity, bones are different, but in particular, skull is very different than other bones. And when we were studying this transcriptomics and proteomics signature, we identified that TSPO is a protein that is observed in inflammation. Its RNA here was increased in Alzheimer's mouse models in 5XFAD skulls compared to the rest of the body. So when seeing this, then we did TSPO PET imaging in human patients, because it is used already in the clinic. We studied this inflammatory reaction in Alzheimer's patients, more than 50 Alzheimer's patients. You can see this increase in the inflammation, not only in the brain, which is expected, also at the skull of this patient. So you can see here very clear increase in Alzheimer's patient's skull, the signal compared to controls. This is very exciting because now we can use this technology to look at the changes in the inflammation, probably at the very early stages of the diseases. So when we have been developing these technologies, we apply into a lot of neuroscientific questions, but we were also interested in opening and solving major problems in other medical fields. A major application for us was cancer metastasis, because majority of the cancer patients, they die from the metastasis of the patients, a metastasis of the tumor. So it's clear that we have to understand and treat this. So how that happens is hundreds, even thousands of tiny tumors coming out from the body, from the primary tumor. They go throughout the body and they hide this tiny metastasis. And when the immune system is down, they form large metastases and kill the person. Therefore, it is very critical for us to understand and visualize this tiny metastasis and make sure that they're treated. If you look at now imaging modalities for cancer study, bioluminescence, MRI, PET, all will look the same. To be able to see something, we have to have thousands of cells together. Otherwise, we won't see anything. So we said we can solve this problem. We see that the tumor into the mouse, 2 months later, it spreads. We made it transparent, and we start mapping cherry expressing cancer metastatic cells in whole mouse body at cell level. But in addition to looking at the cancer metastasis, we also visualize the therapeutic antibody. You can see here in green with the red dots. That is, we could visualize this drugs, therapeutic antibody drugs, in another fluorescent channel. So in a standard imaging technology, you can see here a primary tumor at the leg, but then there are 3 metastases, nothing more. So in particular, in the lungs, you see nothing else actually, but the same animal goes through our DeepMap technology, transparency, light sheet microscopy, and AI-based analysis. Now what we see is hundreds, even thousands of immune cells in the mouse body. You can see now, for example, in the video, so there is the primary tumor at the leg, but there is this hundreds, even thousands of metastases spread throughout the body. You can see all these nodules all around, but a lot of them are in particular in the lungs. You can see really down to cell level their metastasis. Not only this cancer metastasis we could visualize, also targeting of drugs, as I mentioned. When it's targeting the tumors that giving drug, we see that it's in white. When the tumor is not targeted and not treated, it remains magenta. So now, for the first time in the history, we have access to cancer study at cell level throughout experimental models, and really assess the quality of drugs at cell level. This antibody drug, for example, was able to not treat about 20% of the metastasis throughout the body. So this is important information, because now we know that it's not perfect, not good. We have to go back to the lab and improve it and maybe bring a second antibody or drug. So the next level is from looking at visualization of tumors. From here, we have to go into understanding the molecular mechanisms behind this metastasis, that we can now start developing personalized treatments. For that, we developed so-called discourse technology, where we could take out this cancer metastasis that we visualize and sample from targeted versus untargeted regions and run mass spec-based proteomics. So we could do these extractions using robotic technologies that we developed. So there you can see this robot communicates with the microscope during imaging. Wherever the tumor is identified, it can able to go there and then sample, insert a biopsy needle, and then finds this tumor, solubilizes and sucks it out. And then we can do this in multi-regions throughout the animal body. And we can then bring this back to the multi-well plates. From there, we can go and run large-scale molecular profiling proteomics. So that helps us to understand the mechanisms behind these individual tumors. From there, we can start developing drugs and delivering them to the targets. Even the antibody-based drugs, we know that they are not perfect. As I showed, they miss 20% of tumors. So clear that just drugs alone will not help. So we need some smart ways, the carriers, so-called nanocarriers, to deliver them to the specific cells. For that, we have been working with DNA nanorobots, where we can fold them in a different three-dimensional structures and form diverse structures from spheres to the scissors and other things. So it becomes really like powerful, like Lego bricks. We can really build up any kind of carrier. And then they are actually because they are oligos, they are biodegradable, and then we can program them for multifunctions. In addition to that, even gene editing tools can be encoded to these carriers themselves. So these are nanoparticles. However, to study these nanoparticles again in mouse bodies was not easy. People use PET kind of imaging where now resolution is around 1 millimeter. So we are, of course, there's like 6 orders of magnitude difference, so 1 million times difference from millimeters to nanometers. So basically, PET bioluminescence will not show us nanoparticles at all. So we have to at least bring this imaging to cell level that we make sure those nanoparticles, they arrive to the cells of interest. For that, we targeted nano rods here, DNA nanorods with antibodies. You can see this EM pictures on the right-hand side. The antibodies are at the tip of this nanorobots. So these are like targeting structures. So these are like the address of the letter. Then here we targeted them in the mouse body to immune cells. And you can see, therefore, they're mostly going into the spleen, but also lymph nodes as expected. But if we do give the non-targeted version, they are just going through the entire body, but they are also taking mostly into livers. But if we target them to metastatic tumors, you can see that they are going into the tiny metastasis. So now let's look at the whole body of this animal where there is the immune cells targeting. You can see that this DNA origami that is targeted the immune cells, they are mostly in the spleen. You can see a bit whitish because spleen has the highest number of the immune cells. Hundreds, even thousands of immune cells are there. We can really resolve them down to cell level in these clusters. So not only in the spleen, but you can look throughout the body to other organs. For example, in the liver here, you can see also individual cells that are being targeted using this technology. You can see really down to cell level the immune cells in the liver that are targeted. So now we can look at, for example, to the lungs of the animal. So again, we are traveling in the intact animal and looking at where our nanocarriers they went. And as expected also in the lungs, immune cells, they are being targeted with these nanocarriers. So now we have also technology to deliver this drugs at cell level to desired cells. So far, all the experiments I show you, using transparency technologies, is there on mouse. You might ask, would they also work in human tissue and human organs? Yes, actually, they also work in human tissue. It took for us a bit time, but we could also make human organs here, whole human kidney, transparent. And after transparency, again, we could use light sheet microscopy and AI-based image analysis and obtain 3D map of an intact human organ. You can see, for example, the capillaries on the left and on the right and the collecting dots, but also if you dive in, really we see at the glomeruli level details and cell level information. You can see how this capillaries and vessels all around the glomeruli. And now using this human organ maps, what we would like to do to generate engineered organoids and human tissues using bioprinting. So the logic is here, for example, if, as I mentioned, in VASAB technology, we can visualize all these vascular details, and that gives us an architectural map of a human organ. You can think there's really an architectural map of a building, skyscraper. When we have this map, we can bring them to the construction and start generating small-scale organoids, small-scale organs that we can use to test drugs, but as well as we can use these maps probably to generate whole large-scale human organ. That's quite visionary, which may not happen before, I don't know, 20, 30 years. But of course, when we get this, we can generate human organs on demand with bioprinting, probably whole biomedicine will be different than how it works today. So with that, I would like to end. And of course, a lot of research agencies contribute to this science. And that's the team, our AI team, behind a lot of algorithms. We have a robotics team working with biologists and physicists and chemists at the other side. So you are very welcome to follow us on Twitter, so @Lab, so where we usually post our most recent developments before their peer review publications. So with that, I'm happy to take your questions.
Thank you for your presentation, Doctor. Now we can pass into the questions. First, we have some questions. You can start.
Okay.
Okay, I start. Is there any advice you want to give us when we are following our career for reaching the success? What's the advice you could give us?
Wow, that's a big question. I guess what you are doing already is something nice to get involved with the community, to follow what's happening, and not only just at your university, but beyond. Science is very dynamic, and a lot of things are happening, not only in biology, but other fields. And to be able to be informed is important, to check out what's happening in the world in terms of science, what are the major developments, hot topics. This might help, for example, to orient your interest. I guess a recent trend clearly, for example, AI, deep learning, and I would recommend people to definitely learn more about it and use it as much as possible. Work with people that is doing algorithmic developments, not, that means biologists comes together with computer scientists and solves a problem on image quantification. So this kind of emergence of fields are becoming very important. So I guess this is also doable at the local scale where you are and interacting with the people at the university and other departments.
Okay, thank you. Özlem Baydar have a question. Do you have any inspiration for your work? And if so, what is it? Thank you.
Inspirations, it's a good point. I guess it's a little bit checking out a lot of things, what's happening in general in science, not only in my field, but also other fields. That's important. And always think out of the box in a way, let's say, think a bit brave, like things that people might think wouldn't work. Usually there is a solution to make everything working. So for me, I care about ideas, and I would not, let's say, categorize them undoable relatively easily, as long as it's something that can be quite impactful. So it is, of course, not easy, but overall, I guess a lot of that means inspiration would not come in the dream. It means a lot of reading, a lot of communication with other people, being open-minded, not being stuck to your own ideas, saying what I'm doing is the best science, the rest is not very relevant. That is not a good approach in general. Being open-minded, checking things out, talking with different people, trying to create combined ideas might help to bring new next generation biomedical solutions, maybe by following such a path.
Thank you. Ahmet Furkan Çodur asks, first of all, thank you so much for beautiful presentation. What was the moment that you felt upset or angry in your academic life?
So it is, I guess this is not a personal question. This is a question, or the answer would be for everyone. So everyone in academia will have the same problems. Their papers will be rejected, their grants will be rejected, and so on and so on. And this is a very normal challenge, academic challenge. Academia is a bit different than other, fields where, there are some things that you cannot control as well in that regard. Let's say if you are trying to publish your paper, things that it's not in your hand, you don't know the reviewers, editors, and so on. But this should not be upsetting, or could be some for a moment, but it shouldn't be too long. One can always learn from the mistakes and find solutions, so that's important. I guess let's put it like this: what separates the best researchers than the average researcher is not being able to give up. The best top researchers, they never give up, even though they have the same problems, even though they are encountered probably even more problems. They don't stop. They always say, I'm going to find solution, move to the next level. So that's the key in academic career. If you want to do that, it is less important to feel upset or angry or blame others. It is more important to learn and find solution to the problem and move to the next level. And this should be on never-ending circle.
There's a viewer asks, Şeyda Yağcı, the pandemic has also affected education and internship opportunities abroad. How can we close the gap of opportunity?
Yeah, I mean, there's some opportunities not there, some opportunities are there. Like Zoom meetings, now they weren't opportunities, now they are opportunities. And a lot of meetings all around the world, a lot of educational information is online. Let's say if someone now wants to learn AI deep learning, probably you don't need to go anywhere. You can just sit and watch all these hundreds of thousands of nice materials that are being recorded and put online for educational purposes. Of course, you may not be able to do some hand-on stuff like handling mouse, whatever that might be. But I guess there's more opportunities now than in the past to learn, to network. At the end, most of science, scientific development relies on this kind of networking activities. If you want to do, if you want to start your PhD after your undergrad, it is less important that you know how to hold a mouse, I would say, than you know how to find a solution to a problem. Because you will learn how to pipe it in a few days. But if in general, if you don't have the character to solve the problems and not giving up and pushing things, that's not going to be successful regardless.
Exactly. This question is from Emirhan Balcı. First of all, thank you very much for your presentation. It was very useful for us to explain what many of us couldn't understand from the articles during our student years. You said that the studies you are working on can also be applied to human tissue. Is there a specific time for this? And do you actively work for it?
Yes, we are actively working on it. I show you the human kidney. We are working on human heart. We are mapping human heart. And for us, it's important now to generate cellular level map of a human heart. So again, the aim is to use these maps as an architectural maps of generating new organs. It is very interesting to think, actually, if you think about human organs, every human organ is different at cell level. It's not the same. But the general structure is the same. If you look at kidney, it looks like bean, even though it's a bit bigger, smaller. The heart, you know, maybe in a big person it's bigger, in a smaller person smaller, but it scales up. So there is a structure behind, and engineered structure behind. So that means, you know, it's just scaling up the units. If you think about kidney, if you know how to construct a glomeruli with how many capillaries, what is, you know, distance between units, all these things, very mathematical formulations. When you have them, you can scale and then make them even larger and larger. So that's what we are aiming with these human organ maps, really to generate these maps that we have in the core, and we can then make small for drug screen, we can make human organs big for transplantation. Again, this is relatively big vision. It's not only the map problem, but there are additional problems around printing the cells, generating the cells, and so on. But I think this is the first step actually we need to solve in general.
Thank you. I want to ask a thing actually. You use artificial intelligence or machine learning for data or visualizing. Do you use these things, I mean, artificial intelligence or machine learning, in all works, or just now? I mean,
so my lab is a bit different than standard biomedical lab. We do AI almost for every project. That is a bit unique, and we started relatively early. I mean, this AI, of course, is famous in the last 2-3 years, but we started, you know, even 5 years back, all these developments where people had no idea about what deep learning can do. So we now more or less integrated into entire research, our research deep learning, because we do a large data. So not only imaging, but also with proteomics, transcriptomics, there is this large data that's about molecules, and everything is for us in our hand analyzed using deep learning. So more or less, yes, we are at this point, almost there's no project in my lab that is not involving AI.
Thank you. Anıl Emin Oduncu have a question. We thank you for your participation and this beautiful presentation. I wonder that what was your first talks and plans while beginning to study that?
I don't know what the question is really. So the first thoughts and plans, the beginning of the study, like the transparency work, I don't know if that person means that. I mean, this is overall something in science very also important to keep in mind. What we may think very exciting now might be very exciting in a year or two. Things might be very different. So it's important to start somewhere, have some ideas and try to solve it, but also be open to quickly direct the research into some directions which might be interesting. So when we started, initially we had some different questions, different projects, which most of them didn't work. But now we have we found some other things on the way that could make, that could work and solve a lot of those problems. So it is always important to see, again, find solutions, not only just being stuck to always the same problem, but sometimes solutions might be just changing the path and moving into another direction, being opportunistic. So I guess we have to try both, be persistent to solve problems in hand, but also be flexible enough to just change the whole ship and move to another thing. And we have been doing this, I guess, relatively well so far. And I know that it might be scary sometimes if you work on a project a year afterwards, then you want to change, so not easy, but sometimes that might be a better solution than being stuck to it for 5 years.
Thank you, teacher. There was a question from Feyza Keskintaş. Thank you so much for your presentation. I wonder that, are there any online platforms we can take courses from you?
From me, I mean, the major thing that we have is really, or what we do is Twitter you can follow. We have so-called discotechnologies.org website. I can also post, I think if you just Google Disco Technologies, that will give you some of the things that we do. So that is, we have their workshop videos. So if you want to do, I can put chat into chat here. If you want to do, let's say, develop do some clearing there in whatever biological lab, and most of these clearing methods are very easy to do. It doesn't require a lot of effort. So we have their videos, like a workshop. We showed the details of this, the technologies. Beyond that, I don't have, again, much of courses, but there are others. So as I said, you can follow me on Twitter to see what we are doing in general in terms of research and what we share. But there are many platforms that people can learn, I guess, a platform from Coursera, from Stanford, and other places to get online courses. So whatever topic you are interested in, I'm sure there's enough resources online to get yourself master on it. You may need to choose wisely, of course, what to study, what to learn. And in that regard, again, I would recommend definitely learning more about technologies such as AI, such as transcriptomics, proteomics, spatial transcriptomics, this kind of new technologies that would be quite helpful.
Thank you for your suggestions. The questions is from Sude Toslak. She asks, first of all, thank you for this beautiful presentation. Do you provide scholarship to your students from abroad? I actually was gonna ask this question from you to you.
Good point. I mean, in general, this depends on the lab that you want to go. And, you know, I usually provide, I give a scholarship to the students, but that also depends on how long the person wants to come or can come, and whether also sometimes the regulations may not allow. Sometimes there are master students somewhere. If you are registered at a master program and they want to do internship at my institute, rules, they don't then pay, they cannot pay because saying that this person is master student. So there are sometimes regulatory issues depending. This one has to check. I guess at the end of the day, what is more important is to find something that is interesting for you, and that you are, you can communicate why you specifically want to go to this lab and how you want to contribute. Not only just saying, I want to come to internship, can you take me? You know, if you think, such labs might receive hundreds of such applications. So basically it has no relevance if you just ask something like this. I think it's important to be tailored to the lab, saying, okay, I'm, seen this work, sounds interesting, I want to improve, I want to maybe even, bring something from my expertise, saying I'm computer scientist, or I did imaging, maybe whatever, you have to think about a little bit how you want to write an email. You have to prepare a nice CV. CV should look well organized, without typos and so on. That's also important. So you should look a person like someone who is in control, not just doing sloppy work. So a lot of things should come together, that one can do some generate, let's say, interest at the other side and get scholarship. So it's not just even availability is also like a competition among so many applicants, why you have to just very make clear why you should get support but not others.
Thank you, Doctor.
Ezgi Olgun have a question. Thank you very much for your presentation. The question I want to ask is to get some personal advice from you. As a bioengineering student, which programming language do you think would be beneficial for my career if I could not learn? Thank you.
Again, so which programming language? I think it's just, as I said, at this point, deep learning, machine learning, AI is the most relevant thing. There you can find relatively quickly which platforms you can learn machine learning, how to do deep learning. Just focus on this. I guess that will be most relevant. That's it. You know, I guess there isn't really much into it. If you say, how do I learn deep learning? Google, you will find enough information how to do it.
Thank you, teacher. There was a question from Şevval Karabulut. Thank you for the excellent presentation. Do you have any advice for the student who want to move into your field?
Yeah, I mean, this depends on from which field to my field. It's yeah. I guess important thing would be really to understand, to have a good view of what's happening in general. What are the important topics? You know, if someone can clearly express some ideas about, for example, AI and deep learning, I would immediately say this person seems to know more than general biology student, because clearly now biology is merging with AI. And if you're not able to follow this and express this clearly, probably you're not better than average student. So I guess important thing for me, at least for me, would be to see this thinking of new technologies, diversity. And of course, you know, in such cases at this career level of career, you have to be very dedicated. You know, sometimes in life, there's you have to really do, you know, there is to create and do some opportunities. If you make this a couple of times well in life, that can open a lot of things. So you may need to really work hard to generate opportunities if you want to do internship or move to a field or show yourself in some ways. Yeah.
Thank you for your advices. I want to ask a question. What is the moment, keystone moment actually, that moving you one important step further? Do you remember this moment?
Yeah, I mean, good point. Let me see. I guess, yeah, I did internship. That was, I guess, a good thing for me to do when I was at the second year, that I could go to abroad and see, experience stuff and learn a lot of things, even though I wasn't very well qualified at that time, but still was a courage to do it. I guess it's important to reach out to people who can help and advise for anything. So, you know, finding some mentors around you, that's important. You can ask, okay, I want to apply. What do you give as an advice? Is this letter sounds interesting? My CV is good. And you have to really check things with people with who has expertise if you cannot find the best information online. So I guess, you know, some of the things might be, of course, in the career may look like also luck, how things suddenly happens, but usually there is not really luck. I don't believe in it much, or let's say not little, maybe there is little, but if you create opportunities that there is something happening, then it will be, you can say that happens, right? Let's say if you initiate contact and talk to people and ask, and they know you, if you contact 100 people, maybe one of will help you to do something. But if you haven't contacted anyone, if you haven't done anything, well, you can call yourself unlucky, but also let's say motivated or unmoving. You have to move into the target. You have to generate opportunities. So I think that was in general what I was doing a lot, to create always some options and communications that can help me on the way to improve.
Thank you for sharing this moment with us.
Hilal Yunan have a question. What are the things you wish you knew before starting university? Thank you.
Yeah. It is, I mean, I don't think about this question because it's a bit irrelevant. I guess the most important thing is every time of the moment, what we learn and what we do. I guess it's not very useful to think about what I should have learned in the past or no, but I couldn't. I mean, I guess that is in general no use, but of course it might be important to have some reflection moments. If, for example, we miss an opportunity, we can say, oh, why did I miss this? Let's say now, again, if I go back to the deep learning, AI is an opportunity, and if no action, if I made it, but if I haven't done anything, I would go back, yeah, why I didn't start using this technology early on, what was the problem? So one can do this kind of reflections. One has to do, I guess. But otherwise, I guess starting university is, for me now, very far away. So I cannot really have a concrete, and it would be anyway irrelevant to what I'm doing now.
Thank you for your answering. And there was a question from Ufuk Shen. Thank you for presentation. At the beginning, you have mentioned that lung cancer can vary even in single person's lung cell. And while you are investigating tiny tissue, can metastasis be tracked? Or are there any technologies that can provide also full body information by taking another tissues from another parts of the body? Thank you.
Yeah, I mean, in terms of imaging, unfortunately not. So that's the whole body level imaging at cell level doesn't exist other than what we do. Of course, there's one can take biopsy-like things and do transcriptomics and proteomics studies to also study disease heterogeneity. When we think about this, every person has different cancer, and even one person has different cancer cells. There are ways to investigate this so-called the single cell transcriptomic studies. These are important studies, but usually there we have to take out a small piece and then do investigation on that. So that limits us, but still it shows this heterogeneity. It doesn't show everything, but it shows that things are not simple. So overall, there is this mixture of technologies, not only imaging, but there is this transcriptomics and proteomics studies. Therefore, nowadays, even the best or the hottest technologies are the ones that combining them, so-called spatial omics. That means imaging plus transcriptomics or imaging plus proteomics.
Thank you, Doctor. This is the question from Sude Usta. There were master and doctorate students on your laboratory site. Do you accept bachelor degree students to your lab?
We have, internships as well. I have a few internships time to time coming. Problems with internships usually is that there's usually short time, I'm a bit reluctant to myself hire people for just 6 weeks or 4 weeks. We actually have a rule, more or less, we don't hire anyone less than 6 months. And of course, if you're studying at the university, 6 months is not a possible time probably to have. But I would then ask, I would think that if you are really going to go somewhere, you have to generate the maximum time possible, maybe 3 months, maybe 4 months, whatever, stretching it to end. So you cannot say, I'm going to make a 2 months vacation, then 1 month internship. So that would probably not attractive for many people.
Actually, this would be a great opportunity for us and our guests.
Seda Kakaç have a question. First of all, thank you for your presentation. What are your recommendation for a molecular biology genetics student to special in choose engineering? Thank you.
In tissue engineering, I mean, the technologies aren't very different. Tissue engineering, of course, you need to know more about, depends what you want to do. Tissue engineering might mean different things. If it's about bioprinting, then you may need to know about biomaterials and how to grow the cells and engineering of maybe printers and so on. On the other side, if it's about organoids, we can also call it tissue engineering. That's mostly biological, how the organoids field works, what are the major organoids, and how they are used for diseases. So it depends on what kind of tissue engineering field. Again, here there's a few key hot topics. Again, organoids is one of them. One has to know about them well, at least on paper. AI is another thing, of course. On the other side, I guess technologies like transcriptomics, proteomics, spatial omics, these kind of things are keywords and important to know in the core what they mean for biomedical science.
Thank you for your answering, teacher. There's a question from Cemre Hayfeoğulları. Thank you so much for this incredible presentation. There was a part about the mouse brain in your presentation. Do you have a special reason for working with mice?
Well, mice, they are the most commonly used animal model. And that's the major reason why we vote mice, because a mouse, we can imitate a lot of human diseases in mouse. We can create genetic mutations in mouse, which can imitate some diseases. Mouse genetics, of course, is very easy to control. That's important, but also mice are easy to breed. Keep them in the cages, and so you cannot do the same with monkeys. Of course, it first takes a lot of time, more rules around, of course, also not nice maybe, and so on. So mice are at this point, I guess, the victim of science. They're used the most. This might change in the future more and more if we move to these organoids as well as AI. So that means we do computer modeling of diseases more and more than using animals. That's a future hope in general in science, but we are not there yet.
Yes, though mice are actually model organisms. This is another question from Senagül Yılmazer. Thank you very much for this productive presentation. What would you recommend for us to be better in university in our student life? Is there a specific program or something like that?
I guess we covered a little bit of this. Again, important thing is to have some advisors, mentors that you can get information, right? And make contact, communicate with people, follow people, and and I know them more. Then hundreds of thousands of other people, because they try some visibility, right? Even though maybe they're not qualified anything, but doesn't matter. You show some visibility. I think that's important in general, to have some presence. You don't have to do this all the time, but you know, for some key things you identify, I guess to be visible is important. Again, I guess in online there's all kind of information classes one can find, especially nowadays. And there's a lot of, anyway, Zoom meetings there and there. You can join the online meetings, conferences. We covered some of the key technologies to be, which would be interesting to know in biomedicine, from AI organoids to spatial omics, even maybe like technologies like cryo-EM to do protein, 3D protein structure. I don't know, you should know about AlphaFold, what it does, what does it mean for science, this AI-based protein structure predictions. You know, these are very key developments in science in general. To have information about them and to follow them, I think would be relevant to show also your general interest in science.
Thank you, Doctor. Esma Şevval Yeşil have a question. How do you run your social life with scientific research? Thank you.
That's a good point. I guess overall it's important to have some social life, but I guess also social life should be somewhat meaningful for you to be creative. I do photography. That's for me maybe something helps me to relax. The traveling, being alone in nature, whatever. Otherwise, of course, being people with going out with people, you know, smart people that you can chat about smart topics, I think would be also relevant. To have, you don't have to have 1,000 Facebook friends, but a couple of good friends where you can, you know, talk about some generally what's happening in science in the world. I think that would be relevant and important. Yeah, so work-life balance is important, but I guess how we think about work is also important. I mean, for me, there isn't really very split. I'm not really saying my work is very defined. You know, I'm happy to work when I'm on vacation, or I'm happy to have vacation when I'm at work. So, you know, maybe I have the choice that I can do it, but for me, it's not very, very defined, because I really enjoy my work as well. For me, it's a relaxing thing to do in general as well.
Thank you for your answers. There's a question from Buse Ünlü Eroğlu-Gil. What are the methods of accessing the data you use in your studies?
Accessing the data, I'm not really sure what does it mean, assessing or obtaining. So we do microscopy for imaging, we do proteomics for molecular profiling, we do transcriptomics. So we have a lot of different technologies. So I mentioned very early in my talk, technologies that are high throughput. So that means they can really do millions of cells studies, millions of cells at the same time. So we are very much focused on this high throughput technologies. And unbiased, that means we don't need to sit and analyze the data, and data is usually analyzed and revealed by softwares to us. That means, you know, if you, in general, in a major problem in science, you can look at slides, make an image, and you know, if your hypothesis is to have less cells, you count less cells and you have less cells. So this is very typical that people do this to biased investigation to be able to publish and so on. So we choose the methods that will move away from this, unbiased and scalable.
Thank you for the answer. Rabia Köse asks that, thank you so much for the informative presentation. I wondered that what has surprised the most in your research so far? Was there any big conflict between co-workers? Thank you.
Yeah, I mean, surprises are happening all the time. There's change, and I think that's good. You know, things that you don't expect becomes a major part of the science. Again, maybe computer science, AI, I was not expecting it's going to take the center stage, which now it did. Conflicts, of course, happens all the time. That's also normal. One has to learn as a, to be good scientist means to be good manager or good person to deal with. So one has to learn how to deal with conflicts. I think that's maybe additional things when you ask me what I should learn, to learn how to communicate and how to resolve conflicts at best. So there's always solutions. You have to know how to listen, how to express your idea, and so on. So these are very key in general, not only for science, but to get any achievement. And these are actually probably the most important things when it comes to getting higher and higher, because information becomes less important because you can Google everything, you can find solution to everything. But the important thing, do you have the character that someone can sit with you happily, talk about ideas and say, okay, this person is nice, and I give you a task and you solve it, come back to me. So in general, that's an conflict management and how to make best out of social situation is very important, that everyone has to really work on it. Yes.
Thank you, teacher. Altun Tokan have a question. Thank you for the presentation. Do you think that you are doing well with your students, even maybe there are age differences between you and them? Thank you.
I don't think that age difference is important. So of course, there will be age differences anyway. But the important thing, of course, how to understand everyone's situation, to be to look with their status and so on. Of course, not everything is as I think. I might think a high level. In a way, things might be less problematic for me, but for a student, it might be more problematic. One has to. But then, I have also people in my lab who is helping me to manage the students. Let's say there are other people, the students, they talk, the senior people. So there's a different perspective. I think that's important because I may not be able to see and have time for, being from perspectives of a person, because that will require also time and effort, and I might not have it. But if it is not the case, one can also express, saying, okay, can I make this point? I see this situation like this in my shoes, and so on. One should be able to express themselves as well as needed.
So we can say that age differences is not important thing. The important thing is being respectful to different perspectives.
Yeah, sure.
There's a question from Fatma Tarhan. Thanks for your presentation and time. Do you use a special chemical to colorize tissues which shouldn't be harmful for tissues?
Good point. In general, when we do this tissue transparency, every tissues we use is dead tissue. They're fixed tissue. Therefore, harm in terms of harming living tissue is not a question, because tissues is not anymore living. But of course, it should not also destruct the tissue, even though it's fixed. That tissue, it should not, let's say, destroy it. So we had to adjust the dosage. Maybe made screens in the papers you can check. So this happens with the chemical screens, and you troubleshoot. And the ones that changes the tissue, you take them out. You don't use them anymore. The ones that seems to have effect without destroying the tissue structure, the ones that we use, the ones that we publish.
Thank you for the answer. Gizem Nargazali asks that you mentioned transparency technology. Does the use of transparency technology provide treatment in advanced Alzheimer's patients?
Not necessarily. I mean, again, anything we do with transparency, they are not applicable to the patients or living things. So when we make things transparent, that means that's dead tissue. The whole mouse you see transparent is not living anymore. So we cannot make a human being transparent. They will be dead. Yeah. But what we can do is, of course, use the organs from people who died from Alzheimer's. We take their brain out, we make transparent, and we scan them at very high resolution, and we understand at cell level what went wrong in their brain. And that can help us to identify a solution, a treatment for those who are living. And these are the things that we are doing actually.
Thank you, teacher. Mustafa Eren Yıldız have a question. Do you think to come back Turkey?
I come back to Turkey for vacation, so I think that's for now what it is. I mean, in Turkey, in terms of research and science, unfortunately, is not the level where the high level standards. So therefore, I don't see at this point at all any reason why, in terms of scientists, if you have opportunity, you should work in Turkey. Then somewhere there is more opportunity. Of course, I mean, not everyone can just go out and work outside. If you work in Turkey, you have to make the best out of conditions. But of course, at this point, for me, it would be a big downgrade.
Thank you for answering, but we want to see our teachers face to face one day, if it is okay for you to. When you come to Turkey, we want to see you. So
sure. We can think of.
Another question from Ahmet Furkan Çodur. Honestly, do you believe the energy movements before starting to do something, such as thinking positive before the experiments?
I don't know what does it mean, energy movements. I have no idea. But if you think about thinking positive, of course, that's important in general, to be positive about anything. There will be always problems, that's normal, but being positive means not only the beginning, but throughout the process. And, you know, you say something goes wrong, okay, what went wrong? I'm gonna solve this. You know, there's always solutions. I think that should be the approach. The solution will not come from the sky, it will not come from the dream, it will not come, you have to find the solution. And that's the key. And there is always a solution. So that's, you have to keep in mind. I think that's sufficient to make us positive in general.
Thank you for the answer. The next question is from Aleyna Zehir. Have you ever had a hard time experimenting with mice for your research?
Yeah, I mean, mice, of course, I don't know exactly what she means, but in general, of course, using animal models becomes harder and harder. So because of regulations in the EU and countries, it's harder to use animals, mice even. So therefore, I always try to generate alternative methods like organoids, engineered tissues, AI. So the aim is to model the diseases more in cell culture, even in silico on computers, than using animals. But that will take still some time, because to be able to model something, you have to have a good understanding of what you're modeling. Unfortunately, we don't have this information yet. So we have to still use the mouse to get, or human information to get the best out of it, and feed it to algorithms to start learning and making good predictions.
Thank you, teacher. Sude İdem Uğur have a question. What was your motivation for doing research to find a cure for this diseases?
For these diseases, I mean, I guess in general, I mentioned at the beginning of my talk, so we are interested in complicated diseases. Those are cancer, neurodegeneration, and diabetics, for example, heart attacks. So those are the diseases that most of people die from, and there is no cure. And that's my general interest, to bring technologies and solve the diseases that are very complicated to solve only using standard technology. So that's a bit my motivation in general.
Thank you for the answer, doctor. I wonder your COVID process. I mean, do you have any? Do you have any difficulty in your COVID process? I mean, it's a long term, around approximately 1.5 or 2 years. So, is there any difficulty for you?
Of course, COVID in general affected everything, also us. Our research slows down a lot of things. Hire of the students, sometimes people cannot travel, cannot come. The process are longer. Again, you know, these are some opportunities. You know, let's say some problems creates also some opportunities. Again, we haven't used the Zoom meetings and so on as much. Now it's much easier to connect people, to go to seminars. I'm not going, for example, much in seminars directly, but like this, right? So I'm now doing almost every weekend something. Otherwise, I would not do it. Yeah, if you say come to Turkey, I would not be able to come to Turkey every weekend. But now it's an opportunity that we are able to do this, and we learn how to do it. So again, there are challenges, but always some opportunities to create solutions to places where we get stuck.
Yes, actually, even in this moment, we can use these opportunities.
Thank
Yeah,
you.
true.
Thank you for your participation. It was a really beautiful presentation. Thank you.
Sure.
Thank you.
Sure.
So
Thank you. And sorry?
Go ahead.
I just want to say thank you for the invitation and good luck with the rest of the program.
It was very lovely presentation. Thank you so much. So much question. I hope we didn't tire you.
Sure, it was a pleasure. Okay, good luck. Bye-bye.
Goodbye.
At 1 p.m. and see you later.
Hello everybody, my name is Özlem. I'm the Vice President of IEEE GTÜ EMBS. Today, Şevval is moderated this section and I leave the word to Şevval now.
Hello everyone, my name is Şevval Karabulut. I'm the student at GEBZE Technical University. My department is Molecular Biology and Genetics. I'm the preparation student now. And we will continue with our another moderator, Sude.
Hi everyone, my name is Sude. I am first year student of Molecular Biology and Genetics at GEBZE Technical University. Welcome to our section.
Hi, I am Kübra. I am studying at GEBZE Technical University, Department of Chemistry. I am second year.
Okay. And here are Professor Seamus Martin with us, and he will start his presentation if you're ready.
Okay.
Yes.
Good morning, everybody. Let me just get my slides. So, okay. So, see that?
Yes.
Okay. So today I'm going to talk about programmed cell death, and it's a process that I think is very beautiful. It's how cells die in all sorts of situations in the body. And I'm just going to give an overview about this process. I've been working on it since I did my PhD, actually. So we accidentally came across programmed cell death. There wasn't a lot known about it at that time. And over the last 20 or 25 years, now we know about this process in a lot of detail, all of the genes involved, the proteins that the cell expresses in order to kill itself in various circumstances. So I work in Trinity College Dublin, which is right in the center of the city. And Dublin is right on the east coast of Ireland. And this is the Trinity College campus here. It's Ireland's oldest university, and it's a very nice, quite a small campus in the center of the city in Dublin. So I'm going to start off with a little movie. This is how we used to think that cells die. Cells are going along, minding their own business, going about their daily lives, and then one day something really terrible happens. Now that actually does happen sometimes to cells. But that type of cell death is called necrosis, and that's unregulated, and it's not really controlled by a specific set of genes, although there's certain circumstances where programmed cell death can take the form of necrosis, but most necrosis is uncontrolled. But in fact, most cell death that takes place in the body is actually highly controlled, as I'm going to tell you in this talk. So as you all know, the cell is the basic unit of life. We have 10 trillion cells in our body. That's a lot of cells. And during our lifetimes, we make probably 500 or 1,000 times more than that, because a lot of cells and a lot of tissues are undergoing constant cell death. In some tissues, cells only live for 24 hours. In others, it's a bit longer. It's maybe a week. And in other tissues, cells live almost our whole lifetimes. So the lifespan of cells in various tissues in our bodies is very variable, but there is a lot of cell death that takes place in all of our tissues on an ongoing basis. And as I'm going to tell you, this is the way that the body uses to replenish the tissues, to eliminate cells that are not doing their job properly, or cells that are potentially cancerous or that are infected, or for other reasons. So the cells are beautiful and amazingly complex. Each cell expresses probably 15,000 different proteins. And if you take modifications of those proteins, such as phosphorylation and ubiquitination and alternative splicing, you probably have 100,000 different proteins in a typical cell. And we are still really decades away from understanding how this all works together for cells to function. Now, if we consider the body as a box. Here, the outside of the box is our skin. And then we have the lungs and the gastrointestinal tract. And then we have all of the cells in our constituent tissues. And for this all to work, for our bodies to function properly, there are some basic rules. And as you'll see, if you break the rules, then unfortunately, in the cellular society, that is the body, the punishment is death in most cases. So the basic rules are, of course, you do your job in the cellular society, you do whatever you're supposed to do. You stay where you were born. So in the body, when cells are born in a particular tissue, they stay there. There's very few exceptions to that. The exceptions really are cells. Our immune systems are allowed to patrol the body and go in and out of the circulation and into the tissues and inspect what the tissues are doing. But most cells cannot leave where they're born. If they try to leave, then they're killed by programmed cell death. And we know, of course, in cancer, cells do leave where they were born because they're cancerous and they travel to other tissues. And that's a big problem in cancer. It's called metastases. So another fundamental rule is that you don't eat your neighbor. In a multicellular organism, clearly cells trust one another. They don't treat each other as food. And as we'll see, that rule actually breaks down during programmed cell death. When cells are about to die, their neighbors actually eat them because they have changes that take place on their cell surface, on the plasma membrane, that give permission for other cells to eat them. Another rule is, of course, don't divide, don't undergo cell division without permission. That rule breaks down in cancer where cells divide when they shouldn't. Don't allow bugs inside, so don't allow viruses inside or bacteria. If cells become infected, then that's another reason why they undergo programmed cell death. And then finally, not a very scientific description, but don't act weird. So cells that are behaving abnormally, that are expressing abnormal proteins, these are also cells that are killed by a specialized cell we have in our immune system called a cytotoxic T lymphocyte or natural killer cell. And so essentially in a multicellular society, cooperation is essential. Everyone plays their role, everyone obeys the rules, and then the organism functions, quite happily. But if you break the rules, if you do any of the things I've mentioned here, and of course some other things, then typically the programmed cell death machinery will be turned on. And that's just a summary of what I've just said. So in any of these cases, when cells are injured, where they're infected, when they're behaving abnormally, or dysfunctionally, when they're breaking any of these fundamental rules, programmed cell death is typically what we see happening to eliminate the cells. Now I'll show you a quick photograph. I'll return to this later. So in the bottom here, we have a healthy normal cell. And you can see here the nucleus is stained in blue. And then the rest of the cell, in fact, we're staining the mitochondria here, are stained in green. But then when cells undergo programmed cell death, they look very, very different indeed. So the nucleus becomes very fragmented.
I'm so sorry, Professor, but are you moving the slides forward? Because we can see the first slide right now.
You can only see the first slide?
Yes.
Yes.
Okay, I am in the slide format.
Yes,
sorry.
now I can see the other side.
Now you see the other slides. I had full screen display. Okay.
Okay.
All right. Well, I'll continue. This is not the normal display, but do you see my full screen or do you just see the slide?
No, it's not full screen. But it's not okay. You can continue with like that.
Okay. Can you see the same slide or just the first slide now?
No, it's
Same
the
slide, yes.
other slides. It's not first one.
All right. So I'll just go back a little bit. Are you seeing the slides change now?
No, it's not changing now.
Okay.
Freezing now.
If you want, you can close it and share it again.
Let me just, what I'll do just to make sure, well, I'll just zoom up the slide a little and let's see. So So can you see the slides on the screen now? Hello?
Sorry?
Can you see the slides?
Yes, I can see. You don't need to share the full screen.
Okay.
I think the problem is when PowerPoint make a full screen, the slides won't play because of the Google Meet doesn't contain the PowerPoint. That's why the problem is starting. Is this okay? Maybe you can go without full screen?
Yeah. Can you see the slides changing? You can. All right. Well, I'll just go back a little bit, but this was what I was showing a few minutes ago when I was saying the body is effectively a box with the cells inside, but I'll just continue from where I was. So this is what programmed cell death looks like. You have cells and they have a very unusual appearance. So can everybody see this? Can you all see what I'm showing on the screen? Yeah?
Yeah, we
Yes,
can
we
see.
can.
Okay. All right, so the cells here on the left-hand side that I'm indicating with the arrow, hopefully you can see that. Well, that's a normal healthy cell with the big nucleus stained in blue, and then the rest of the cell is stained in green. But then the cell here at the top, where the nucleus is very condensed and it's fragmented into small pieces, and now the cell is staining red because there's a dye that binds to apoptotic cells, to cells undergoing programmed cell death. There's a dye that reveals a change in the membrane, and I'll talk about that a little later. So cells undergoing programmed cell death look very unusual, and programmed cell death enables cells to be eliminated with surgical precision. And the particular type of programmed cell death I'm going to talk about today mainly is called apoptosis. So that was first recognized really in the 1970s, and very little was known about the molecular machinery that regulates this type of cell death at that time. And as I say, over the last 25 to 30 years, now we know a lot more about this process. So again, can you see what I'm showing now on the screen?
Yes, we can see now.
Okay. I'll just zoom out a little bit. So our tissues are undergoing constant remodeling due to, of course, cell division taking place on the left-hand side here, that's constantly allowing new cells to be produced. So, for example, in your immune system, you have the neutrophil population, which every 24 hours, the neutrophil population is renewed almost completely. So neutrophils have very short half-lives of between 16 and 24 hours. So they're born, and then within about 24 hours, they die. Here you have just a little cartoon of cells undergoing programmed cell death. So in certain tissues, there's a constant throughput of cells being born and cells dying. And that really keeps the cells, or that cell population, healthy. And there's a general correspondence between the likelihood of cell injury to a tissue and the rate of cell death in the tissue. So cells on the surface of our body are epithelia. Those cells undergo programmed cell death quite a lot. Also, quite a lot of the cells in our immune system undergo cell death a lot. But then there are tissues such as the brain, where neurons live for a long time. Cardiac muscle lives for a long time. So cells in different tissues have very different lifespans. As I mentioned earlier, our bodies have 10 trillion cells, which is a lot of zeros after the 10, as you can see there. But then in our lifetime, we actually produce about 1,000 times that number of cells as tissues basically undergo replenishment in this constant cycle. So why do cells die? As I've said earlier now, cells die because they become injured, because they become infected. They malfunction because of protein misfolding or protein oxidation, or accumulation of mutations in genes that encode those proteins. Cells compete for oxygen, for nutrients, and that selects the fittest cells, and then those that don't compete successfully die. And then there are special circumstances in which cells die. For example, as I'm going to show you an example of now, during development, in many tissues, you overproduce cells, and then you eliminate the cells you don't need or you don't want, and leaving you just with the cells that you need in the final form of the tissue. So all of this is to say that cell division and cell death are very delicately balanced at all stages of life. And particularly in the adult, you have this balance between the rate of cell division and cell death that keeps us obviously at a constant size and keeps our tissues healthy through our lives. And there are cases where cell death becomes dysregulated. So for example, in cancer, which I'm going to mention throughout the talk, it is often the case that cells accumulate mutations in genes that regulate programmed cell death. And so cells don't die when they're injured and don't die when they're breaking the rules the way they normally should be. And that just refers to that there. So in cancer, you have an excess of cells that are dividing and a deficiency of cells that are dying. But in certain other diseases, such as neurodegeneration, Alzheimer's disease, Parkinson's, you have an acceleration of cell death and cells die inappropriately. And so obviously that cell population contracts. And here's an example of a dramatic enlargement of the lymph nodes. These are lymph nodes from a normal mouse. And as a result of a mutation in a single gene that regulates the lifespan of T and B cells, and that gene is called FAS, or CD95, you can see that within 4 weeks of birth, the lymph nodes of a mouse dramatically enlarge, about 50 times their normal size, as a result of a failure of programmed cell death in the T and B cell population. And now you accumulate a lot of extra T cells because the T cells are not dying the way they normally should. And these mice develop an autoimmune disease. So that's the spleen of a wild type mouse. And then again, this is the spleen of a FAS deficient mouse because again, you have an accumulation of extra lymphocytes. So that's a dramatic, I think, illustration of what can happen if cells fail to die at the rate at which they normally die. So there's lots and lots of situations now where we know that cell death is programmed. So during development, I'll show some examples of this. We have lots of programmed cell death happening during the formation of complex structures, such as the limbs, the developing neural system and immune systems to form blood vessels. You initially make a solid tube of cells, and then you eliminate the cells in the center via programmed cell death. In the adult, we have lots of ongoing cell death, as I've mentioned, and lots of tissues. And then also in response to damage or infection, programmed cell death is critical for eliminating infected or damaged cells. So programmed cell death keeps our body healthy. Just to give you one example of cell death in development. So during development, the limbs, they develop initially from little bumps, protuberances, that go larger and larger. And you essentially overproduce cells initially. And in the process of developmental sculpting, you eliminate the cells at the places where they're no longer needed to form the final form of the digits of the fingers and the toes. And that's a good example of where cells are only transiently required during development in order to make the final limb structure. And then you very precisely kill off the cells that you don't need in order to make the actual digits. And something very similar happens in our developing immune systems. About 97% of T cells die by programmed cell death during development. So only 3% of the T cells you make are actually fit for purpose. And there's a process called T cell education that happens in the thymus. And that's where all the T cell death happens in a regulated fashion during development. So I'll return also to discuss this system in detail later, but this is an embryo of a Caenorhabdus elegans, a nematode worm that's very often used to do genetic screens. And if I can just play this movie, this is the C. elegans worm developing, and you can see very quickly you go from 2 cells to 4, 8, 16, 32, and eventually 1,090 cells are born. But 131 of those cells always die during development at the same times and same places. And over on the right-hand side, there are little smooth disc-like cells that just appeared transiently. Now the worm is moving around in its egg case. Those smooth cells, maybe I'll go back a little. Those rounded cells are actually cells undergo programmed cell death. They appear very transiently. There is one there. There's another. But the point is, almost 10% of the cells that are born, in fact, more than 10% of cells that are born in the worm, die during development. They're just required transiently during development, and they always die at the same times and places. And studying the genetics of regulation of programmed cell death in the worm has been really important for identifying some of the genes involved. And as we'll see, the same essential genetic framework is conserved to ourselves. So I'm going to go through now some of the really the machinery of how cells die. As I already answered this question. In most cases, cells do not die by accident. They die by design. Cells express all of the proteins necessary to kill themselves. And cells are constantly monitoring the health of the cell, and they switch on their own programmed cell death machinery if there's a problem. And if they can't do that, some viruses, for example, bring in inhibitors of programmed cell death into the cell to try and prevent the cell from doing that. Then there's even a specialized type of cell that will execute the cell by switching on its programmed cell death machinery. And I'll discuss that also later in the talk. So there's three basic types of cell death that I'm going to cover. Cell murder is essentially necrosis, where the cell doesn't choose to die or participate in its own death, but it has death imposed upon it just by an overwhelming injury. And that does happen sometimes in the body. So for example, a burn injury, or if you are involved in a serious accident, if you were maybe hit by a car, well, the type of cell that happens when you get a very severe injury that's very sudden is typically necrosis. And as I'm going to tell you, that it really is not a very desirable mode of cell death to happen in the body for lots of reasons. And I'll explain more about that later. So the type of programmed cell death is more like a cellular suicide. So apoptosis, which is the major form of programmed cell death in the body. In this case, the cell is actually an active participant in its own death. It senses that there's something wrong, and the cell participates in the decision to die and actually encodes a set of proteins. So there are genes that encodes proteins that whose sole function it is to kill the cell at the right time. And then there's a type of cellular execution. There are cytotoxic T cells and natural killer cells in our immune system, and they switch on the programmed cell death machinery in cells if the cell is behaving abnormally and doesn't switch on its own programmed cell death. So these are, as we'll see, somewhat similar. Cells die in all cases, but very different in terms of the way the process looks and the outcome of the process. So the most desirable endpoint from the body's point of view is programmed cell death, because it's a much friendlier form of cell death, and it does much less damage, and it doesn't activate the immune system, as we'll see. Now, I use this joke to illustrate this. So this person's not dead, they're meditating. It's an advanced form of meditation involving putting yourself in this position for long periods of time. And I like to think of this as apoptosis. It's highly controlled. It's good for you. This person has ended up in a similar position, but the upstream stimuli are very different, and this is a less healthy way to end up. So that would be more akin to necrosis. So it does actually matter how cells die, because in both cases, this is a cell undergo programmed cell death, and so is this. But as we'll see, when cells die by programmed cell death, they're recognized by the body's tissues before they can burst open and before they can cause damage and disturbance to other cells in the vicinity. When cells die by necrosis, they release all sorts of nasty toxic enzymes that causes further cell death and also activates the immune system, because when cells rupture by necrosis, they release cytokines, which have been called DAMPs, damage-associated molecular patterns. And these cytokines then activate the immune system, and they cause inflammation. And inflammation is not a good thing if it happens a lot. So DAMPs are released by necrotic cells, and then they activate further cytokines, and you get strong inflammation. And really, a lot about apoptosis is to avoid the release of DAMPs, to avoid activation of the immune system unnecessarily, because we have apoptosis happening in all our tissues all the time, and our tissues would be in a constant state of inflammation if we were constantly releasing DAMPs and activating our immune systems. So the key DAMPs that our bodies express are members of the interleukin-1 family. There are 7 of these cytokines: interleukin-1 alpha and beta, IL-18, IL-33, and then there are 3 IL-36 cytokines. And these are molecules that are not normally released from healthy cells. They're kept within cells, and when cells burst open, they release the IL-1 family cytokines, and then they're very good activators of inflammation in lots of different tissues. So that's one of the major reasons for having a programmed cell death machinery, is to avoid this inflammatory response that you get to necrotic cells and enable the tissues to remove the cells when they die, quite silently without involving the immune system. I'll skip that. So chronic inflammation is a big problem in many diseases. In cancer, for example, most tumors are known now to have a major inflammatory component that helps the tumor to progress and become more aggressive. But lots of diseases, diabetes, cardiovascular disease, arthritis, and of course autoimmune diseases all have a major inflammatory component. And we now know that the failure to regulate cell death properly seems to be a driver in some of these diseases. So I'm going to look now at the regulation of programmed cell death. What is this molecular machinery that I'm talking about? So again, just to revisit necrosis for a moment, to contrast this with apoptosis, the things that cause cells to die by necrosis are severe traumatic injuries, really very high concentrations of chemicals or poisons, extremes of heat or cold. So obviously a burn injury, as I mentioned earlier, causes necrosis, high dose radiation, and percussion injuries, injuries involving a lot of force. And what typically happens during necrosis is it happens very rapidly. Cells become injured, they detach from the substratum that they're living on very quickly, they swell, and then they rupture, releasing their contents. And this is what necrosis looks like. Again, this is a movie that we made in the lab. These are cells exposed to hydrogen peroxide. And this movie is over 1 hour duration. And you can see that hopefully the cells are swelling, they're producing these blebs, and the blebs burst open and release the contents. And everything is over pretty quickly. Like I said, this all takes place within an hour. And if this happened in the body, it would cause a lot of inflammation, and it would cause a lot of collateral damage to other tissues, because cells contain lots of nasty enzymes, proteases, DNases, lipases, carbohydrases, that if released into the extracellular space, will cause death to cells that weren't injured in the first instance. I like again to illustrate this with a movie, made a couple of years ago when I was at a conference. Let me see if this will play. Whoops. It zoomed up a little, so I'll have to zoom it down. These are some members of my lab. So this is what necrosis looks like. The cell is having a good time, and then suddenly something really terrible happens, and there's really no chance to avoid that. Now, in apoptosis, it's very different. The cell participates in the decision to die. The cell senses that it's injured or stressed or damaged in some way, Basically, very mild injuries can cause a cell to turn on this programmed cell death machinery, and or detection of infection. And in some cases, there are natural cytokine-like molecules, members of the TNF family, that don't injure a cell at all, but they just turn on the cell's programmed cell death machinery. And in apoptosis, what happens, cells round up, but then they condense, they produce lots of blebs. The chromatin becomes hypercondensed. The nucleus breaks up into lots and lots of small fragments. And then the cell actually collapses into these small little vesicles called apoptotic bodies. And critically, neighboring cells and macrophages recognize the cell as being, as in trouble, undergoing programmed cell death. And they can engulf that cell before it has an opportunity to rupture and release its contents. So in apoptosis, you have a very different outcome. The cell packages itself for disposal, and then it's simply removed by its neighbors. The neighbors are given permission to eat the cell before the cell can burst open. And this is what apoptotic cells look like. Hypercondensed chromatin. You can see blebs starting to form here, but the cell has not ruptured or released anything yet. And this is another example of a scanning electron microscope image. All these tiny little vesicles that are being produced, these are normal healthy cells surrounding this apoptotic cell. So during this time, membrane alterations happen, and one of the major ones is the exposure of a lipid called phosphatidylserine that's not normally present on the surface of healthy cells, but apoptotic cells expose this, and neighboring cells or macrophages recognize the phosphatidylserine and use that as a means of gaining permission to eat the cell. So this is what the process looks like. Again, a movie made in my lab. These are cells treated with donirubicin. And this movie is shot over 24 hours. So this cell has started to undergo the process of apoptosis here on the left-hand side. And its neighbors are all still very healthy. And at this point already, the cell would be eaten by a macrophage if there was one here in the culture. But cells stay in this process, but they're still alive, but they're displaying all these blebs, and they've entered apoptosis. And here you have a cell, and another one that's completely undisturbed by the death of these cells that started many hours ago. And then finally, this cell, many hours after the other cell started to die, this cell now starts to die. These cells were treated with a chemotherapeutic drug, donirubicin, that is widely used to treat cancer patients. And most chemotherapeutic drugs that are in use actually cause programmed cell death, apoptosis, to happen in the cells. So just to remind you again, as I've said, typically the endpoint of apoptosis is neighboring cells or macrophages recognize the changes on the apoptotic cell, and they selectively remove that cell so before it bursts open and can activate inflammation. And as I mentioned a few minutes ago, the change that happens on the surface of the membrane is phosphatidylserine, which is normally confined inside the cell, is flipped on the membrane and externalized on the outer surface. And we can detect this using a protein called an Exin-5. We can make this fluorescent, so we can monitor this change taking place in cells. And this is an example. We've stained cells here for an Exin-5, which has a fluorescent dye added to it. And normally, all of the cell populations are negative for an Exin-5. This is when they become positive. So when we trigger apoptosis, we can see now a lot of cells move over here into the positive quadrant, and progressively more cells stain with an Exin-5. So they're externalizing phosphatidylserine as they undergo apoptosis. And here's a movie of this process. An Exin-5 is here stained in red. You can't see it at the beginning. The cell is still healthy. The green stain is cytochrome C within the cell. And then we've also added to the medium a dye called DAPI, which gives a blue stain when it enters the cell, and it will stain the DNA. And so as the cell undergoes programmed cell death, it's still healthy here. And you're going to see the green cells suddenly become diffuse. Now it's rounding up and blabbing, and now the annexin 5 dye is binding, and you can see then the nuclear fragments within the cell. So annexin 5 enables us to identify very specifically apoptotic cells due to the phosphatidylserine externalization. And so that's really the fundamental difference between apoptosis and necrosis. In necrosis, you have rupture of cells, release of DAMPs, and then those DAMPs activate macrophages, and you get cytokine produced, and you get an inflammatory response. In apoptosis, you get phosphatidylserine externalized, the cell packages itself, and then it breaks up into apoptotic bodies and is recognized by macrophages at an early stage. And you get no inflammation, and even an anti-inflammatory response can arise. Now, there's a slightly different type of apoptosis that happens when the cell cannot turn on its own cell death machinery. And this is in the case of, for example, when a cell is infected with a virus, and the virus might be encoding something to block the turning on of the cell death machinery. And in this case, we have natural killer cells or cytotoxic T cells, and they can deliver a signal to the cell either in the form of Fas ligand or TNF ligand, or they can introduce molecules into the target cell to turn on its cell death machinery. And I'll give you an illustration of this. These again, so these are cancer cells in the plate, the flat cells, and then the more rounded cells are natural killer cells that we've added to the culture. And you're going to see that the more rounded cells are moving around a bit. The natural killer cells can patrol and look for cells that they don't like. And what's going to happen here is this cell is going to attack this cell here, and it's going to turn on its programmed cell death machinery. So it's degranulating now, and it's delivering cytotoxic granules into the cell that's going to turn on the programmed cell death machinery in the target cell. And finally, this cell then dies by apoptosis. And that happens in the tissues in our bodies when cells are infected or they're making something that, maybe is suggesting to the immune system that they're cancerous. So what is the molecular machinery that governs all this? Are there genes that regulate apoptosis? What do we know about the understanding of this process? Well, a lot of work was done actually about 30 years ago, starting in the lab of Sidney Brenner in Cambridge, and then a couple of his postdocs, most notably Bob Horvitz, continue that work, working on the nematode worm C. elegans. It's a model organism. It has about 1,000 cells. And they noticed when they started studying the development of this worm that about 10% of the worm cells died by programmed cell death. As I showed earlier now, during development, they traced the birth and death of every cell during development, and they found that 131 cells died during the development of this worm. And they decided to do mutagenesis studies, do genetics, to see if they could inactivate the genes that controlled cell death. And they did that successfully, and the Horvitz lab published a series of papers showing that they could identify mutant worms where the programmed cell death in the worm was disrupted. And so the older developmental cell death was either blocked or accelerated. And this is just comparing a wild type worm here with one of the mutant worms. And at the end, we're going to see that this mutant embryo basically has cells that should have died, but they didn't die. And so these are the extra cells here that essentially persist in a mutant worm. And they identified lots of different mutants. And after a lot of experimentation, they identified this genetic framework where a gene they called SED3, cell death defective 3, another gene called SED4, they promoted programmed cell death. And then there was a gene called SED9 that blocked cell death, and a gene called EGL1 or Egling deficient 1. And that accelerated cell death by inhibiting SED9. Now we now know that Programmed cell death regulation in humans and other mammals is very similar. These are the SED3-like genes, the caspases. This is the SED4-like gene called APAF1. And then the SED9-like family is the BCL2 family, and the EGL1 family is the BH3-only proteins. So worm programmed cell death is regulated by homologs in humans that are very similar to the worm. Now, it's much more complicated in humans. And in fact, now that it's all been worked out in a fair degree of detail, this is actually the molecular machinery. I won't go into it in any detail, but we know there are 3 major routes to activating this machinery through either receptors on the cell surface that are called death receptor. I mentioned one of them earlier, FAS. There are other members of this family called a TNF receptor or a trail receptor. Cytotoxic T cells can switch on the machinery by delivering proteins into the cell, most notably granzyme B. And then we have an intrinsic way of monitoring the health of the cell. So that intrinsic pathway is called the BH3-only driven pathway. And that acts by opening pores in the mitochondria. And all 3 pathways converge on the caspases that are a family of proteases that when they're activated, which is what happens during programmed cell death, they effectively coordinate dismantling of the cell. All the caspases are switched on and they cleave lots of different proteins in the cell. And a critical checkpoint in mammalian programmed cell death is the BCL2 family. And many tumors overexpress members of the BCL2 gene family, such as BCL2 itself or BCLX. And if that happens, then the programmed cell death machinery can be kept off. So many tumors have elevated levels of BCL2 family proteins, and they block the activation of the cell death machinery in mammals. And this mitochondrial checkpoint has emerged as being really a critical step in the activation of the programmed cell death machinery in ourselves and in other mammals. Now, when caspases, which are proteases, are switched on to coordinate apoptosis, they cleave hundreds of different proteins, and that has a result of activating nucleases, for example, or dismantling key cellular structures or deregulating the activity of certain family of kinases and other proteins. And so caspases, they, as I say, they effectively dismantle the cellular architecture during apoptosis, and they ensure that the membrane changes take place that enable our bodies to recognize cells as dying. And we've done some proteomics on this, and each dot here represents a protein, and the circles here are proteins that are degraded. They get cleaved by caspases, and the squares here are the degradation products. When you compare healthy cells and apothetic cells, there's many, many proteins that get cleaved by caspases. And my lab and other labs have studied this for some time, and we now have a fairly good picture of what happens during apoptosis when caspases get switched on. Some of the proteins they cleave, and you know what the consequence that are. What consequences does this lead to? These are not caspases, obviously, but a friend of mine left their 2 children alone with a can of paint when they answered the phone call, and when they came back, the children had very helpfully redecorated the sitting room. So this is kind of what happens when caspases are switched on. They cause a lot of mayhem in the cell that results in programmed cell death. But again, I want to remind you that one of the major purposes of caspase activation, really, apart from killing the cell, is to ensure that the death that arises is highly regulated and is non-inflammatory. So you avoid inflammation and you minimize disturbance to the tissue. And again, that's a slide I've shown earlier. So apoptosis is really as much about avoiding activation of the immune system as it is about killing cells. So what we're currently working on is something that really is related to cell death. it's actually inflammation. And normally things that kill cells, such as chemotherapeutic drugs, ideally, you want to get those drugs switching on the cell death machinery. In cancer therapy, the major goal is to kill cells. But you don't always successfully kill them, of course. That's a major problem in cancer therapy, that cells resist the drugs that are being used to treat the cancer. And unfortunately, one side aspect of that is when you try to kill a cell, you stress the cell. And if you don't successfully kill the cell mounts a stress response. And we and others have noticed over the last few years that when you stress cells, they make inflammatory responses if they don't die. And those inflammatory responses can actually assist the tumor in becoming more aggressive and in surviving. So we've called this type of inflammation stress-associated inflammation. Very little is known about how it works. We know very well how infection and injury drives inflammation through so-called pathogen-associated molecular patterns in the context of infection. We know how they're sensed by a range of receptors, such as toll-like receptors. We know how necrosis causes inflammation through releasing DAMPs, and they're sensed by IL-1 receptors. But we really don't know how cell stress causes inflammation. And this is very important in the context of tumor-associated inflammation. Tumors, as I mentioned earlier, they build up an inflammatory environment around them. And that helps the tumor very often by turning on what's called a wound-healing immune response. But how tumors drive this stress-associated inflammation really isn't known. So there's still a lot to discover. This is actually a photograph taken from the Hubble telescope. It's a globular star cluster. It's very complicated. Obviously, we know that deep space is extremely complex. And this is something quite similar, but it's not deep space. It's a high resolution microscopy image of a cell. You see all of these protein clusters. All of these protein clusters are obviously doing something different. So cells really are immensely complex, and there's still a lot to discover. There's still a lot to learn. And this is a lower resolution picture as you zoom out. And so this is sometimes, it feels like what we do. We're still, I think, we have a relatively crude understanding exactly how cells work. So there's still a lot of interesting problems out there to solve. I'll just finish up by saying, because sometimes I'm asked this, why I got involved in research. I got involved in research because when I started working in a lab as an undergraduate and then as a postgraduate, I really loved being in the lab, being at the bench. I felt like I was seeing very often things, you know, that nobody else had seen before. It can be very exciting, rewarding to work in a research environment. And there's a saying that if you find something that you love to do, you'll never work another day in your life. And that is very true. Research, certainly for me, has been extremely rewarding and exciting career and very challenging. You're constantly learning. No 2 days are the same. And it's like, in a way, having a hobby as a job. So, of course, the other reason to do research is because we want to cure disease. In order to do that, we have to understand how cells work. And, you know, we're still quite far away from obviously understanding how cells work. But of course, there's been many breakthroughs that enabled, you know, lots of diseases, including cancer, including lots of autoimmune diseases, to be effectively cured or treated much better than we were able to treat them previously. So I'll finish up at that and acknowledge the people, of course, who actually do the work in my lab. These are people who, this was the group a few years ago now. It's changed a bit since then. But I've had a group of amazing PhD students and postdocs over the years. And I'd just like to thank them for their excellent efforts and basically highlight their work. And then finally, if you're interested in immunology, I write a textbook called Essential Immunology, and the 13th edition is available at the moment. And I'll finish with that, and thank you.
Thank you, Professor. It was really inspiring and excellent presentation. And if you wish, we can start the questions that our audience asked.
Yep, no problem.
YouTube chat. And before the start, we have questions that we want to ask you.
Okay.
And first of all, I want to ask you, we know that you have an international reputation in the field of programmed cell death. And how was your process of determining your field of study, and what were the research methods in this process?
Well, my process was, first of all, as an undergraduate, I did a research project related to fish development, related to understanding how we could identify different species of fish, believe it or not, because I'm a fisherman. I like fishing, and I was fishing yesterday, and so it's my hobby, and I take it very seriously. And I knew that from my own, experience with fishing, that it was quite difficult to identify certain species of fish without killing them, and we like to put the fish back alive. So I did a project when I was in my final year undergraduate to use a method, isoelectric focusing, to try to identify the fish species using taking a tiny sample and using that method in order to identify the different species of fish based on their protein composition. And during that project, I just realized I really loved working in a lab. And I just loved everything about the process. And then I decided I wanted to do a PhD. And I started working on programmed cell death during my PhD accidentally, because my PhD was about looking at differentiation of tumor cells after treatment with retinoic acid. That was a therapy that was being developed at the time. And when I was working in this a few months, I noticed that during the process of differentiation, there were cells that looked weird. And it turned out they were cells dying by programmed cell death. And so I started reading all I could learn about that. And there wasn't a lot known about that process at the time. And I was really fascinated by this. And then I started, my PhD turned into working on programmed cell death. And because it was a lot to discover, we made a lot of, interesting discoveries, and I published quite a few papers during my PhD. And then I went to London to study as a postdoc with a well-known immunologist, Ivan Royce. And at that time, the HIV pandemic was had just started. And so I worked on HIV-induced cell death. For my first postdoc, I learned a lot of immunology there. And then I went to California for my second postdoc. And I had an amazing time while I was training. And then I came back to Ireland to join Trinity College in Dublin, and where I've been ever since. So that's been my journey. I didn't choose to work on programmed cell death. I think it chose me. So.
Yeah, it is really good to learn about your travels and hobbies, actually. And here is another question. I know you are a board of member of the European Cell Death Organization. How does it feel to have an experience like that, and are there any difficulties with it?
Well, I've been an ECTO board member for quite a long time now. ECTO runs a meeting every year, and it's a group of scientists all interested in programmed cell death, obviously. It's interesting to be involved in running a meeting, and you know, there's obviously it's always a bit of negotiation about choosing the meeting program and meeting venues and raising funding for the meetings. So it's an interesting experience, and it's good to meet with colleagues. There are members on the board from Italy, from Spain, from the UK, from Belgium, Switzerland, you know, everywhere essentially. And it's good because it exposes you also to, you make friends and colleagues in different countries. And of course, the board meets usually once a year, and we travel to different countries in Europe and get to see, you know, how everybody lives and works, and different foods and different cultures. So yeah, it's an interesting experience for sure.
It sounds really good to have an experience like that, and I actually want to live a life like that. It was my dream, actually.
Well, of course, a lot of this has changed during the pandemic in the last 2 years. So yeah. My life used to be much more interesting, traveling almost every second week to conferences and, you know, well, to give talks in various places. But we're getting back there again now, thankfully. Conferences are starting to happen again, but the last 2 years have been, there's been a bit of sensory deprivation compared to before. Yeah.
Okay, thank you so much.
Can I just add, of course, it's not all fun and travel. All of this depends on, you know, working hard, being really good at what you do. You get to do some of these things if you, of course, excel in your field. So I should add this because, of course, it is great fun being a scientist, but you don't get invited to much unless, you know, you do good work, right? So there is that, you know, there is that aspect that I do need to emphasize. I tell my own students that you get lots of invitations to speak if people want to hear what you have to say, and they will want to hear what you have to say if you show them that, you know, you've done some interesting work. So it's not all about going to, you know, nice places and, you know, nice meals and so on. There's also, of course, as you know, the hard work has to be done in the lab as well. And when I was a PhD student and a postdoc, I worked very hard, but it was enjoyable work, you know. I loved being in the lab, and then these things happen to you as a reward, if you like, for your hard work. So I do have to say this because, you know, it's easy to look at the kind of the red carpet and say, oh, I want to be on the red carpet. You get there for doing the thing that, you know, makes you recognized. So don't forget to work hard as well.
Thank you for your sharing your experiences as well. And here is another question.
Another question from Özlem Baydar. What is the most suitable cell type for these studies? Which cells do you use in your research?
What is the most, can you repeat the first?
Suitable cell.
Well, really it depends. There's lots of different cell types you can work on. And, you know, a lot of labs have their favorite, you know, model cell lines that they work with. It really depends on what you want to do. So for example, if you want to do a lot of knockdown studies where you're knocking down expression of specific genes, or you're knocking out genes by CRISPR, there are certain cell lines that are much easier to use CRISPR to knock out in or knock down. HeLa cells are very good in that respect. But if you're interested in the immune system, you may want to work on a transformed T cell line or on primary T cells. So it really depends on, you know, what's the problem, what's the approach you want to use in order to address the problem. It's a bit like sport, you know. There's lots of different sports, and if you're playing tennis, you need a tennis racket, you know. If you're playing hockey, you need a hockey stick. So with research, there are different problems you're addressing and different ways you approach addressing those problems. So there's no best cell line, and there's no best system. So if you want to do genetics, even if you want to understand how something works in humans, it may be much better to start using a nematode or a fly in order to do the genetics that you can't really do in human cells. So.
Thank you so much, Professor. Another question from Ufuk Shen. Are there any technologies that are used to force cancer cell for apoptosis, or is it just another daily work for immune system not called as apoptosis?
Are there any technologies to force cancer cells into apoptosis? Well, all of the existing chemotherapeutic drugs we use actually kill by apoptosis. It's just we need them to work more efficiently. And there are now some new targeted therapies based on our knowledge of the cell death machinery that mimic proteins within the cell death machinery. So towards the end of the talk, I mentioned BH3 only proteins. They're critical for driving apoptosis in one of the pathways I mentioned. So BH3 only proteins are proteins expressed by our own cells to turn on programmed cell death. And now there are compounds that have been designed to mimic those proteins, and they're called BH3 mimetics. And there's a series of these that are in clinical trials, but one of them has been approved about 5 years ago called venetoclax. And that is a new type of chemotherapeutic agent that switches on the programmed cell death machinery very precisely. And venetoclax is used to treat, I think, CLL, chronic lymphocytic leukemia, and other malignancies. So there are therapies that have been developed specifically to turn on the programmed cell death machinery and improve the switching on of apoptosis using the more conventional chemotherapies. Yes.
Thank you so much, Professor. It's up to your Kübra.
What are the internship opportunities at your university or other universities in your country? Can you share information about this?
About internship opportunities? Well, of course, I get a lot of applications for internships, and that really depends, I guess, on some universities have internship programs, but not so many really. You have to remember, it's actually, of course, it's great to take in an intern in the lab if they're good students and, you know, they learn fast and, you know, they work hard and so on and they're no trouble. But it's identifying students really who realize, you know, who first of all are going to be easy for the lab, you know, to host. And a lot of students I find, they ask for an internship by telling me how great it would be for them to visit my country, visit my university, and, you know, have this great experience. Well, I know all that. Of course, it would be great for the students to do that. But you need to tell somebody, why should they choose you? What's so great about you? You need to sell yourself. So students often write looking for an internship, or in fact, indeed, looking for a PhD position. And they often, in their letter, tell me nothing about why them. I get an internship application maybe every second day. And so every year, most labs would get maybe hundreds of applications for internships, and they can maybe take one student. So you need to sell yourself. You need to say what skills, I don't mean experimental skills, but what qualities, you know, you would bring to the lab. You know, are you creative? Are you, you know, hardworking, ambitious? You know, why you? So there are internship positions available, but most of them are kind of ad hoc, informal. They're not programs that are organized, but you have to persuade a lab to give you an opportunity to visit for maybe a couple of months. And they need to know why would they give you that opportunity, right? So I think it's very important that when you're writing that letter, you need to make it very clear. Why are you such a great student? What are your grades? Give examples of things you've done during your schooling or university education that shows that you're a fantastic student and you deserve that opportunity.
Okay, thank you for answering. I have a question. What can be done when the programmed cell death process is interrupted?
So that's what we're really, working out. So what can be done is to identify what the problem is, where the interruption is. And in many cases, as I mentioned in the talk, the interruption is often one of the BCL2 family proteins, BCL2 or BCLX, MCL1. Very often, the levels of that protein is elevated. And in those cases, molecules, as I've mentioned now earlier, BH3 mimetics can be designed to neutralize the elevated levels of those proteins and overcome this interruption. And so that's where most of the focus is. There are other checkpoints such as IAP proteins, inhibitor of apoptosis proteins, and those proteins, can also be neutralized with IAP antagonists. And then there are other ways, such as manipulating the molecules that normally would switch on the cell death machinery, such as TNF. And for example, TNF inhibitors, antibodies directed against TNF, have been very successful in the treatment of many immune diseases. So there's lots of things that can be done.
Thank you, Professor, for your answers. And here's another question from Yasemin Albayrak. Thank you for your wonderful presentation. I get most of my questions in my mind, but I wonder about your current researches. How about the bad parts? If cells affected with the best stresses, are you able to anticipate and prepare for what might happen?
If cells are, sorry, I missed a little bit of the question. If cells are affected by stresses, is it?
Yes. If cells affected with the best stresses, are you able to anticipate and prepare for what might happen?
Well, you are, for example, if the stress is caused by chemotherapy, you can anticipate it because you know you're going to deliver the stress, you know you're going to deliver the chemotherapy. And if you want the best outcome, what you want to try and do is minimize the stress-induced inflammation and maximize the cell death that occurs in chemotherapy. So we're trying to understand how cells really, after perceiving stress, we're trying to understand how they make cytokines. What are the pathways involved there? How do cells sense stress? And that's not a simple problem. We've been making some progress on this. We published a paper a year ago in Developmental Cell describing one pathway that leads from stress induced by the chemotherapeutic drug paclitaxel, or Taxol, as it's called sometimes. So Taxol is used to treat breast cancer. It's first-line chemotherapy to treat breast cancer. So millions of women are treated with Taxol every year. But that drug can cause this type of stress-induced inflammation. And so we can anticipate that this happens. And we can then devise strategies of minimizing that if we identify the pathway involved. So we know that a receptor on the cell called TRAIL is critically important in basically driving the Taxol-induced inflammation. So that's where our work is going right now. We're we're trying to understand the sensing pathways for stress and how they are connected to inflammation. And so there's lots of challenges there.
Yeah, it was really understood. Thank you. And another question from Ahmet Furkan. Is it possible to stop the cellular death processes after the cell has already started to being death or while deathing?
Not really. Once the mitochondrial checkpoint, the back channel, has been opened, a cell is really committed to death then. So it's quite difficult to interrupt the process beyond a certain point, but you can block the process if it's upstream of that. And so, you know, there are, for example, within the death receptor pathway, the extrinsic pathway, we know several points in the pathway that can be targeted and can be stopped. But most of the time, in most of the diseases, actually, we're trying to enhance cell death rather than block it. Of course, the exception would be neurodegenerative disease. And, you know, really, I think that the focus there is more about minimizing the causes of cell death, which is very often toxic protein aggregates. They're trying to minimize the accumulation of those protein aggregates, and rather than blocking cell death per se. So most of the time, as I say, in cancer and autoimmune disease and other forms of immune-related conditions, we're trying to enhance the cell death rather than block it.
So that it looks impossible to stop the.
Well, it's not entirely impossible, but it depends the point in the pathway. Once mitochondria are involved, and it's downstream of mitochondria, then it's very difficult to block it. But above mitochondria, then it's maybe possible, yes.
Yeah, okay, I understand. Thank you.
Thanks for information reply. Another question from Feyza Keskintaş. What do you plan to build your next career on? Will you continue on this research topic, or do you have another topic in mind?
I think well, I work on another problem in parallel, and I'm interested in also members of the interleukin-1 cytokine family, interested in how they're activated by proteases. And so that's a research line that my lab has worked on as well in parallel for the last 15 years. And we're very interested in how IL-1 family cytokines become activated and inactivated in the context of the immune response. But that's quite enough, actually. So I don't plan. I think there's lots of problems to solve still in the areas that I'm in. And also, I'm editor-in-chief of a journal called the FEBS Journal. That keeps me very busy. We have, you know, about 100 manuscripts submitted to the journal every week. And so only about 5%, maybe 10% of manuscripts get accepted at the journal. And I lecture in the university in Trinity College. And also I write a textbook, Essential Immunology. And I like to go fishing. So I have plenty to keep me busy.
Thank you so much, Professor. Another question from Sude Uğur. Firstly, thank you for your informative presentation. When unprogrammed cell death is prevented, does the damage caused by disease like Alzheimer's be permanent or can be cured?
I think, you know, there's possibilities to I hesitate to use the word cure, but certainly, of course, there are cures for certain diseases. There's progress taking place in these conditions all the time. And I think certainly the damage can be ameliorated, so minimized. And, you know, so there's very interesting work going on in these neurodegenerative diseases. And as I say, there's, I think, some interesting work going on to minimize the production of the, you know, the fibrils and amyloids that are produced in these diseases in order to minimize the damage that takes place. So I think I would be optimistic that yes, you know, these diseases will be converted to milder forms, you know, when we know a lot more. But I think it's complicated, and it's going to take probably another decade or more to make major therapeutic breakthroughs in these areas. Neurodegenerative disease has always been really a difficult problem to crack. But, you know, I've witnessed, for example, when I was a postdoc, I worked on HIV infection, and at that time, HIV was just a new virus, and it was invariably a death sentence. People were dying within 2 to 3 years of HIV infection when I was a postdoc in London. And I've seen that disease within a space of 15 years go from being a fatal illness to being essentially just a chronic condition by identifying, you know, HIV protease inhibitors that enable people to live for decades, you know, infected. And obviously we've come through a pandemic that started off having absolutely no treatment options available. And here we are now within 2 years of, you know, this pandemic breaking out. And we have, you know, 4 approved vaccines, 3 of them, or 2 of them, which use extremely new technology, you know, RNA. One of them, or actually we've more than 4 approved vaccines now, maybe 6 or 7 approved vaccines. One based on recombinant protein, which is just the Novavax vaccine that was just recently approved. 2 based on adenovirus, and 2 based on RNA molecules. So the point is, we're making progress all the time, and that's a great thing. And so I would be optimistic about cures for, or at least much better treatments for neurodegenerative diseases in the next decade.
Thank you for answering. Another question from Fatma. Thanks for sharing information about this interesting topic. I have understood cells die in injury or infections, but I couldn't understand the logic of cell dying when organism is developing.
Yeah, well, there's a lot of logic actually, because you know, you can't build a complicated organ just by growing cells in very specific places. It's like a process of sculpture. You make, first of all, a less complicated organ, and then you delicately remove things you don't need. And I'll give you another example of how you know, this makes sense. So in our immune system, our body cannot anticipate what are all of the foreign things that the immune system needs to recognize. So how the immune system works is your body makes receptors by random reconfiguration of genes. So it makes T cell receptors and B cell receptors by random reconfiguration of the T cell receptor and B cell receptor genes. So you make tens of millions of different T and B cell receptors. But because they're randomly reconfigured, many of them don't work properly. And you can't anticipate that. And so, and also many of them are autoreactive. So you make all of these T cells by random reconfiguration. And then you select just the cells that work. And most of them don't work. So, but how can you make such a complicated system in advance? And the brain works somewhat similar. Here's another, so with brains, neurons have to innervate, they have to make synapses to connect and, you know, to educate themselves and retain memories and functions. And what again happens is you have many neurons and just specific subsets make the right connections. And that's why, for example, some of us are very good at remembering things and some of us are not. And some of us know a lot and some of us don't know a lot. And because our neural networks, you know, are making contacts in a kind of in a Darwinian way, you know, based on you know, essentially a competition for the fittest cells, for the best cells. And then the ones that fail to make contact, they die because they're not innervating anything. And, you know, it's that's the way it works. There's clearly no better way to set up that system because that's the way our bodies have evolved, have developed, you know. I certainly don't know a way that I could predict all of the organisms that I might encounter in my lifetime, except through a random reconfiguration of immune receptors, so that I make an infinite number of receptors based on probability. But then if I'm going to do that, many of the receptors won't work properly because they're randomly reconfigured. And so in the immune system, you make lots of extra T cells, but many of them don't work, so you get rid of the ones that don't work. That's the way it works. It makes sense to me anyway.
I want to ask one question too. The studies you are involved in are quite a lot, and this is very impressive. How can you make time for all this? Really, Professor, I want to learn your secret. Please share with me.
How do you make time for work hard? Work hard, but also I think you need to focus. I think you need to decide what you're really going to put your effort into, and then become good at that. And after a while, things kind of get easy, you know, because it's like if you look in the cockpit of a large, you know, Airbus or, you know, Boeing plane, you know, and you look in the cockpit where the pilot sits, and there's all these switches and controls, and it looks very complicated. But when you're an experienced pilot, you know where all the switches are and what they do, how they work, when you turn them on and off. So it's easy. But when you're training, of course, you have to be dedicated and focused to learn how everything works. So, and I think the secret is, as I say, find something that you really enjoy and then really dedicate yourself to that. You know, if you want to be a great pianist, you have to practice many hours every day and focus, you know. And then after a certain amount of time, it becomes very easy to play the piano. But it doesn't start off easy.
Okay. Thank you so much, Professor. It was a great honor to meet you.
Yes, thank you for this wonderful and informative presentation. And it was definitely an interesting topic, and it was an honor to see you at our event, Professor. Thank you so much.
Thank you. Thanks for the invitation.
And next session will start at 2:30 p.m.
Okay. Bye, everybody.
See you. Sir.
See you, Professor.
Okay.
Thank you so much again.
Thank you so much.
All right. Thank you. Bye-bye. Bye.
Hello everyone. This is our fourth session today. We're really excited for this session again. I'm going to leave the word to Şeyda. How are you, Şeyda?
I'm fine, thank you, Ezgi.
I'm fine too. Have a nice session. Good luck.
Thank you.
Hello, Professor Malarias. And dear participants, I'm Feyza Keskintaş. I'm a member of IEEE GTÜ Technical University, EMB Society, and I'm a first grade student of molecular biology and genetics. And I leave the word Şeyda.
Thank you, Feyza. Hello, dear participants and Professor Malarias. I'm Şeyda Ayarcı. I'm a second grade student of molecular biology and genetics. And I leave the word again, Feyza. But before that, I wish you all a good session.
Today, this session's guest is Professor George Malarias, whose field is electrical engineering. But for this session, he presented us the technology for bioelectronic medicine as subject. I leave the word to you, Professor. I wish a good presentation.
Thank you. Thank you very much for the kind invitation. As I was saying earlier, when we were chatting just before the session started, there is no bigger honor and bigger joy for an academic to be invited by a student society. So it's a pleasure to be with you. I'm coming from Cambridge. It's a special day today because the sun is shining. This doesn't happen very often here. So it's great to be with you virtually. So let me share my screen. Yes, give me 1 second. That's what I want. Yes. All right. So can you see my screen?
Yes, we see.
Excellent. I'll go and have a pointer. Yeah, here. Can you see the pointer?
Yes, we see.
Great. Wonderful. So let's start. I put a title that is very generic, Technology for Bioelectronic Medicine. I will explain what bioelectronic medicine is and why it needs new technology, and what we as electrical engineers or as engineers, physicists, scientists, whatever, can do to help in this topic. So I'll go for an overview of research in this discipline, and then I will give you some examples from work in our lab. So let's see what medicine is. And this is the engineer's view of medicine. So we start our life and then we progress through life towards death, the length of the journey being determined mostly by our genetic makeup. However, there are factors that can accelerate this journey. A poor lifestyle can send people to an early checkout. Pathogens with the COVID pandemic, very much a contemporary topic, can also shorten our journey on this planet. Accidents, the same thing. In this context, medicine is a decelerator. It tries to turn back time and give us more time on this life and better quality of life. How does medicine do that? What are the modes of intervention? There are the classical ones, such as, for example, pharmaceuticals. You go to a doctor, you feel sick, they will prescribe a pharmaceutical. This can be done for a variety of reasons. Can be done for prevention, for example, vaccines. Again, very much a contemporary topic with the COVID pandemic. You get a vaccine to prevent you from getting sick. It can be done to control the symptoms of a disease. For example, dopamine is given to people who suffer from Parkinson's disease. It doesn't cure the disease, but it treats the symptoms. So it stops the tremor, it helps with walking, all that. Pharmaceuticals can also have curative outcomes. For example, antibiotics. You take antibiotics, they kill the bacteria that cause the disease, and the patient is cured. Another mode of intervention, which is classical, is surgery, radiotherapy. This is kind of subtractive way to operate. You go in and you cut, you burn, you replumb. So this classical mode of intervention can also be used for prevention, for symptoms control, and for curative outcomes. An example of preventative surgery is a prophylactic mastectomy. This is done in people who are in danger or at risk of having breast cancer. It can be used for symptoms control. For example, the installation of a shunt. This is a little tube that drains cerebrospinal fluid from the head into the abdominal cavity. This is done in people who have hydrocephalus. It can also deliver curative outcomes. For example, in tumor resection, if surgery is performed early enough and the whole tumor is removed before it has had a chance to metastasize, then it leads to curative outcomes. So these two are the most common, the most classical modes by which medicine is practiced. However, in recent years, there have been some advances that are very exciting. One of them comes from regenerative medicine. And here you go in and you add tissues to the body. For example, as a curative outcome in people who suffer from leukemia, this is a blood-borne cancer, stem cell transplants can have curative outcomes. In this vein, bioelectronic medicine also represents a novel mode of intervention. It can be used, for example, to prevent disease, to prevent death. And one example is the implantable cardioverter defibrillator. So let me explain. This was in the news recently. Actually, last summer there was the football tournament, the Euro, and one footballer from the Danish team suffered cardiac arrest during the game and collapsed in the field. And he was fitted with an implantable cardioverter defibrillator and started playing again. He played his first game again yesterday. So this is a device that has an implantable pulse generator, as it's called. It's shown here. And it's implanted under the skin on the chest with leads that go and get inside the heart. Now, these leads have electrodes that you might be able to see here. Those electrodes monitor the beating of the heart, the electrical activity that comes from the beating of the heart. And if they detect that the heart stops, then this will give a high current pulse and restart the heart, preventing thereby death. Having said that, most applications of bioelectronic medicine are for symptoms control. One example is deep brain stimulation for tremor. This is shown here. This is a patient who suffers from tremor. He's been implanted with a device that again has a pulse generator that is sitting under the skin. And then with leads, electrodes are placed deep in the brain in a structure that's called the subthalamic nucleus. And through stimulation with current, the symptoms of tremor get much better, are treated to a good extent. So this is with the device switched off. You see that this patient has difficulty in writing, in holding a cup of water. in functioning normally. And then with a flick of a switch, the tremor is considerably less pronounced. So these devices have been used for treating motor disorders such as dystonia, Parkinson's disease, tremor, as is shown here. They are being used to reduce the frequency and severity of seizures during epilepsy. They're being used for neuropsychiatric disorders such as obsessive compulsive disorder and depression. They're being tried in the treatment of obesity and many other indications. So arguably, this field goes back to the work of Luigi Galvani in the 18th century. This is a famous by now experiment where he applied a potential on a dead frog and got the legs to twitch. And then 200 years later, there were already medical devices. This is a pacemaker. In about a year before, this device became fully implantable. So at that time, patients had to carry this cart with electronics that had the battery and the control electronics with them at all times. Today, these devices have been miniaturized and can be inserted inside the heart using a very simple procedure. So if we start the clock for bioelectronic medicine in 1958, which is the first year when a fully implantable pacemaker was developed, then a decade later, there were cochlear implants. These are devices that are implanted in the inner ear to help people with deafness hear. Then a decade later, there were spinal cord stimulators for helping people who suffer from chronic pain. Then a couple of other devices on peripheral nerves, on the brain, which came to the forefront in early 2000s, in the eye and other positions in the body. And more recently, this type of stimulation of peripheral nerves is used to treat diseases that go beyond the spectrum of neurological ones. So, for example, autoimmune diseases such as type 1 diabetes, Crohn's disease, rheumatoid arthritis are believed to be treatable with electrical stimulation. So the trend is definitely accelerating, and there are more and more devices that are gaining approval from the regulatory bodies to be used for the treatment of disease. It's actually coming to the radar of very big companies such as Google's parent company Alphabet and pharmaceutical giants such as GSK. These two companies formed an alliance that is called Galvani Bioelectronics in order to treat disease not with pharmaceuticals, but with miniaturized implants that will do electrical stimulation of nerves. And then the founder of SpaceX, of Tesla, the founder of Facebook, they all got into this game. So it's a game that obviously also has commercial value and is attracting a lot of interest. It's also attracting a lot of hype. A lot of Hollywood movies portray bioelectronics, and it's mostly a scary person trying to do damage in our civilization. The truth is more like what you see here. This is a boy who is wearing a cochlear implant. This consists of an external part shown here. This picks up the sound through a microphone, processes the sound here, and then uses this antenna to convey the information into an implant inside the head in a part of the system which is called the cochlea. And through electrical stimulation, this guy can hear for the first time, and just look at the wonder in his eyes as he can hear now for the first time. So bioelectronics can have very beneficial, many benefits in people. There are ethical issues. Any medical technology raises ethical issues. When you act on the brain, which is the seat of consciousness, is the organ that gives us the sense of self, the ethical issues can get even more important. And the message that is important to pass here is that when you develop medical technology, it has to be done in constant communication with society and all the stakeholders. This is from an old movie, and this is the quintessential crazy scientist who's working in a dark basement and is trying to galvanize, as it was called, or reanimate a dead corpse by applying electricity. And obviously, this is not the way to move forward and to develop technologies. This is unacceptable to have this type of work happening. But through collaboration with society, through open dialogue, through involving all the stakeholders, devices like this were developed. And this is, again, the cardioverter defibrillator that I showed you earlier, that can reanimate someone whose heart has stopped. So we can use bioelectronics for good and to develop medical technologies that benefit humankind. It has to be done in collaboration with society. That's why events like this one that you're organizing today are very important. So that was a bit of a long introduction into what is bioelectronic medicine and what it could do. Now, let's look at the current status. What are the limitations? Why don't we see this incredible technology deployed at scale? There are some formidable limitations. First and foremost, we do not understand how the brain works. It's a very complicated machine that consists of over 80 billion neurons that are connected. Each neuron can be connected to 1,000 other neurons. So the numbers are astronomical. The complexity is enormous. We do understand how two neurons communicate with each other in a reasonable level of detail. For example, a neuron is a cell that has these extensions that are called dendrites that accumulate information from other neurons, again, as many as 1,000 other neurons. This information is compiled, and then the output of this computation is an action potential, which is like a voltage pulse that travels down this process, which is called the axon of the neuron. And then the information arrives at the synapse, which is where this axon comes very close to another neuron. And then it releases neurotransmitter here in the synapse that either excites or inhibits the postsynaptic neuron. So these are phenomena that if you pick up a neuroscience book, you'll be able to read in good level of detail. And there is a semi-quantitative understanding of how these things work. However, what is not understood is how we go from this cellular phenomena to macroscopic behavior. We do not understand how this cell communication leads to our behavior. In order to bridge this gap, electrophysiologists use electrodes that are placed in or around the brain in different configurations. For example, you can go into the brain with penetrating electrodes. This is called stereotaxic electroencephalography. And in this case, you can, if the electrodes are small, you can record activities of single neurons. You can place electrodes on the cortex, on the surface of the brain. This is called electrocorticography. And here you can read large assemblies of neurons. You can read the rhythms that come from many networks of neurons. Or you can put electrodes on the skin, on the scalp, And in that case, you can record much larger parts of the brain underlying the electrode and get a bit more average type of data. So to call on an analog, here you're seeing the trees, but you're not seeing the forest. Here you're seeing the forest, but you can't see the individual trees. And corticography is somewhere in between. Now, another thing that is not understood is what is the mechanism of neuromodulation? Why, when we stimulate with an electrode in the brain, why do we change behavior? So these are big picture problems that will take generations of scientists to understand. Your generation will do hopefully a lot of work on this topic. You'll advance the field further, and maybe it will take another generation, the students you'll train, and another, to gain a good understanding of how the brain works and solve these major puzzles. However, there is something more technological that needs to happen as well. And these two go hand in hand. We need better technology to interface with the brain in order to understand how it works. There are a lot of issues that I will go through one by one and explain what is the problem and what can be done to address it. But the message I want to leave you with is that this is a field that is defined by technology, and it's limited by technology. So new technologies are urgently needed in order to establish a good interface with the brain and allow us to study it and understand it, and also deliver better treatments. So that's where the introduction ends. I will now go through these topics and explain in a bit more detail what are the limitations and what work needs to be done. So let me start with the first one. The brain is very soft. It has the consistency of jello, and it also moves. It pulsates during blood flow, and it jiggles inside the skull every time we move our head. So how do you make contact with such an object? Electrical contact and mechanical contact. So there has been a lot of work on this topic over the past decade. And here are two examples. The first one comes from the group of John Rogers. At that time, he was at Illinois, the University of Illinois in Urbana-Champaign. Now they moved to Northwestern. So they made electronics that were so thin that they could conform very nicely to the surface of the brain and make very good mechanical contact. So this is a brain, and as you see, it's not flat. It has topography and it's soft. So you cannot interface with it with any type of electrode. You need very soft and very conformal electrodes to make good contact with it. So what they did is they made electronics that were so thin that you could not lift them off their substrate. They would collapse under their own weight. So they developed a technology using silk, which is a biocompatible material. They would pour silk on top of these electrodes to give a mechanical stability. Then they could lift them up, transfer them to the brain, and then dissolve the silk and leave a tattoo-like device conforming nicely to the brain. Then there was a group of Stephanie Lacour at EPFL who took this a step further. They used silicones. These are elastomeric materials that can stretch, so rubbery type of materials. And they developed electronics on those. This is tricky to do because when you're processing your electronic device, the elastomeric substrate can change dimensions because it can swell, for example, in the solvents that you use to process. So it is challenging to develop this technology, but they have succeeded. And you now can have electronics that not only sit very conformably to tissue, but can also stretch with it. So when this rat walks and jumps and does what rats do, these electronics that sit on the spinal cord can move and hold good mechanical contact. So these are two examples from work that was done that largely address this limitation. There are many other examples of very good work in this area, but they do, by and large, this problem has been addressed reasonably well. We now have electronics that can make a good contact with tissue that is soft and can follow its movement. Another limitation is that the signals that are produced by neuronal communication are very small and also diverse. What do I mean by that? When two neurons communicate, when the action potential comes down the axon, there are small ions such as sodium and potassium that go in and out of the membrane. These represent ionic currents in an electrolyte. The electrolyte is the cerebrospinal fluid, but they're very small in magnitude. And you have these ionic fluxes in an ocean of ions. So measuring them is very tricky. They're also diverse. So you have the small ions going in and out of the membrane. You have neurotransmitters coming out at the synapse. You have hormones that are circulating in the cerebrospinal fluid. You even have components of the cell being exchanged in the form of exosomes. So you need to make sensors and use multiple techniques in order to understand this neuronal communication in bigger level of detail. So here I will cite one example from our own work that shows how we can couple between ionic and electronic carriers and gain a better understanding of these signals and be able to capture them more accurately. What happens when, again, a neuron fires is ionic currents come in and out of its membrane. So what you're trying to detect is the motion of ions in an electrolyte. The electrolyte is the cerebrospinal fluid. And you do this by coupling ionic motion to electronic motion inside a material, let's say silicon, for example. An ion will be coupled electrostatically with an electronic charge inside your electronic material. So when this ion moves, then the electronic charge moves as well, and you pick up the displacement current. However, if you try to do this coupling between ionic and electronic charges using silicon, there is an issue that when silicon is immersed in an electrolyte, you'll form an oxide, or you'll put a nitride in order to protect silicon from the electrolyte. So this insulating barrier spaces away the two carriers and decreases their interaction. That's why most electrodes are made not out of silicon, but are made out of noble metals such as platinum. In this case, you do not have an insulator, and ionic and electronic carriers can come very close and interact quite strongly. But even in this case, their interaction is along a two-dimensional plane. What we developed is materials in which ions can penetrate into the volume and couple with electronic carriers in the whole volume. So you go into a situation that looks like this one here. I'm sorry, my clock is broken and goes a bit berserk. It will stop in a moment. So these materials are made out of what is called conducting polymers. I'm not going to go into the details, but consider them like sponges that have electronic conductivity and can accept ions into their structure and couple in a three-dimensional fashion. So in these materials, you have a much closer interaction between ionic and electronic carriers, and you can be detecting ionic fluxes with higher sensitivity than you can in this. So if you want to go more into the details, these materials are characterized by a capacitance that scales as a function of volume of the material. So the thicker you make your electrode, the stronger the interaction between electronics and ionics, and the higher the capacitance that you'll get. Just to give you an order of magnitude of where things stand, if you put a film of this material, this little sponge that is just 130 nanometers thick, then you are going to have a capacitance that is 2 orders of magnitude larger than the capacitance of a flat metal film of the same area. So that means you're coupling electronic and ionic carriers with 2 orders of magnitude higher strength. That means now that you can make your electrodes much smaller and still be able to detect these small ionic currents. So this is shown here. These are small electrodes of the order of 20 microns. Just to calibrate you, 10 microns is the size of the soma, the body of a neuron. And these electrodes can be deposited on very thin plastic films that are conformal to the brain. And so this is an electrode array shown here with small electrodes, and it's shown to conform to radii of curvature that are consistent with the brain. And then when you put this array on the cortex of a rat, you can detect these signals here that correspond to single neuron activity. So we can make very small electrodes today that can record from the cortex single neuron activity. So the maximum resolution you can hope for. So this technology moved to humans, and this is shown in a clinic here, recording these spikes, which are single neuron activity, from the surface of the brain of a human patient. So this is used to map the brain before surgery, and at an exceptionally high resolution, down to single neuron signals. Now, this work was already a few years ago. Since then, we've been working to make these devices optically transparent so that you can combine electrical activities with optical measurements. This is shown here. This is a transparent electrode array. It can measure these electrical activities of neurons, but at the same time, we can shine a laser through it and do what is called super resolution microscopy. This is a particular technique which is called structured illumination microscopy, and it can detect markers that are down to molecular level. So look at that. You get incredibly high resolution. These are markers that appear at the synapse, and you get chemical specificity, and at the same time you get the electrical activity of neurons. These devices can also be made transparent to MRI, so that you can combine them with structural and functional MRI. And through these multiple techniques, electrical measurement, optical measurement, MRI measurement, you can start putting together a better picture of how the brain works. So these are just examples of work that is being conducted to address the fact that signals are difficult to detect, and there are many signals to be detected. Another issue is that bioelectronic medicine, as it's practiced, it's highly invasive. You need to place electrodes into the brain, on the spinal cord, and often multiple surgeries are required to move the electrodes or to replace them. So it would be good to develop technologies to avoid that. One example is in spinal cord stimulation. Here, electrodes are placed on the spinal cord and stimulate the spinal cord electrically in order to relieve pain. And this is given to people who suffer from chronic pain, where they can have constant, terrible pain sensation. And through this neuromodulation technique, through electrical stimulation, this pain is replaced by a more agreeable feeling. There are two technologies here. One uses this one-dimensional electrode arrays. These are called percutaneous arrays. These can be placed on the spinal cord through the space between vertebrae. So this device is inserted between the vertebrae and then pushed up. But unfortunately, it doesn't work very efficiently. It covers only a small part of the spinal cord, plus it can move after implantation and lose efficacy altogether. There is then this device. This is a two-dimensional electrode array, which is called a paddle electrode. And this provides very efficient therapy because it stimulates at multiple points on the spinal cord. It doesn't move, and so it's a very efficient treatment, but it requires a laminectomy. To place this on the spinal cord, you need to remove the bone, part of the vertebra, in order to put this in. So a patient today faces a choice. Do they undergo quite invasive surgery and then have a device that works well? Or do they have facile placement of this device without surgery and then but then risk a device that is not as efficient? So here we received inspiration from the field of soft robotics, and more precisely from pneumatically activated soft robotics. This is an example of work from George Whiteside's laboratory at Harvard, where they made this soft object. And by using pressurized air, excuse me, they could make it move. So the thing that you take away from this is that you can pneumatically activate a soft object to change size. So what we thought, maybe we can integrate fluidic channels with our soft electrodes and get them to change shape so that we can make them as percutaneous arrays that can be inserted in the spinal cord without a laminectomy. And then once inserted, we can actuate them to open up and deploy and become paddle electrodes. So this is shown here. This is a paddle electrode that can be then rolled up to look like a percutaneous array. If you design it appropriately, then its electrical characteristics don't get affected by this operation. And this is shown in an in vitro model, how you can have it rolled up and then actuate it, and it will open up. So the device design here consists of two planes. We have parylene, which is a plastic material on which we put our electronics, and it's conformal and thin and all that. And with lithography, we can define the electrodes. And it's connected, it's attached to a PDMS, a silicone-based component that contains the fluidics. One of the design considerations here was to make sure that when the device is pressurized, it doesn't expand in this direction because it would put a lot of pressure on the spinal cord, but rather expands laterally. And you can do this through design of attachment structures where you attach the top and the bottom of the fluidic together so that they cannot blow up. And in this case, we send the pressure towards the sides, and then that can help unroll the device. So to test this, we went to a human cadaveric model that recapitulates the anatomy, and we had to learn how to reconstitute all that. And this is shown here. This is a needle that can go through the vertebrae and deliver the device. And the device then is pushed up on the spinal cord. This is a fluoroscope, is a device that uses mild X-rays to image the device. And once inserted in position, then it can be manually with a syringe expanded and visualized. So this is shown here. This is the needle that is inserted in the vertebrae to release the device. The device is then placed up the spinal cord in the position where it needs to be. And then it can be expanded. These are two X-ray markers that coincide when the device is unrolled, but as the device is rolled up, but when it's unrolled, then they move laterally to show the extent of expansion of the device, as shown here. And what we can do then is we can go and do a laminectomy, so open up here, and then look at the device as it sits on the spinal cord. And again, this is the site where the device was injected up by our surgeon. And this is one of the X-ray markers, and the other X-ray marker is there. So the device sits nicely on the spinal cord as expected after expansion. So this is now, we just received funding to give this to living humans as a way to translate it to patients at scale. So this will go to first in human within 2 years. And after that, we hope to be able to transfer it to a company to move it to patients at scale. Now, the last few minutes, I would like to talk about the foreign body reaction, which is a very tricky thing. And I'll show you some unpublished work from the lab. So what this refers to is when you place a device in the brain, this is bad not only for the brain, but also for the device. The brain attacks the device and coats it with a layer of scar tissue, rendering it less active. There are many, many ways that people are trying to avoid or minimize this foreign body reaction. For example, by using very thin, very soft devices, by coating them with layers of gel that will release anti-inflammatory medication, and many others. But by and large, this remains a problem. So there is a concept out there in the literature that is called the living electrode concept. And it goes as follows, is that if you have your electrode and you coat it with this layer that I mentioned earlier, the conducting polymer that is used to enhance ionic electronic communication, then you can coat it with a gel that contains living cells and then implant the whole thing. So now what the body sees is it doesn't see your electrode, it sees a gel that has living tissue. And maybe in this case, you can fool the body into accepting the electrode and not try to attack it. And in this case, you can also have better communication. Imagine that you implant neurons that then go and innervate the native tissue. Or imagine that you implant muscle cells and then neurons from the body can innervate those muscles. So that could even lead to better communication. I'm very excited about this concept because it changes the paradigm. Now you're not just implanting electronics, but you're implanting electronics and tissue together. So you're bringing together regenerative medicine and bioelectronic medicine. Another way you can think of this is that you are implanting tissues that are going to integrate with the body and repair some of the damage that was done by disease or injury, and use bioelectronics to control this repair. So this brings you in a new type of thinking frame. We can start talking about regenerative bioelectronics, which is again at the interface between bioelectronic medicine and regenerative medicine. So here, to provide a proof of principle demonstration, we used stem cells that are derived from human-induced pluripotent stem cells. So the idea is you take cells from a patient, you turn them into pluripotent stem cells, and then you forward program them to different somatic cell types. For example, you can produce neurons, myocytes, oligodendrocytes, and other types of cells. This is shown here. This is muscle being formed through this forward programming process, and it takes a couple of days, and you end up having really nice myotubes formed on your device within just a couple of days. So the idea here is to work on a peripheral nerve injury model and use a device. So in this case, you have a severed nerve. You have the axons of the motor neurons that are looking to form a target, and you present them with muscle cells that are grown on your device. So then this motor neuron axons will innervate the muscle, and when the brain fires signals going through these axons, they will stimulate the muscle, and you will record here muscle signals. So that was the theory, that was the hypothesis. This is how the device looks like. It's attached on the nerve. It has electrodes that are depicted by these spots here, and we can interface with different parts of the nerve. So the idea is that we take the device, grow muscle tissue outside the body, and then implant it on the sciatic nerve here, attach it, and hope that it will innervate. And these are the data. These are very recent data, yet unpublished. This is histology that shows that these muscle cells that we're presenting into the body survive inside the body, survive the process of implantation. And more exciting is that we start recording after 4 weeks, which is roughly the time that is expected for this innervation to take place. We can now record these beautiful muscle signals when the rat is thinking about moving. So this is, again, recorded on a severed nerve, and we're recording activity from muscle on the device that is being produced by the brain. So very exciting first demonstration of this concept, and we hope this will lead to novel ways to treat peripheral nerve injuries. So let me conclude. What I tried to do today was to convey my enthusiasm about bioelectronic medicine as a way to prevent and treat the symptoms of disease. The technological challenge here is to establish a stable and effective interface between electronics and tissue. We are always trying to work at interfaces between disciplines. The interface between bioelectronics and soft robotics can provide solutions that reduce the invasiveness of surgery. I showed you here a spinal cord implant that uses concepts from soft robotics to expand inside the body. I think this can help us dream and think of other devices that can go into the body through a small pinhole surgery and then expand or even move inside the body, there's a lot of work to be done. Even more excited about the interface with regenerative medicine, because here you change the paradigm. You don't just implant an electronic device, you implant an electronic device and tissue. And this means that you can start repairing damage by disease or injury and lead to curative outcomes. So because I'm talking to electronic and electrical engineers, I thought I'll add one more slide. What are the challenges and the opportunities if you're not interested in bioelectronics? How does the existence of bioelectronic medicine change your world? For example, implantable electronics are transitioning to thin film technology. This means that electronics that are flexible and stretchable will be receiving increased attention. So you'll be seeing a lot of work in that direction. Making electronics visible inside the body through integration of X-ray marker technology is challenging. And the packaging of electronics, how do you attach them? If something is very flexible and conformal, how do you attach it to cables that come outside the body? How do you insert that in the body? This technology needs to be developed. The fact that bioelectronics and regenerative medicine are converging has implications for additive manufacturing. For example, hybrid fabrication such as 3D printing and bioprinting can combine to give rise to new types of devices. And then those of you who might work on novel materials, such as polymer semiconductors, should be aware that while these materials are great for understanding electronic properties, their translation to the clinic is difficult because you need to spend an enormous amount of effort to take a new material from the lab to the clinic. So with that, let me thank my group and collaborators. I showed you slides from the work of these people. This included medical professionals, so neurosurgeons, Damiano Barone, who co-leads the lab, Jiang Lei, and I forgot to highlight Yilin Wu. These are the guys who helped with the surgery. The animal work was done by Alejandro and Sam. And then we have Vincenzo, Amy, Ben, and Sagnik, who are the engineers, and collaborations with different groups at Cambridge. Chris on the expandable implants, Gabi on the imaging of the devices, and Mark on the stem cells. So thank you for the kind invitation. It's a pleasure to be with you. I'll stop here, and I'm ready to take questions.
Thank you so much for your great presentation. We are so happy. And can we ask the questions from YouTube chat too? Okay, I start with the first question. As a natural sciences student, how can projects be done in common with your field?
Yeah, so the field is very interdisciplinary. In our lab, we have physicists, we have material scientists, we have engineers, electrical, chemical, even mechanical. We have biomedical engineers, quite a few of them, neuroscientists, and medics, all under one laboratory. Nobody comes into the field having the whole picture. So we have to collaborate among us. We have to collaborate with people outside the lab and learn from each other. And this is a process that takes a long time. So the advice is that when you're an undergraduate, you should focus on learning your own discipline very well, and also keep an eye out for other disciplines that you might consider exciting. And at a graduate level, then you can start using your undergraduate knowledge to gain an approach to other fields through collaboration. There is no undergraduate on bioelectronics, right? And everyone, a physicist, a chemist, an electrical engineer, a chemical engineer, a neuroscientist, can contribute, can have valuable contributions in this field. But that's done at a graduate level or further.
It has been an enlightening perspective for us. Thank you for your answer.
Thank you.
Our next question is from one of our participants, İdil Polat. Thank you for this great presentation. Do you have any advice you would like to give us so that we can be successful individuals in our careers?
Yeah. I mean, it just follow your dreams and aspirations, right? It's in academia, in science and technology, we are lucky to have a work that is also our hobby. And it's great because you wake up every morning and you're happy to go to work. There's always something new to understand, something new to discover. And it's a great pleasure. It doesn't happen in all professions. The thing that I keep telling my research students is that the biggest danger we run as scientists is that of burnout. Because we love our work so much, we tend to focus on it 24 hours a day. And that's not very good. You can kind of lose a bit perspective. So it's very important to also take care of yourself, make sure you take breaks, make sure you have a good social life. And science, technology is super exciting. Don't let it become all-consuming. You should also have a good social life. You should also go out with your friends, and that's the key advice that I give to people. And again, the people that I interact with, the young students who come in, they're all very dedicated, very much into their profession. They really enjoy science and technology. They're off to a great future. They're off to works where they contribute to society. They take a lot of pleasure from the day-to-day work. What I tell them is make sure you don't burn out.
I think your advices will be very helpful for all of us in our careers. The next question is from Buse Ünlüeroğlu. Thank you very much for your presentation. It was an excellent presentation. What level of knowledge do you currently have about bioelectronics being studied for Parkinson's patients?
So bioelectronics in Parkinson's has been developed several years ago in terms of implantable devices in the brain to stop the tremor and the shuffling gait, as it's called, to help with the motor neuron aspects of the disease. Historically, it was found as an accident. So it was known that the deep brain structure, which is the subthalamic nucleus, is where a lot of fibers crossed that go from the motor commands of the center to the periphery. And there were surgical techniques to go in and create a lesion at that part to help people with tremor. So a scientist in France was trying to, rather than cutting with a knife, to use electrodes implanted in the brain to burn that tissue. But they found that way before you have enough power to burn the tissue, the symptoms became better just by electrical stimulation. And this, of course, is much more preferable than damaging tissue permanently. That's what gave rise to deep brain stimulation being used in Parkinson's disease. It was kind of an accidental discovery. So since then, a lot of progress has been made. There have been people who received implants in early 2000s, so they are among us now for over 20 years with implants, and they still work. The mechanism of neuromodulation is not very well understood. So it's known that that part of the brain, if you scramble the signals through electrical stimulation, you have beneficial effects, but the details are not known. The current technology consists of implants with 4 electrodes, and these are implanted with precision of about 1 millimeter, and you try to hit a structure which has the size of a green pea, so a fairly small structure. And a surgeon can place the electrode array, making sure that at least 1 electrode is inside that structure. And then a clinician will try to adjust the stimulation, find the appropriate pair of electrodes between these 4 electrodes that are implanted to send current from 1 electrode and collect it from the other, so that it gives maximum relief of symptoms with minimum side effects. So after implantation, the patient will undergo some exploratory setting adjustment for a period of 1, 2, 3 months until the right parameters are found to optimize the balance between control of symptoms and minimizing side effects. In terms of technology, there are now stimulators that have 40 or even 80 electrodes, but programming those becomes very, very difficult. It's impossible that a clinician will be able to try all these combinations. So that's where machine learning can play a big role. You can imagine implanting such a complex stimulator to a patient and then giving them a little handheld device that says, is this better or is this worse? And then through some exploration with machine learning, you change a setting, for example, and then the patient can say yes or no. And then it learns to identify the right set of electrodes that give maximum relief. So I think this is where things are moving in the future.
Thank you for your clear and understood explain. And the next question is from Ufuk Shen. Thank you for presentation. Soft robotics seems useful for modeling. Does this process occur fast? Is it hard to simulate? And also, which programming language are you using? Seems interesting.
We're not experts in computer modeling of these devices. We use things such as COMSOL, for example, to just look at static properties. I know there are much better software environments to explore with soft robotics, but we're not experts. I wouldn't know which ones.
And thanks for your answer. And the next question is from our participation Ezgi Olgun. Professor, it was a presentation that I watched in one breath, just like last time. I have a lot of questions that I want to ask you, but I will try to keep it short in order not to be unfair to other friends. Do you have any studies on neurogenesis? How far do you think neurogenesis can take us in human history? Some say we can skip years on the timeline, and would you ever be considered to come to the Turkey for visits?
Sure, yes. So let me answer the last bit first. Of course, very happy to come visit anytime. I've been to Istanbul well, it was 2014 was the last time I went, and had really excellent tour. I toured 3 universities and had fantastic hospitality and food. So yes, gladly come back anytime. On the neurogenesis bit, so this is a very important question. What I talked about today had a flavor of electrophysiology, which means we're trying to use electronics to either record neuronal activity or stimulate it. And typically, electrophysiologists work at frequencies that go between 1 and 1,000 hertz. That's the region of activities that correspond to neurons when they fire. There are other communities that look at what electric fields do to the brain. For example, there is a community that does electroporation, where they try to use electric field pulses to open up the cell membrane, either temporarily to get drugs in or permanently to break up a cell. They tend to operate at much higher frequencies. Then there is people who are trying to stop neuronal division by using what is called tumor treating fields. They operate at frequencies typically around 200 kilohertz, and there is a device called NovoCure, where you put electrodes outside the head to try to act on glioblastoma, which is a lethal brain cancer. And the theory is that by applying these fields, you stop the process of cell division and you can control the growth of a tumor. So the point I'm trying to make is that in addition to recording and stimulation of neurons, there are other things you can do. You can affect cell division. You can guide the fate of stem cells. You can porate cells and deliver genetic material. These are all very exciting tools, and we're at the very beginning of trying to figure out how far we can go. Among the things that have been demonstrated today is, for example, you can control innervation. You can stimulate a neuron and get it to produce processes and innervate with other neurons in a controlled fashion. So that bodes very well for things such as repair of spinal cord after injury. You can trigger proliferation of cells. Many things you can do. So very exciting field. We're only scratching the surface. I expect that this will keep us busy for many years to come.
And thank you for your answer again. And we would like to see you in Turkey, and maybe you can join another event face to face.
That would be great. Yeah.
The next question is from Boran Toktay. Professor Malarias, do you think one day can think as we do? Also, I want to ask, does adding more tech to human body make us less human? And thank you. Professor, a friend of mine has muscular dystrophy. Is there a way to solve it with bioelectronic engineering? I know it's genetic, but is there any way to stop or slow down?
Yes. So thank you for the question. About AI thinking as we do, that's a very big question. And again, it's beyond my expertise. I can, from what I've heard from experts giving talks, is that it's evolving very rapidly. And it might reach the point where at some point in the not so distant future, it can outperform humans in very complex tasks. There is the von Neumann, there is the Turing test, where if you converse with someone behind a wall and you don't know whether it's a human or a machine, and after having a conversation, you cannot tell whether it's human or machine, then it has reached a level of intelligence that is equivalent to that of human. I don't think we're quite there yet. Even the best AI environments that I've interacted with, you can tell it's a machine, right? When you call to get your credit card unblocked or to talk to a company, if it's a machine, very quickly you understand that it's not human. But it's evolving very fast, so I don't know. I think it will reach that stage in the not so distant future. And there are a lot of ethical issues that are associated with this. Adding machines to the body also is very fascinating, especially the brain. There are a lot of papers on that topic exploring the ethical issues. For example, there are reports by patients who had implanted devices that monitored brain activity in order to warn about an oncoming seizure. If a company that provides such a device goes bankrupt, then the device must be explanted, must be removed. And patients who had that happen to them, they stated that they felt like they lost a part of themselves. So it seems that the brain is uptaking the device as part of who we are. Another example is in stimulation for depression. So electrical stimulation deep in the brain can be used to treat the symptoms of depression. And people with these devices, when they're turned on, they are sometimes unable to feel the whole range of emotions. So, for example, if you go to a funeral, it is normal to feel sad and depressed, right? And if you have a device now that stimulates your brain and prevents you from feeling depressed, that is not normal. There are people who, upon brain stimulation, they become, they take risks. For example, gambling. They become addicted to gambling. And there are statements from people who are only able to understand the impact that this has to them and their family only when the device is switched off. So there are enough examples out there to highlight the ethical issues of acting on the organ that gives a sense of self. And those, of course, should be very carefully considered and balanced between the benefit and the risk that a new technology brings. We work on a project where you can put electrodes on the brain of someone who suffers from locked-in syndrome. This is caused by an injury in the spinal cord pretty high up close to the brain that disconnects the brain from the rest of the body. So while the brain works, that person cannot communicate externally. They cannot control their mouth and all that. So the idea is to read the part of the brain that gives rise to speech and take these signals and interpret them by a computer and use a computer to vocalize. So then a question is, how do you make sure that you only vocalize the parts that are intended to be vocalized? We don't want every thought in our brain vocalized, right? You think, oh my God, what is that person wearing? How can they go out wearing that thing? You don't want that to be vocalized, right? Or anyone who sent predictive text messages. Sometimes you type something and the computer thinks you're typing something else. These are things that need to be considered in the development of technology. It's a very exciting area where you take into account not only technology, but also ethics and society and how it impacts society and all that. It's a game of many stakeholders, very exciting, and requires that type of broad thinking. Now, on the second question about muscular dystrophy, I'm not sure. I do not know what is the status of bioelectronics on that topic. I would start by of course, talking to a doctor who can point you to if there is any way of electrical stimulation, they might be able to point you to it. I would also look at Medtronic being the key company for neuromodulation, to see if they offer any devices for muscular dystrophy. I do not know.
Thank you for your answer. Your explaining is inspired for everyone, including me. And the next question is from Ruket Epozdemir. When a problem turns into an interface with robotics, how can be fixed? Can artificial intelligence or deep learning be used?
Yes, so both AI and deep learning will play a big role in interfacing machines with humans. I can give you a good example in brain-computer interfaces. So brain-computer interface can be used in people with paralysis where the spinal cord is severed at a particular location, and that can disconnect the brain, for example, from the arm. You cannot move your arm. In that case, there are examples out there where an electronic device is implanted on the cortex, on the motor cortex, and picks up brain activity that corresponds to volition. So when that person thinks, I want to move my arm, the neural activity that corresponds to this thought is picked up by the electrode array, can be interpreted by a computer, and then the computer can move a robotic arm. And there are videos out there that you can see where someone wearing an implant controls a robotic arm with their brain, and they can bring a cup of coffee and drink it and all that. Now, this is highly invasive technology that goes into the brain and is trying to read signals at single neuron level. So you want to read a signal that corresponds to the motion of this finger, then another signal, this finger, that finger, to be able to create an elaborate and highly articulate interface. And that's difficult to do. Alternatively, you can use electrodes that sit on the surface of the head. Those are much less invasive, but they pick up signals that are very average. You cannot pick up signals from the neuron that moves this finger versus that finger. But with machine learning, maybe you can train a computer to interpret these very complex signals at the level of not, I want to move my little finger, but at the level of, I see that glass of coffee, that cup of coffee, and I want to pick it up, and let the computer figure out how to execute that movement. So rather than using our brain to plan the details of the motion, extend the arm, open the arm, close the arm, pick up, contract the arm, bring it close, lift it up. Instead of the brain having to do all that, the brain just sends a command to the computer, fetch me that cup of coffee and bring it to my lips. And then the computer learns how to control the arm to do that. So I can see the future of brain-computer interfaces being less brain and more machine. The brain sends the high-level command, and the computer executes the minutiae of the motion. I think that will help design less invasive and more performant interfaces.
Thank you. I think it was so understandable for all of us. The next question is from Emirhan Balcı. Do you think that the use of bioelectronics in the human body and its control from the outside may cause ethical problems in the future?
Yes. So the safety of implantable devices is a very big topic. How do you prevent them, for example, from being hacked, right? And if you have a device that interfaces with your brain, how you prevent that information from being stolen. It's similar with all medical data in a sense. You know, these days there is a lot of movement towards making all your medical history available to your doctor, to yourself, putting it on a cloud so that it can be picked up by a hospital and used as needed. And this is a great thing, right? You want your medical history to be available to your doctor, to your pharmacist, to yourself. So when a pharmacist is giving a drug to you, this gets checked against any allergies you might have and all that. So that's a good thing, making your history available, but available only to the right people. You don't want some of the details being broadcast in public, or some of the details of your previous history necessarily going to a future employer or to an insurance company. So these are ethical questions that are very general and have to do with medicine. Now, when with bioelectronics you act on the brain with a device, then there are some additional ethical issues that are becoming important. One of those is agency. Agency means who's the agent, who does something. So if I do something, you know, it's me who did it, right? But if I have a device implanted in my brain, then it gets a bit blurry. Is it the device that controlled my brain and made me do that action or not? So should we be talking about shared agency between brain and machine? When is it appropriate? And where does the human end and the machine begin? That boundary needs to be clearly defined. And of course, there are also questions of liability. What happens when someone has a brain implant and behave in a particular fashion? Is it the fault of the human, or is it the fault of the company that made the implant? So all these issues are very complex and need to be explored.
Thank you for your all answers. They are so important for us, and it was an excellent presentation. Thank you so much.
Thank you. A pleasure, a great pleasure to be with you, and enjoy the rest of the weekend. Thank you for having me.
Professor, I wish we had more time because I have a lot of questions. But next time I'm going to make sure the whole day will be yours. I can listen to you all day.
Very welcome. You're very, very welcome. It was a pleasure. Take care. Bye-bye. Bye.
Hello, friends. Our next session will be start around 5 minutes. See you later.
Hello everyone again, I'm Özlem, and Hilal and Aleyna will moderate our current session. First, how are you, Hilal?
Thank you. I'm fine. And what about you?
I'm fine too.
Okay. Hello and welcome again to the fourth session of the SoScience event. I'm Hilal Nurdunen, student at Gebze Technical University, and I'm in my prep year just before studying molecular biology and genetics. And
Hi everybody, I'm Aleyna Zehir. I am student at the Gebze Technical University from the Department of Chemical Engineering at the first year.
Dr. Umut Gürkan is among us and attending from Case Western Reserve University. On this session, dear doctor will inform us about innovation-enabled, accessible precision medicine approach for underserved populations. Sir, if you wish, you can share your presentation.
Okay, thank you very much. Okay, can you see my screen?
Yes, we can see.
Okay, would you like me to get started? Should I start?
Yes, please.
Okay, thank you very much everyone for inviting me today. My name is Umut Gürkan. I'm an associate professor at Case Western Reserve University in the Case School of Engineering. I'm in multiple departments: mechanical and aerospace engineering, biomedical engineering, and also Department of Medicine. And our work is focused on developing and translating diagnostics and cures for underserved populations. Here, I will focus on how we do this, like how to develop and translate new technologies, new medical technologies for people who need them the most. Our research is highly translational, meaning that we work on the fundamentals, we work on developing a basic fundamental understanding of diseases, problems, health problems, and we work on developing solutions that can be translated. Translate meaning take them from the lab, from the research phase, and commercialize them as products through companies, and then so that people can use them. The people who need these solutions can use them. So this means translation. So I have to start with disclosures. Disclosures meaning I will talk about different technologies, different products that we have developed, and these are actually now commercially available, most of them through companies. And as an inventor and investigator on these technologies, I have interest in these companies, meaning that they have licensed patents and patent applications from our lab, and I'm involved with these companies through employment, stock ownership, consulting, research funding. So these companies are shown here, and we have different types of companies that are operating in different modes. For example, we have Gazelle, He Makes Health. Gazelle is the product name. He Makes Health develops point-of-care microchip electrophoresis diagnostic technologies for sickle cell disease, anemia, thalassemia. Thalassemia is also known as Mediterranean anemia, which you may know about, and also hemoglobin A1C for diabetes. And this technology is now available in many places in the world. And this is a company that is focused on low resource global health applications. The other company that we have developed is Biochip Labs. So Biochip Labs is a microfluidic red blood cell biophysics and blood rheology related product company. And it is available in the United States right now. And it essentially provides new type of tests to measure red blood cell health. A third company that we are working with is Exatic, and their product is ClotChip. This company has products that are in clinical trials right now that is focused on dielectric spectroscopy based point of care tools for clotting and bleeding disorders. Another company that we have started, and it is now it has products in the market in the United States and worldwide, is DxNow, and their product is iMod, and this technology helps sort high quality sperm for in vitro fertilization applications. So I will today focus primarily on the first two companies, Himex and Biochip Labs. Just a second, give me 1 minute, please. Okay, I'm back. Before I start talking about how we develop these new diagnostics and treatment technologies or treatment methods, I would like to briefly talk about why blood cells, red blood cells, and hemoglobin are very important. So in our bodies, in human body, there is actually more than 1.5 gallons of blood, which is about like 6 liters or so. And there are 3 million red blood cells that are made every second in our bodies. And there are 25 trillion red blood cells in our body. And these cells are actually 84% of all the cells in the body, red blood cells. And there are 270 million hemoglobin molecules in a red cell. So hemoglobin is a very important molecule, which I will talk about later, that is responsible for carrying oxygen to the tissues. And in one day, our blood travels 19,000 kilometers. So that's actually the distance from New York to Los Angeles. If you live in the United States, you would know how far it is. Our blood actually travels that 4 times in a day. So blood is currently continuously flowing and interacting with the blood vessel walls and carrying the oxygen to the tissues. So it's a very complex process. And the cardiovascular system, blood cells, blood vessels, they form a functional unit. It takes 3 minutes to circulate 1.5 gallons, to the 6 liters of blood in the body. And if you look at the length of the blood vessels, and if you put them end to end, that's 60,000 miles. And the smallest capillary or smallest blood vessel in the body is 5 micrometers. And the largest one is 1.5 inches, which is about 6 centimeters in diameter, which is a really large diameter, actually 5 centimeters roughly. And the cardiovascular system is composed of arterioles, arterioles, capillaries, and the capillaries to the veins. Capillaries are the smallest blood vessels in the body. If you look at the red blood cells are a tiny bioreactor. They transport gases, oxygen, carbon dioxide. They regulate vascular flow through NO, nitrous oxide. They can actually dilate the blood vessels by regulating the NO mechanism. And they also detoxify hemoglobin. Hemoglobin is a toxic molecule because it contains iron. Iron is not a real biological molecule, so it can damage cells, tissues if it is not detoxified. And red blood cell is responsible for all these things. So the reason I'm starting with the fundamentals when it comes to hemoglobin, red blood cells, blood, as engineers, we have to understand the fundamentals very well, even if it is biology or even if it is human physiology or disease, it is important to understand the fundamentals so that we can develop the best solution, the most important, the most significant solution for a given problem. So it's important to focus on the fundamentals, even if it is not area of expertise. So red blood cells actually look like donuts. They are like round, and they excrete most organelles to be more deformable. So they don't have nucleus. So if you are familiar with cells, in general, they have nucleus. The nucleus has the DNA, and it is responsible for producing the RNAs that go and produce the proteins. So none of those things exist in red blood cells. So they cannot make new proteins. They have minimal self-repair, and they have a lifetime of 120 days. And they also don't have mitochondria. So they cannot produce ATP the way most other cells do, and they can only utilize glycolysis pathway to produce ATP. And they don't stick to surfaces, and they also evade immune cells. So immune cells don't attack red blood cells. They are quite neutral in the body, and they need a lot of energy, ATP. And I will come back to this in a few minutes. And it is very important to have healthy, fit red blood cells for healthy microvasculature. So our smallest blood vessels, which are 10 times thinner than a single human hair, very small blood vessels. So their health depends on the red blood cell health. So if you have healthy microvessels and healthy red blood cells, as you can see here, the blood will flow really nicely and freely through these small vessels, and you will have unrestricted healthy flow. But if red blood cells or the microvessels, endothelial cells in the microvessels or the blood vessel walls are not healthy, then red blood cells can get stuck, and this will stop blood flow, and some parts of the tissues can be hypoxic, like indicated with purple or blue color here. So that can happen if the red blood cells are not healthy. And many diseases actually affect red blood cell health. The most severe ones are anemias, such as sickle cell anemia, and there are infectious diseases that affect the red cell health, such as malaria, sepsis. There are toxins, heavy metals, mercury, lead. They affect red blood cell health. Blood transfusion, if you receive blood transfusion regularly, then the blood transfusion products can cause red blood cell damage and unhealthy red blood cells. Diabetes has been associated with red blood cell health. Kidney disease, cardiovascular disease, almost every disease has some kind of relationship to red blood cell health. And healthy red blood cells live for about 4 months in circulation, but some of them actually cannot live that long. And the reason is certain diseases. For example, severe blood disorders such as sickle cell disease. In that disease, red blood cells live only for 10 to 20 days. In healthy conditions, 120. And there are other disorders such as sickle cell trait, the carrier state, and the kidney disease. In those cases, red blood cells live less than 120 days, but we still don't know for many diseases that lifespan. It is also very hard to measure red cell health in humans, red cell lifespan in humans. And there are actually different research focus areas when it comes to red cell health and how to measure and understand red blood cell health and red blood cell lifespan in circulation. One application is blood banking, transfusion medicine, synthetic blood. So there are actually people working on making synthetic blood, like artificial blood. Drug delivery. Some people use red blood cells as drug delivery vehicles. Since red blood cells can evade the immune system, they are also able to go into the cancer tissue, tumors. You can actually use red blood cells to deliver drugs to those regions. It's a very important research area. Another important area is understanding how diabetes, for example, affects red blood cell health, and how we can actually measure, for example, A1C, glycosylated hemoglobin in diabetes. And shorter lifespan and hemoglobin variants actually can skew the results. So if you have shorter lifespan red blood cells, then your A1C reading may be off. And we actually have research. We actually do research in almost all of these areas, and try to better understand the red cell health and also how to use it for different applications. If you look at anemia and other red blood cell disorders that affect other diseases that affect red blood cell health, you can actually see a summary here. The anemia affects more than 1.6 billion people worldwide, 25% of the world population. And hemoglobin disorders affect about more than 500 million people worldwide. And if you look at where anemia occurs in the world and where these hemoglobin disorders, for example, sickle cell disease, occurs in the world, they are more or less similar areas. Africa, India, Middle East, and low and middle income countries typically, they suffer from these disorders. And if you are creating solutions for these diseases, you have to take this into account. Your solutions should be accessible, usable, and basically affordable by these regions of the world, especially Africa and India. That's where more than 99% of the people with hemoglobin disorders, for example, live in the world. And if you look at the hemoglobin mutations, inherited hemoglobin mutations, we are interested in these diseases because they represent the most severe form of red blood cell disorder. So red blood cells are unhealthy, very unhealthy, if you have a hemoglobin mutation. And common mutations are shown here. For example, sickle cell disease, sickle cell trait, and hemoglobin SC disease, S-beta thalassemia. This is essentially a single mutation that results in a single amino acid difference in the way hemoglobin is made. You can actually see the different types of mutations here. Normally, for example, you would have glutamine here as in the normal hemoglobin type. But if you have one mutation that causes a valine here, for example, you can actually have sickle cell trait. But if you look at it, there are two alleles, as you know, in the chromosome. And if you have mutation in one of them, then you are a carrier. If you have the mutation in both of them at the same location, then you would have the homozygous version and you would have the disease. If you have different mutations in each allele, then you would have a different type of hemoglobin mutation. That's a combination S and C. And if you have a deletion, for example, of one allele, then you would have one mutation and one deletion. It would cause beta thalassemia. So these mutations can happen in different ways. You might actually have a different amino acid, or you might have a deletion altogether. So there are hundreds of these different mutations. And if you look at the combinations, that you can actually have thousands of different hemoglobin, inherited hemoglobin mutations. So this is a big problem. And almost all of these mutations actually emerged due to malaria. These mutations, when you have them in the carrier form, for example, this form, it actually protects you against malaria. So malaria parasite, as you may know, eats hemoglobin to survive. And if you have a mutation in the hemoglobin, then malaria parasite has been shown to not really survive very well. So it actually helps protect you against malaria parasite. That's a long discussion for another time. So normal hemoglobin is hydrophilic, which means that it likes water. So hemoglobin is very nicely soluble in water. And in both oxygenated and deoxygenated states, as you know, hemoglobin is responsible for carrying oxygen, and it should be soluble in both forms to be able to carry the oxygen from the lungs to the tissues. Sickle hemoglobin, on the other hand, for example, it's a mutated hemoglobin. It's hydrophobic when deoxygenated, and it precipitates in water when deoxygenated, and it polymerizes in the red cell in hypoxia. Polymerization means it precipitates. It's almost like forms of water forming ice crystals. So the sickle hemoglobin in deoxygenated states forms these ice crystal-like structures in the red blood cell, and it damages the red blood cell. And the effect on the human health of these hemoglobin disorders is really significant. For example, if you look at the sickle cell disease and trait, which is the most severe hemoglobin mutation, it's quite painful and lethal. In the world, there are 300 million people with sickle cell trait and about 30 million people with the disease. And if you have the disease in the world, on average, it actually causes a 50-year reduction in the lifespan. If you have this mutation, you would probably live compared to the average lifespan in the region, in that location, you would probably live 50 years less, which is a significant reduction. But in developed countries, in the United States, Europe, with advances in diagnostics and treatments, that reduction in the lifespan is about 30 years, which is still a lot. And we need better solutions. We need better diagnostics, and we need better treatments and cures to improve the lifespan in this disease. And hemoglobin mutations are in the growth. It's expected that there will be 30% more people living with hemoglobin mutations by 2050. So I would like to focus on hemoglobin. We are now going deeper into the red blood cell. Now we will look at how hemoglobin works, because we need to understand these really basic fundamental aspects of red blood cells and hemoglobin to be able to create solutions. So I will now talk about Bohr effect. Bohr effect is an important property of hemoglobin. It is essentially the way hemoglobin carries oxygen from the lungs to the tissues, and it changes its affinity to be able to capture oxygen in the lungs and then release oxygen to the tissues. So hemoglobin has two states, T state and R state. T state on the left has a low oxygen affinity, so it is good for unloading. And R state on the right has a high oxygen affinity. It's good for loading. So it likes oxygen more in the R state. For example, when the blood is passing through the lungs, it loads the oxygen efficiently. And when the red blood cell is in the tissues, it unloads the oxygen very easily because of the T state. And this happens through pH. And this effect was actually discovered almost 100 years ago by Christian Bohr, and that's why this effect is known as Bohr effect. It essentially shows that the pH of the surroundings of the hemoglobin affects its oxygen affinity. And this happens with carbon dioxide solubility in the blood. So when you have high carbon dioxide in the environment, for example, in the tissues, that reduces the pH. It creates a more acidic environment, and hemoglobin assumes a low affinity state. So it is able to deliver oxygen if there is more carbon dioxide in the environment. And the opposite is true. In the lungs, there is less carbon dioxide because lungs are open to the air, and it can actually go to the R state at higher pH values to capture more oxygen. So this is a very unique and very important property of hemoglobin for it to carry the oxygen from the lungs to the tissues. Now, another important aspect of this Bohr effect is how the oxygen saturation depends on the oxygen concentration and how hemoglobin actually modifies its affinity to optimize its essentially hemoglobin saturation. Hemoglobin saturation on the left indicates what percentage of the hemoglobin is saturated with oxygen, and the x-axis here shows the oxygen concentration partial pressure. So at higher oxygen partial pressures, as the hemoglobin becomes more saturated with oxygen, it becomes more difficult to load more oxygen to the hemoglobin. It's almost like charging the batteries. If the battery is charged, for example, more than 95%, it takes more time to charge it to 100%. But it takes less time to charge it, for example, from 50 to 70%. So same concept here. As the hemoglobin becomes more saturated with oxygen, it takes more time to saturate it even further. But hemoglobin has this affinity modifying ability to make it easier to load and easier to unload. So this is a very important property of hemoglobin. There are actually other things that modify the oxygen affinity. So this is, for example, 2,3-DPG. It's an allosteric effector. It's a small molecule that can actually modify oxygen affinity. So 2,3-DPG can be produced by the red blood cell. I will talk about this later. And when the 2,3-DPG levels are higher in the red blood cell, it can actually reduce the oxygen affinity of hemoglobin further. For example, when you go to a high elevation where there is less oxygen in the air, your red blood cells would produce 2,3-DPG to reduce to oxygen affinity so that your tissues can still have the same amount of oxygen, even if the oxygen in the air is less. And then later on, if you stay at that high elevation for a few days or weeks, then the body will produce more red blood cells to carry the oxygen that is needed by the tissues. So if you look at now the oxygen affinity, the curve with increased 2,3-DPG and the reduced oxygen affinity, you may actually also have in a certain disease condition premature oxygen offloading, and also you might actually have this persistent reduction in the oxygen saturation. It might actually be bad if your hemoglobin, for example, crystallizes or polymerizes in low oxygen conditions. So I will come back to this in a few minutes. So it is really important to understand the role of 2,3-DPG to be able to, again, develop solutions for these diseases. As I said earlier, 2,3-DPG is higher in high altitude. If you go to high altitudes, in deep oceans, for example, whales, when they dive deep into the ocean, when there is no oxygen, of course, in the environment, their red blood cells produce 2,3-DPG to reduce the oxygen affinity so that it can actually use all the oxygen that is stored by the hemoglobin. So that's how they can actually dive deep and then stay at those deep parts of the ocean for long durations. And in sickle cell anemia also 2,3-DPG levels are high. So imagine that if you have sickle cell disease, your red blood cells might be behaving almost as if you are at high altitude or deep in the ocean. So it's a really stress state. It makes the red blood cells tired and also exhausted and overactive all the time. I will come back to this later. There is also the opposite of 2,3-DPG, which is higher oxygen affinity, and fetal hemoglobin, for example, the hemoglobin that the babies have before they are born, that hemoglobin has higher affinity because it should be able to extract oxygen from mother's blood. I will talk about this later. So now we are actually going even deeper into the red blood cells and the hemoglobin, and we are looking at what modifies the oxygen affinity of hemoglobin. And we already talked about pH, lower pH reduces oxygen affinity, higher 2,3-DPG reduces oxygen affinity. And if you have both of these conditions met in sickle cell disease, for example, then you would have low hemoglobin, sickle hemoglobin solubility, you would have low oxygen affinity, and this will actually cause lots of problems in the cells. Hemoglobin molecules will crystallize, polymerize, and this will essentially cause all the problems. So in essence, in order to prevent hemoglobin polymerization, in order to improve the red blood cell health, you need to understand how these molecules work and how these things are modified so that you can create solutions. We actually have ongoing work in this context, and a student from Uganda, Zoe, is actually working on this project, and her project involves understanding the hemoglobin oxygenation, deoxygenation kinetics, and also hemoglobin affinity modulation. So how can we modify these affinities to make the red blood cells healthier by preventing hemoglobin polymerization? And there are drugs such as voxelotor and etovopirat. These are the drugs that actually modify these properties, and actually we can assess how they improve the hemoglobin polymerization and red blood cell health. Another aspect of the 2,3-DPG is the impact on ATP, the energy produced by the red blood cell. So if you are familiar with the glycolysis pathway, the glucose is converted to pyruvate and lactate, and in the process ATP is produced. ATP is the energy source of all the different processes that happen in the red blood cell. So it's a very important molecule. So if a cell doesn't have energy, then it cannot do anything, right? So in the sickle red blood cells, this is actually a one way of red blood cell producing 2,3-DPG. So in the glycolytic pathway, if the red blood cell uses, for example, an LR shunt, and this shunt is also known as Lubering-Rappaport shunt, it actually can produce more 2,3-DPG and less ATP. So red blood cell can actually produce 2,3-DPG to modify hemoglobin affinity, but it sacrifices ATP if it does that. So, for example, when you go to a high altitude location, your red blood cells produce more 2,3-DPG, but then they will have less ATP, so they will have less energy to do other things. Another modifier of ATP production is PKR, an enzyme known as pyruvate kinase R. It's an isoform of pyruvate kinase. It's exclusive to red blood cells, and this enzyme also helps create ATP in this step, but PKR activity has been shown to be much lower in sickle red blood cells. It's because of the oxidative stress in the red blood cell. So there are actually two sources of ATP in the glycolytic pathway, and both of these sources are compromised if you have an inherited hemoglobin disorder such as sickle cell disease. So one way to improve red blood cell health is to address these shortages of ATP. And there are actually certain research studies ongoing right now to understand how this would improve things. But if you look at the overall impact of all these processes on the red blood cell, you will see that actually there is ATP-related issues, sickle hemoglobin polymerization-related issues, and oxidative damage, and they can actually cause lots of problems. For example, lack of ATP can cause the malfunctioning or not enough functioning of the ion channels, and this can actually cause the red blood cells to dehydrate. So they lose the water, and actually they can become dense. And oxidative damage, the reactive oxygen species can damage the membrane, cell membrane. It can actually make the cell membrane more fragile, and it can actually cause vesiculation of the membrane. And this can also cause an improper management of the adhesion molecules on the surface, and the red blood cell can become sticky. And it can also impair membrane repair and also deformability. Lots of problems that can happen in the red blood cell. And this is essentially our more basic research, understanding how these processes take place in the red blood cell. And what we do in our lab is we develop new measurement technologies for these problems. So we always say that if you cannot measure it, you cannot improve it. You have to be able to measure these properties if you want to develop new methods to treat them, cure them. And we actually do this by measuring, by developing measurement tools for red blood cell adhesion, deformability, density, hemoglobin composition, rheology, the way blood flows in the blood vessels, especially in the microcapillaries, the small blood vessels. And we actually measure these properties in different clinical states with different treatments, for example, adhesion inhibition, hemoglobin modification, and also genetic-based therapies, gene therapies, if you have heard about them. And I will talk about those briefly. So one way to do this is actually to mimic the human microcirculation in the lab environment, and we call this microcirculation on a chip. We actually created microfluidic devices that mimics or that represents important features of human microcirculation, the smallest capillaries in the body. And we do this by looking at the size of the microcapillaries, the flow conditions, velocity, pressure. These are like the engineering aspects of the project essentially. And we create these conditions by representing these parameters in a microfluidic device. We also use the surface functionalization. We functionalize the surfaces with endothelial molecules or cells, and these are the students who have worked on these on this project over the years, and you can see the publications here as well. So the main idea here is how can we represent the blood and blood vessel as a functional unit, and how does it come into play, in these diseases. And it is important to be comprehensive in this case to represent the interactions between the white blood cell and the red blood cell and the blood plasma and the endothelium. So we actually use different aspects of these interactions to mimic these properties in the microfluidic devices. We also use the normoxic and hypoxic state. So blood sometimes flows in normoxic environments. For example, in the lungs, it is normal oxygen levels, but in the tissues, hypoxic, which means low oxygen availability in the tissues. So we can actually mimic both of those conditions in these devices. And by controlling these parameters, for example, hypoxia, we can create the unhealthy state of red blood cells and microcirculation in these devices, in these chips. So one of the questions is, for example, how can you reveal the worst behavior of these cells or the total accumulated damage over their lifetime? And here the concept is actually we let them flow in a microcirculation on a chip device in hypoxic flow, and then we can observe how these cells behave in those conditions. And we have actually shown over the years that hypoxic flow causes more adhesive red blood cells. They also cause stiffer red blood cells, and they also increase the blood viscosity. Blood becomes thicker, almost like honey. If you use it as an example, if it is in a hypoxic condition. And we have also shown that these properties are really subject dependent, which means that certain people have certain characteristics of hypoxic flow, and it also associates with clinical severity. And different therapies actually can modify these properties, which is good. So now let's talk about the ATP in the red blood cell, the energy source. ATP is very important in all the cells. I mean, that's how we live. Our cells can produce ATP from glucose and other things, and ATP is used by the cells for different functions. And in the red blood cell, one of the functions of ATP is to maintain the hemoglobin, maintain the membrane properties of the red blood cell. So there is actually a molecule known as phosphatidylserine, and this molecule acts as a signal for white blood cells to come and essentially kill the red blood cell when the red blood cell is pretty old. So if PS levels on the membrane, for example, increases, it is a good signal. It's a signal that the red blood cell uses to have itself removed from the circulation. But in the case of ATP shortage, then this PS exposure or PS on the surface increases prematurely, even when the cell is not ready to die. And that happens because of this competition between two enzymes, phelipase and scramblase. Phelipase is an ATP-dependent enzyme, and scramblase is a calcium-dependent enzyme. If there is not enough of ATP in the cell, then the phelipase will not be able to flip the PS to the inner leaflet of the red blood cell. And if you have more PS on the surface of the cell, then it's going to signal that it is the end of its life. And that actually causes many of the red blood cells to be cleared from the circulation early. And we are actually working on understanding the link between how these properties can actually change adhesion and deformability and what governs it. And it is actually quite important to see that stiff cells adhere more, adhesive cells are stiffer, and they are all interrelated. And these are the students who have been working on these projects over the years. So another important property of red blood cell is adhesion, its adhesive properties, and how these red blood cells can adhere, detach. The question is, for example, if they stick to a surface, is it a permanent adhesion or would they come off, or is it a dynamic event? It is a really important question because it has certain implications, as I will talk about later. So we have actually shown by using really advanced microfluidic devices and imaging technologies to show that red blood cell and detachment are dynamic in flow. So adhesion happens, deformation, detachment, adhesion, deformation, detachment. So it's not like a permanent process that happens once and stays that way. And we have actually shown that with the collaborators here, and Utku is a PhD student in our group, we have shown that actually this dynamic process, and actually you can use it to modify the disease. And this actually brings us to abnormal adhesion of red blood cells and what you can do to prevent it. You can actually see a summary of all the abnormal adhesion events that happen between red blood cell and the blood vessels here through different adhesion mechanisms. And you can actually use targeted therapies to target these adhesion events. And we have again shown over the years, Utku, Ellison, Eilish actually worked on these projects. We have shown that by using anti-adhesion molecules such as anti-P-selectin, you can actually prevent these adhesion molecules. You can use these strategies to prevent adhesions, or you can actually reverse the adhesions, prophylactic or acute type of methods, essentially to manipulate or to reverse these events. We have also shown that there are actually more complex mechanism that takes place that involves adhesion and blood viscosity. For example, again, a PhD student in our lab, Erdem and Erina, they have actually shown that ICAM1 adhesion essentially ICAM is an adhesion. ICAM1 is an important adhesion molecule that is expressed on the surface of the blood vessels. This adhesion type between sickle red blood cells and the endothelium is mediated by fibrinogen. Fibrinogen is a protein that increases blood plasma viscosity. So if you have high levels of fibrinogen and ICAM1 expression on the endothelial surface, then you can actually have high adhesion levels. And hypoxia makes these things worse, and also associates with clinical states, hypoxic clinical states in these patients. So this part of the presentation is a little bit more clinical, but it is important to understand how these fundamental changes in the hemoglobin and the red blood cell impacts human health. And you need to make a connection between the fundamental biology and the clinical state, and see, how the improvements that you make can actually improve the disease at the end. So one interesting engineering problem for us was the density. So if the ion transport due to lack of ATP is compromised, then the red blood cells will be denser. So they will lose water and their density will increase, right? So, and the question was, how can we measure this? There are no methods actually in the available right now to measure single cell density accurately or precisely. It's very challenging if you think about it. We have actually developed a new method with our collaborator, Dr. Gözde Durmuş at Stanford, and Utku Göreke is a PhD student in our lab. They actually work together to develop a magnetic levitation-based microfluidic density measurement method. And this paper was just published in Lab on a Chip in 2022, and it's actually the cover art in this issue. So if you're interested in density measurement, you can actually go check the Lab on a Chip journal. It's this month's, like this issue's cover article, how to measure single red blood cell density precisely. We have actually shown that in sickle cell disease, there are dense red blood cells as shown here, less dense cells. This is, for example, a patient who has been transfused with healthy blood, and you can actually see the different groups of red blood cells with different density levels. So these cells in the red circle are the denser cells, which cause the biggest problems in the body. So they are the densest cells that lost all the water in their cytoplasm, and they are now very stiff and dense. And another aspect of red blood cell health is the integrity, or how intact the membrane and the membrane skeleton is. And if the red blood cell, or any cell actually, if their integrity is compromised, then they will actually, their surface, their membrane will start vasculating. It essentially means that the membrane breaks into smaller pieces, and these are known as extracellular vesicles or exosomes. They can have different sizes, like from nanometer diameter to micrometer diameter, and this is not something that you want. And these products, hemolysis products and these vesiculation products, can also affect endothelial health. And because of excessive hemolysis and extracellular vesicle release, most patients with sickle cell disease, they lose their spleen early in their life. Spleen is the organ that captures all the unhealthy red blood cells from the circulation. And if most of your red blood cells are unhealthy, then the spleen is overwhelmed. Spleen cannot really, you exceed the spleen's capacity to, remove red blood cells from the body. Spleen swells, and it is removed by surgery. So if you don't have the spleen right now in your body, then all these things will happen in the circulation. You will end up with all these hemolysis byproducts, which is really bad for the circulation. And we have also studied over the years how the membrane integrity in the red blood cells compromises deformability and essentially the red blood cell health. And we have actually looked at many things on the membrane skeleton, but the most abundant, the most important protein turns out to be band3, and band3 is the most abundant protein on the cell membrane, in the red blood cell membrane, which is an important transport molecule, and it plays a role in hemoglobin polymerization, reduced ATP, dehydration, oxidative damage. So we want to focus more on that. And in collaboration with Purdue University and Dr. Phil Lau, who is a very well-known researcher in red blood cell and band3, we have shown that a tyrosine kinase inhibitor can be used to target band3. In other words, band3 is actually overphosphorylated in sickle red blood cells, which means that it is overactive. When proteins are phosphorylated, they become active. And if a protein is overactive, it might actually do more than it is supposed to in a certain situation, and that's not always good. And we have also shown that deoxyhemoglobin band3 binding together with band3 overphosphorylation compromises red blood cell membrane integrity, deformability, adhesion, and other effects. And if you target this overphosphorylation, you can actually improve red blood cells. So there you have a molecule, a target mechanism, a mechanism that you can target, and actually it can improve many properties of the red blood cell. So this is something that we have done and published in British Journal of Hematology. If you're interested, actually, you can take a look at this work. Now, let's take a step back and look at a few other aspects of hemoglobin. And based on all the things that we have learned, how can we now develop better cures, better treatments and cures that actually address the fundamental problem in the red blood cells, which is essentially its oxygen affinity and polymerization? Can we actually use genetic methods, genetic gene editing methods to cure these problems? One important observation that we should make here is the difference between adult hemoglobin and fetal hemoglobin. Fetal hemoglobin is the hemoglobin that we are born with. Babies, before they are born, while they are still in their mother's body, they actually have a different type of hemoglobin known as fetal hemoglobin or hemoglobin F. Fetal hemoglobin is quite interesting because it has much higher oxygen affinity, so it is easier for fetal hemoglobin to capture oxygen. And adult hemoglobin, that's the mother's hemoglobin, has lower oxygen affinity, and the difference between the affinities of these two hemoglobins makes it possible for the baby to essentially retrieve oxygen from mother's blood. Like, think about it, the baby in the womb doesn't really have access to the lungs or the outside air, so it collects oxygen from mother's blood, and this is only possible because baby's hemoglobin has higher affinity. Okay, so it is a different type of hemoglobin that the babies are born with, and after they are born, in the first few weeks to months, all the fetal hemoglobin, which is essentially the alpha and gamma chains that's shown with the blue line here, the gamma chain production goes down and fetal hemoglobin production goes down and adult hemoglobin production goes up. So it takes a few months for the baby to fully produce adult hemoglobin, switch from fetal to adult. Okay, so this is quite important, and it has been used as a really smart strategy to cure hemoglobin disorders. I will explain that next. Another important observation that the researchers did over the years was some people in India, they have another mutation that affects hemoglobin production, and it essentially increases or keeps the fetal hemoglobin production at high levels, like higher than normal levels. And if you have and this mutation is known as hereditary persistence of fetal hemoglobin, that's essentially in these people, fetal hemoglobin production doesn't go down, so they keep producing the fetal hemoglobin. And if you have this mutation and it also sickle cell mutation, which is rare, but quite a bit, it's quite large numbers in India, considering the population of India. If you have both of these mutations, then you actually can have a moderate type of sickle cell disease. And this actually has shown people that if you have fetal hemoglobin in your red blood cells at the same time with sickle hemoglobin, then you will have a milder disease. It's almost like you have no disease. And when they did the research, they realized that fetal hemoglobin actually prevents the polymerization of sickle hemoglobin in the red blood cells. And that actually improves the disease state. So the idea was, like, if you combine all the things that we have learned so far, can we induce this mutation in people who have sickle cell disease by genetic therapy such as CRISPR-Cas9 gene therapy, and then have a milder phenotype? So that's one of the approaches of gene-based therapies in curing and essentially curing and having a definitive treatment of the disease, like forever. And this is known as reversal of hemoglobin switching. The idea is, can we trick the nature to produce some fetal hemoglobin instead of the mutated sickle one to essentially achieve a cure? So this typically happens, in adults, like younger, older kids and adults. And the idea here is, can we target the BCL11A transcription factor that silences fetal hemoglobin production? Can we go delete it with CRISPR, for example, and then let the body produce more fetal hemoglobin? And when you have a mixture of fetal hemoglobin and sickle hemoglobin, then you would have a milder phenotype. So that's a very interesting concept if you think about it. I won't go into the details of how that is done. It's a very interesting and a really huge field. If you're interested in genetic therapies to cure diseases, you can actually search and find many presentations online and publications. But I would like to focus on, as engineers, what is our role? Like I talked about lots of biology, hemoglobin, red blood cells, and like the fundamentals of the disease state, and some of the work that we do in those areas. But what can we do as engineers to help these diseases better? And one of the things that the engineers are needed now is essentially improving the accessibility of these genetic therapies. So they are still in clinical trials. They are not approved yet for use by the general public, but these therapies are quite complicated. The way they work is they collect blood-forming stem cells from the body, from bone marrow, from a patient, and then genetically modify them in the lab using lentiviral vectors, for example, viral vectors and other transfection methods. And then you culture them, you grow them, and then you put them back into the body. And this process here that happens outside the body is very complex, requires really expensive infrastructure, expert people who can do these type of things. So it's very expensive. And it actually requires very complex and expensive infrastructure, as I said earlier, like complicated instruments, expensive instruments, and it actually can cost millions of dollars to do one cycle of this therapy for one patient. So it can cost $1 to $2 million per treatment, which is very expensive. And another issue is these are not in vivo delivery. It's not like the vaccine. Like we got most of us got this mRNA-based COVID vaccine, right? So it's essentially an RNA that you inject into your body with a needle, and then your immune cells are programmed to fight against the virus when the virus comes into the body. Can we actually do something similar? For example, can we have an in vivo gene editing strategy to go and edit these blood-making cells in the bone marrow? And some questions remain. For example, bone marrow, if the bone marrow is not healthy, which is the case in these patients, how can we reach the bone marrow and deliver the therapy to the bone marrow, right? So there are lots of engineering problems here, drug delivery, biomanufacturing of these cells, genetic modification of these cells in the lab environment. There are lots of questions that remain. Another aspect of this, the gene therapy research, is resource limited settings right now. Like as I said earlier in my presentation, most people who live with these mutations, genetic mutations, live in low and middle income countries, in Africa, India. And currently, even though there are clinical trials to develop gene therapies, none of them are available in those regions. So these regions are actually excluded from the research, the clinical studies, clinical trials, and that's a problem. Like 99% of the people who need these diseases live in the blue regions that are shown here, but then they are not even included in the research, so that's an issue. For this reason, we actually established the Global Gene Therapy Initiative, and we are working with different organizations in the world to start HIV, sickle cell thalassemia gene therapy trials in Africa and India in the next few years. And we are a part of this group to develop technologies for this purpose. So if you look at our precision medicine approach in sickle cell disease, like combining everything that we have learned so far, it actually starts with the screening and diagnosing this disease to begin with. And there are actually certain treatments available right now that are more affordable, accessible. For example, there are pill-based therapies, hydroxyurea. They are not very effective, but they work well. And it has been used by millions of people. So it is widely accessible. Another option is red blood cell transfusion, which is, again, accessible, but it works for some people, not for everyone. The more advanced therapies that are being developed right now in sickle cell disease are adhesion inhibitors, hemoglobin modifiers, and the PKR activators that are shown here, and gene therapies. If you look at the prices, screening can be achieved really in an affordable way, and we have developed Himex Health and Gazelle technology to address this need. Essentially, with the diagnostics, we can support the screening efforts in sickle cell disease, and also we can improve the management and monitoring of these widely accessible treatments such as hydroxyurea and transfusions. So Himex Health and Gazelle platform right now is focused on those, like improving the screening and accessible, available treatment methods. We have started the Biochip Labs. It's a company located in Cleveland to support the in vitro preclinical studies, like the drug development studies, essentially, and also clinical trials that are ongoing with these new drugs. That essentially measures how these therapies are working, and can we actually develop better therapies by mimicking the disease on a chip, essentially. And another direction that we are focused on is for these hemoglobin modifying and gene-based therapies, can we develop companion diagnostics? Can we develop tools that can measure the improvements that these therapies are resulting in? So that's another focus area that we have. So now I would like to focus a little bit more on the engineering aspects and translation and commercialization aspects of these efforts. The first thing that I would like to share is the idea that market essentially defines how much you can raise and how much time you can have to make a product. For example, if you think about all the different companies and the technology that I presented in the previous slide, you need to spend time and effort and money to develop these technologies. There is the research aspect that happens in the lab, but when the technology leaves the lab, it still needs a lot of time and effort and money for it to become a product. That's known as the product development. And the product development is essentially defined by the market. That's the idea here. So for example, if you look at point of care diagnostic devices as an example as a case study, almost like a case study example here, it would, in the United States, it would take about $34 million and 6 years to develop such a product on average. And if you are raising money to develop such a product as a startup company, for example, then you need to raise the money, raise this much money, and then you need to have 6 years to develop this, like 34 million and 6 years. And if you do the math with a conservative return on investment, investors would want to make money, right, from their investments, and they would want to see a large market for them to make this $34 million investment. So your market size should be at least 10 times this, to 340 million, for example, if you are considering a 1 to 10 return on investment. So that's a rough calculation, rough math, but it can give you an idea about how much you can raise considering the market. So the market actually defines, that's why the market size defines the amount of money that you can raise and the time you have. Okay, so it's important to know the market. It's important to know the potential market for the product that you are developing. Another idea that I would like to share with you is when it comes to product development, like commercializing technology, the ideas are not that important. Actually, there is this nice quote by Scott Adams that good ideas actually have no value because the world already has too many of them. And the market actually rewards execution, not ideas. So as engineers, as inventors, it is very easy to come up with new ideas. Like you can come up with ideas, like 10 ideas in a day. But what matters is you focus on an idea that makes the most sense and you work on the execution of that idea. How you take it from the idea stage to a product stage or a device stage. How do you commercialize it, and what are the things that you do along the way? Okay, so these are really important take-home lessons if you are interested in translation and commercialization. So if you look at the traditional translational pathway that most people follow or most companies follow, it would look like this. You start with an unmet need. So you do your research, you talk to people, and they say that there is this problem and we need to solve this problem. So there's that need. And what most people do is they go and invent something and create a prototype of a device or a technology. And then they take that prototype and do some preclinical studies, like studies in the lab with animals and so on. And if things look good, then you advance to the clinical studies. You use this technology or device with people in clinical trials or clinical studies to see if it is working. And if everything looks good, then you focus on manufacturing and you follow certain quality standards to manufacture this device. And then you try to get regulatory approval for this device and then launch it to the market. And like, just imagine this, you do all this work, you spend all this time and money, millions of dollars and years, for example, and then you realize that there is not enough market, right? Nobody wants to buy it, or the market is not clear, or maybe you anticipated like $1 billion market, but only 10 people are interested in buying it, right? So that would be a disaster. So if you don't have customers for your product, then you don't have a product essentially. So what we do essentially to to make this process more equitable, more accessible, we made some modifications to this process, and we call this adaptive innovation and accelerated translation approach. And our goal is to minimize the funds raised, minimize time to market, and focus on need-centric and problem-oriented technology development. And we actually follow a more informed, disciplined, bottom-up approach. It's not like you invent something, an idea comes to your mind, and then you are excited about it, and you go do the work. When we come up with an idea, when we identify an unmet need, we find ways to discredit that idea. We also, we try to discredit that idea and the unmet need by talking to people, by doing research. And if you do everything you can and still the idea holds, the unmet need holds, the approach holds, then it's a good approach. So what we essentially do is while we look at the unmet need, we also look at the market, potential market. Like for this unmet need, for this product, what is the market size? It can be small, it can be large, doesn't matter. But it's important to be aware of the market and the reimbursement, who is going to pay for this, and the IP space. Are there patents in this space? Customers, who are the customers? Is the customer the patient, nurse, the doctor? Like, who is it? And it's really important to look at all these aspects before you basically focus on an unmet need and develop a solution for that unmet need. Another thing that we look at is, especially in the invention prototyping stage, we look at the regulatory considerations. How will this device or technology will be regulated? Who is going to approve it? You cannot just develop something and go use it with humans. Some government bodies such as FDA, WHO, they look at it, CE mark, they look at it and they actually review it and approve it. Another aspect that we look at is manufacturing. Early in the development phase, in the invention prototyping, can we take into consideration how this device or technology will be manufactured for accessibility, for ease of use, and so on? And if you can actually bring all these aspects to early on to your invention and development phases, then actually it helps you down the line. Another aspect is, can you actually start the clinical studies as soon as possible after you do your prototyping? Keep the preclinical trial studies short. Essentially, if you have to do animal studies and in lab studies, can you keep them as short as possible and then start the clinical studies immediately? Like, you need to just go and do the real work as soon as possible so that you can actually figure out if the solution is working or not. And if you are actually developing solutions for Africa, India, you need to engage local experts, clinicians, regulatory experts as early as possible. So if, for example, if you do all your work in the United States and then go and try to, commercialize it in Africa, then it may not work. And while you do all these things, if you can actually also develop a quality management system like ISO standards, then you will have a much shorter time to market. So you can actually do this in a much more informed way. You don't just invent anything that comes to your mind. You don't, come up with an idea, get excited about it, and without thinking about the downstream implications, you don't start working on it. So this is what we try to do in our lab. If your goal is to translate and commercialize, you should be focused on the downstream events. Most of the academic labs are stuck in this first two steps. Most academic labs, they come up with an unmet need, they invent something, they prototype it, they publish it in a journal, and most of them don't even go to preclinical. Some of them go do some preclinical work. Some of them do some clinical work, very limited, but most people are stuck in the first few steps. Like 95% of the academic research is stuck in the first few steps. They cannot really push their research and technologies to the clinical study, manufacturing, regulatory market, because they don't do the things that they need to do early on, essentially feeding market into unmet need, feeding regulatory to invention prototyping, manufacturing to invention prototyping. If you don't feed those downstream requirements and considerations to the early on research activities, then the likelihood of success in commercialization is very low. Okay, so this is our approach when we do these type of things. If you are interested in this type of work, you can actually go and read this article that we published recently. So this is a retrospective case study of successful translation research. So we have translated a device, Gazelle hemoglobin variant that's used for diagnosing sickle cell disease, anemia, and so on, that's available now commercially worldwide. You can actually go and read this article published in Journal of Clinical and Translational Science in 2021. And it will give you a timeline and our approach, on how we do this. So you can actually see a figure from that article here in this slide. So some of the other things that we work on with HEMEX, for example, is we have a platform technology that actually allows us to do many other things. So it's important to focus on platform technologies if possible, so that actually you can build on it. You can actually come up with new diagnostics like COVID-19, for example, that is now working on the Gazelle system. You can actually develop technologies for other things, like diagnostics for other things, infectious disease, anemia, respiratory diseases, and so on. So that's the idea of this slide, is to have a platform technology if possible, so that you can build on it. So this is most of our team who did all the work. You can actually see that we have a highly diverse team from all over the world, and these are the students who have been actually working with us on doing all these projects. And this is all the support that we have received from different organizations, companies, government organizations, foundations who support our work. And this is the end of my presentation. Thank you very much for your attention, and I would be happy to have any questions if you have them.
Okay, thank you for your nice presentation. We have one question.
Yes, please.
Okay. What did you find most difficult while carrying out your projects?
Yeah, so it's a good question. So the most difficult aspect, actually, let me tell you this first. If you care about what you do, and if you are really passionate about what you do, then nothing feels difficult, right? So it's not, everything is difficult, but it doesn't feel like it is very difficult to you. Like it is, it's challenging maybe, there are challenges that we have to, resolve and we have to address, but I wouldn't say that it's difficult. If you are ready, and if you work hard, and if you are prepared, nothing is difficult. They become easy. But you face challenges as you work on these things, like challenges, limitations, problems along the way. I think the biggest challenge is for our students, for our researchers in the lab, finding that unmet need that makes sense. Finding the clear unmet need and defining that unmet need is the beginning of everything that we do. So inventing things is easy. Developing things, prototypes, devices, those are easy things. But if you are not working on a real problem, a real unmet need that people care about, then nothing else matters. So many of the academic researchers, many of the around the world actually, they actually, people come up with certain unmet needs, and they think that it's a big problem in their minds. They think that they found an unmet need or the problem, and they spend years and decades working on it. But in a broader sense, if that unmet need or if that problem is not acknowledged or shared by many other people, then it's not a problem or an unmet need. So identifying the unmet need is probably the most challenging part of the project.
Okay, thank you. I agree with you that point.
Actually, I have a question too. You won lots of awards like Rising Star. What did you think of the most important factors affecting your success?
Yeah, so that's a good question. So again, working on problems that matter. Like we don't define the problem, and we don't define what we should really focus on. we can choose, but those things are actually defined by others, like defined by, children living with sickle cell disease in Africa, for example. Focusing on things that matter the most. So that's number one. Number two is collaborations and mentorship. So, working with many researchers from all over the world, from with different backgrounds and different interests, being able to work with a wide variety of researchers from different disciplines, with different experiences, different academic preparations, mentorship and advising these different types of people, it becomes very important. And many of these awards that you mentioned, they value primarily two things, in my opinion. Are you working on a problem that solves real problems of the people? Like, are you working on something that would solve people's problems, like unmet need, essentially? And while you are doing this, are you training and educating and mentoring the next generation of researchers and inventors? So if you are working on something that's really important, and if you are teaching other people how to do this, then these two things create the biggest impact. Like, number one, if you are working on a real problem, then you may actually end up saving people's lives, and you might actually improve human lives. That's very valuable, important. And number two, you are actually training the next generation of inventors. Like, I cannot do everything, and I will not be able to do this forever. But if you actually spark lights in other people, and if other people, your students, trainees, become interested in these type of activities and in this type of research efforts, then you are actually growing the number of people who can do this type of things, right? So mentorship and working on real problems are the two reasons why I think I got those awards.
Okay, thank you for your answers, and thank you, of course, for your attendance too.
Thank you. So, any other questions? Are we all done?
And I guess time arrangement is finishing.
Okay.
We have a lot of questions.
Sorry, Ezgi, I cannot hear you.
We have a lot of questions from our participants, but we don't have enough time. I'm sorry for that.
Can you send me an email with those questions? I'm interested in seeing what people are asking. Okay.
Okay, I will send you, Doctor.
Thank you very much, and thank you for your invitation. I hope you enjoyed my presentation and you learned some new things. So it was a pleasure to be here.
Thank you, sir. Have a nice day.
Thanks
for your
time. Thank you. It was a very useful presentation for all our participants.
Thank you very much. Have a good day.
Okay. So next 5:30 p.m.
Hello everyone. This is our sixth session. I hope you're enjoying these events. I'm going to give the word to Ufuk. Hello Ufuk, how are you?
Hello, Ezgi. Thank you. I'm fine, and I hope you are well.
I'm fine too, thank you. Have a nice session.
Thanks. So, greetings, everyone. I'm Ufuk Şen, a freshman at Gebze Technical University, who is studying molecular biology and genetics.
I am Sudeydem. I'm a prep student at Gebze Technical University, and my department is molecular biology and genetics.
So, as you can see, Sudeydem and I will be moderating the ongoing session. Our guest is Professor Metin Akay, founding chair of Biomedical Engineering Department and John S. Dunn, endowed chair professor at University of Houston and studies biomedical engineering and neuroengineering. Professor Metin Akay is also the current president of IEEE EMBS. You may begin, Professor. You have the words.
Okay. Well, first of all, thank you so much for the invitation. Happy Sundays to everybody. I'm delighted to be here. I think whenever I receive any invitation from Turkey, my native country, I am pleased to accept, and I hope that the soon pandemic will be over. We will contain the pandemic, and we will be visiting and interacting. Also, Gebze Technical University, I'm part of the Gebze, and I serve as an advisor to the Molecular Genetics Department, the Research Institute. They have been doing a fantastic job over there. So today's talk, I decided to talk about the more healthcare innovations and entrepreneurship because the world is changing so fast and so quick. And as Ufuk mentioned, I also serve as the president of the largest and oldest biomedical engineering department in the world, with the members 12,000 plus globally. And as well as I serve as the president of International Academy of Medical Biological Engineering, which is a part of the International Federation Medical Biological Engineering. So I will try to share my humble experience with you this afternoon. EMBS, IEEE Engineering in Medicine and Biology Society, was built in 1950s, 52, and then changed the name in 1963. And I will share also where we are heading with the new vision for the society. As I mentioned, we have more than 12 members. I think to me is the most important is the future of the society and the young people, dynamic people, interactive people. They are really the future leaders, and we have currently 2,000 plus student members. I love to see this number reaches to 5,000. We have chapters, student clubs globally, each more than 100, and we have this morning update. We have more than 110 countries participating in our conference in our society. For students, I strongly advise to get involved with the society early on. This is very important for networking, exposing yourself to new emerging areas, and it's also to build confidence. Society sponsors several international summer schools. One of them, I believe the oldest one, biomedical imaging in France and biomedical signal processing in Italy. BioX, it's mainly focused on entrepreneurship and the biodesign, and this year is going to focus on data science. Was held first time in New Hampshire, the Hanover, as a co-sponsored by Dartmouth College when I was there. And then Istanbul in Turkey, we had it. And 3 years ago, we have been holding this conference.
Sorry for the problem, but are you changing your slides?
No, can you see the International Summer School on screen?
We are still seeing the first page of your PowerPoint. It may be because of the when you full screen your own presentation, sometimes Google Meets have as error like freezing.
Okay, let me go back again. Let me do the window. How is I now?
We can see now. Can you change the slides or backward? Yeah.
I know. Okay.
That
This
well.
is summer school I was talking about. This year, first time we are going to have it the data science space. It will be held in Hania, a beautiful part of the Crete, and the medical devices, biosensors in Hong Kong. This is also very innovative device-oriented summer school, and the neural engineering. First time we had in Shanghai, and computer models in medicine. Journals. Our society sponsors 13 journals. 3 of them are gold open access. One of them actually is a very brand new journal, is the IEEE Open, the Journal of Engineering, Medicine and Biology. The founding editor-in-chief, Paolo Bonato. This is completely online journal. I think we don't have the impact factor, but we are going to have it this year, at the end of this year. Other journals here, Translational Engineering and Health Medicine, Transaction Neural System Rehabilitation. As also we have several co-sponsored publications. One of them, Transactional Medical Imaging, I believe, which has the impact factor 12 right now. And there are several others. And of course, our flagship journal, Transactions on Biomedical Engineering, and the Journal of Biomedical Health Informatics is impact factor is close to 6. The current editor-in-chief, Dimitris Fotiadis, is also doing an outstanding job for this journal. We organize conferences globally. If you look at the screen, and it goes all the way, and we also had this conference 2001 in Istanbul, Turkey. I hope that one day we will come back to the Turkey or the region. And the last one was at Guadalajara. And this year we will be hopefully face-to-face in Glasgow. And in 2023, we will be in Sydney. 2024 will be in Orlando. And 2025, we will decide this year. Society has several technical committees, and we open these committees to junior and also the student members. They may not have vote in the committee, but I want your voice to be heard, which is very important. And also it gives a chance for recruit the next generation leaders. I think that's the motto for the society. This is Dimitris Fotiadis, who's the editor-in-chief of Transaction Biomedical and Health Informatics. One thing that's very important for me, I hope that its slide is there, is the diversity inclusion. I think it's a moral and ethical responsibility to promote women and minorities in the field of engineering and science, and I'm highly committed to have equal number of the keynote and plenary speakers at our conferences, at our journals. As a society, as the executive committee, we are keen to follow up these procedures, and I think we will prevail. It's their commitment. They have done tremendous to move the society to next level. We should continue and having them in our board, and we have to increase their involvement, their promotion. We should do everything in our power for this. We have also the Best Chapter Awards every year at our conferences, and for Ecuador was one of the winners for the conference in Canada. I think it's I strongly encourage your chapters to be nominated or to nominate. We can check the procedures. I think you have very dynamic chapters in Turkey. I became president beginning of 2021, and I look at our mission and vision statement. The society has been doing great, and the new leaders, new leadership executive committee, we decided to have the focus on more healthcare innovation and I'm going to share that what we have done 2021, what we are going to do in 2022 as well. First of all, our mission is to foster, to promote the biomedical engineering innovation and research and education globally. We are a non-profit organization. I believe that our society, also the culture and the ecosystem of our society is extremely welcoming, and we care about the members and we care about the students. I think this is, again, encourage you to get involved. I think mention is something touches my heart, the promotion of women and the diversity inclusion globally. Our society really appreciates the role of the women scientists and engineers. They were extremely crucial for success of our society since the beginning of our society, 1952. As a president, I'm fully determined to continue to promote female scientists and engineers. I will do everything in my power to get them involved with our conferences, publication, not just attending, but playing leadership role for our conferences, for our journals, for the technical activities of our society. The action item was the public forum. We decided to have the public forums in emerging new areas, open up this public forum. It was free of charge, not only society members, but general public. So the new initiative was to really share knowledge and have very fruitful discussions about urgent healthcare issues. And then discuss about how engineering innovations can really be translated into the practice to have effective healthcare solutions to these grand challenges. I believe that these forums are very useful. It's a very unique platforms for not only engineers, but also scientists and the clinical practitioners, students to focus on healthcare innovations. I also believe that there is an urgent need for a paradigm shift in healthcare because the engineering innovations are indeed really help to economic growth. It's also, I believe that these forums have exposed our students to many career and networking opportunities in the global healthcare market. First one, we focus on the COVID-19 healthcare screening, tracing, and treatment. And it was held 2020 at the end, and it may be one of the most comprehensive forum because we had 4 days, every day is 4 to 6 hour presentations, and from the expert in the field, from the healthcare part, screening and tracing and treatment part. We had new papers published by the IEEE transaction on biomedical and health informatics, and I can send a copy of to Ufuk to share. And also we have the professionally interviews with a few selected speakers, and video is available. I will send it to you, and we will work on this. Second initiative was the focus on data science and engineering in healthcare. When I look at the jobs in the field of biomedical engineering, healthcare areas, next at least 10 years, we will continue seeing an enormous, enormous growth of the jobs in the areas of data science and engineering in biomedical or biomedicine or healthcare areas. Therefore, we decided to have second forum, again, 4 days on data science, and it was including the fourth symposium, including medical imaging, precision medicine, digital health, and brain and neural systems. Also, all these talks we recorded. These recordings are available for our members. And if you have a hard time to reach, send me email and we will send you the correct link to look at. This form, as I mentioned, is the for the symposium. They are co-chaired by the leaders in the field. And as you can see, the names are posted there: medical imaging, Andrew Lane, Amir Amini, and the precision medicine, Colin Brennan, Ali Tnazlı, digital health, Paolo Bonato, Jeff Palmer, and the brain neural systems, Erika Ross, and Paul Sajda. We had actually 36 speakers, 36 keynote speakers. They're from leadership, from industry, research institute, academia, and also these are the number of the attendees, and it's almost 2,000 people attended. The third forum we focus on about the healthcare industry. When we have conferences or when we have events in our department, we always pay attention to have the industry representation. Reality is, most of our students, they get a job in companies. Some of them, they decide to go 15, 20%, go to medical school, and some they continue to graduate school, 30% plus, and the remaining, they get job in industry. So it's very important to listen the trends in the healthcare technology. So therefore, we devoted one more public forum, again, 4 days. The focus on innovative medical device industry, digital health industry, biopharm, biotech industry, and the neurotech industry. Unfortunately, we do not have the videos for those, but we are going to have position papers and also the interviews with some of the speakers, and they will be released very short, soon. I will share with you also when it's available. We have 26 industry leaders. Some of them are CEOs of the companies, some of them vice presidents, research directors, or the chief technical officers. They were there. And for the healthcare, the industry, and I had a fantastic people. They were working in organizing, and the Dr. Winevsky, she's the CEO, co-founder of the Prof USA, Carl Brennan, CEO of the Cell One, and the Ali Tnazlı, Biospin right now in Europe is the CEO, Jacqueline Poe, CEO of Enlargement Health, and VG2 CEO Bruce, Erika Ross, the research director, Abbott Lab, and Giorgio, president of the Platon Associates. I'm not going to go to all of them, just to give the flavor. This is the medical device, and we have the GE leadership, Netflix, Medtronic, Material, NIH, and the Blombertech. We go fast here. But one area that I would like to bring your attention is the digital health is especially based on smart sensors, smart variables, and the second area is the smart analytics. I think is going to really help to improve the quality of the healthcare and also reduce the cost of the healthcare. Also, they provide an incredible access for the patient remotely. And we have, if you look at them, all of them are highly accomplished, despite they seem to look very young people. And Steve is the founder of the Cyber Health, is also MD degree. Wellbeing Digital Cow is in Hong Kong, and Heidi is in the president of the Tour. It's just the vision and also the dedication and the resilient people. I think they're going to move to healthcare into the next level. See the biopharm, biotech industry groups, and also we had a lot of the leaders. I would like to take your attention somewhere else. If you look at most of our speakers were females. I think they were highly achieved and the leaders in the field, and they serve the CEOs, founders, or research directors or involved heavily with the research activities. Some of the colleagues, for example, like David White, they are actually professors. He's the professor at MIT, also has the company. I think this is a new thing that to translating the research into the new field is very, very important. We also build the, I decided to have a special distinguished lecture programs. These distinguished lecture programs are the great leverage for us to reach out to the community, or if you are organizing conferences or even the student conferences or workshops, and your chapters can invite our speakers, and the society partially sponsor their activities. I think it's a great opportunity for our chapters, and we have the colleagues from the Abbott Lab, the Dr. Ross, and CEO of the founder, and the Triona is in Europe, and the Anita, she is the head of the life science strategy at the HP, and the Natalie I mentioned, she's the founder and chief technology officers of the Pro USA, and Jadwiga, she's the director and head of the computational biology of the Pfizer, and Dr. Tnazli, again, he moved from California to Germany recently, and he's brilliant mind actually. And Mike leads the vice president for the corporate technologies and innovation at the Medtronics. Okay, let's talk about this is the slide, is actually an old slide I borrowed from the World Health Organization to look at which kind of disease is continuing impacting our daily life, our health, and also our economy as well. If you look at the first 4 diseases at the top, the highest cause of the deaths globally, they are all chronic disease. And it's mainly cardiovascular, respiratory disease, chronic obstruction of the pulmonary disease. You don't see a pandemic there, but it's the pandemic is a part of also the infectious disease, and is also related to chronic disease, is affecting mainly the immune compromised the people globally, and also the people, they do not have an access to clean water, the isolation. It's affecting mainly the lower income the people. So the engineering innovations can be great help to really translate into the developed technologies, help to reduce the chronic disease, and they reduce the burden in the hospital, and they give the freedom to patients at home environment to take care of at least some of the activities. I think it's a great help for this reason. Second biggest growth is the cancer research. As you can see, the cancer has been the global disease. Lately, we are not talking so much because of the pandemic, but there is a lot of efforts globally to develop vaccines. In fact, the BioNTech, the groups, they were heavily involved with the vaccine development for the cancers before the pandemic. There are other groups, they are working in that area. The third one, to my best knowledge, is we have unprecedented level of neurological disease, because thanks for the technologies, we live longer. Life expectancy significantly improved globally, and especially in developed countries. And we are talking about somewhere 80, 85 men and women respectively, the years. And the developing countries, this number could be slightly lower, but to me, it's very important to quality of the life. So therefore, the new technologies are needed, and the new way we have to look at how we design the drug, and it's very, very important to take care of the neurological disease, because the many diseases such as Alzheimer's, dementia, and are really increases with age, but even the young people may have it, but mainly the older people, they suffer. So Alzheimer's is one of the, still we do not have really the concrete solutions, but there are a lot of great efforts everywhere in the world to really develop the better drugs, better technologies for neurological disease. Biomedical engineering, I also put something inside the healthcare engineering, and I believe that is these two names are synonyms between each other. So the healthcare is and the biomedical engineering are extremely related, and I believe that we will pronounce the healthcare engineering in the near future more and more. The job growth still for biomedical engineering is increasing, and this is some of the reports from the U.S. Bureau of Labor Statistics. This is holding also the other part of the world, and I also believe that biomedical engineering still is one of the fastest growing engineering field. My bias, biomedical engineering is the future of the engineering, because at the crossroad of all the engineering disciplines, biology and medicine. The enrollment for the biomedical engineering program, including the pandemics actually, there's still significant, the number of the applications are coming, and in even Europe, more and more biomedical engineering departments are opening, and also significant increase in the number of the biomedical engineering departments Asia Pacific area. But when we look at the healthcare innovation and market, these numbers before the pandemics, of course, the pandemics affected these numbers slightly, but I believe that we have to look at the positive side. We will contain the pandemic. Human being is great to adapting to new conditions, and we may be living it together. But I think we will contain, and there could be others. The disease may come up, infectious disease, but humans will prevail. Annual revenue before the pandemics for just the medical devices was around $500 billion per year. This is the only investment and the activities in the United States alone. Of course, still the orthopedic engineering and cardiovascular engineering are get the lion's share on this. But the other biggest part is the biopharm engineering, or as I put there, the biopharmaceutical science is almost $1 trillion per year. The highest growth area in drugs and oncology. It's very important that culture also change in biopharm industry. They hire more and more biomedical engineering students. They hire more and more engineering students for develop the better platform to test the drugs. Also, better the computational the programs to test the drug effects for modeling purposes. So when you enter any biopharm engineering, biopharm companies, when you talk even from leadership, you should not be surprised when you see lots of medical doctors, lots of engineering trained biomedical engineers. Of course, you will see also the biopharm background people. This is the healthcare cost per capita. These numbers could be old. I think I know United States is almost, we are in the $10,000 per people per year spending for healthcare. Switzerland, Sweden, Germany, Canada, they spend less money compared to United States. But the United States, the healthcare is not the best in the world. Before the pandemic, the last time when I look at them, healthcare quality was the US ranked number 34 or 35. Of course, the pandemic shows that how healthcare system was not prepared to fight, and also the immediate hospitalization became a huge burden in hospital setting. The countries like Nordic countries, Sweden is one of them, Norway, and also the Switzerland in Central Europe, they spend less money, but the healthcare quality seems to be much better than the United States. This means that spending more money does not mean that you have the best healthcare, but this average number does not really reflect the benefit for the different part of the society. Different part of the society, what I mean, high income, middle class, and the low income. If you look at the GDP-wise, is the U.S. economy slowed down a little bit, but still I believe that we are coming close to $20 billion, to $20 trillion. And China has the largest market, but the GDP is right now is getting closer to 15. Japan, Germany, UK, France, India, and Italy. I would like to bring something to your attention. If you look at India and Italy, their contribution to their GDP is just about $2 trillion or close to $2 trillion. I think we need to be careful that there is no way any country will reach this level unless they produce. They're all they have a significant contribution. I'm just talking about the healthcare device, healthcare technologies, healthcare innovations. Leading countries, not surprisingly, go the same way. United States, China, Japan, Germany, UK, France, goes on. Between India and Italy, there's one more entity. This entity provided or they helped to build more than 30,000 companies, employed 5 million people almost, and also their GDP contribution was $1.9 trillion. Name of this company is Massachusetts Institute of Technology. I'm talking about the university, one university, because this university is the focus is translational research. We do shift from the science is extremely valuable. These ideas into the product, turn these ideas to help the people, turns these ideas to help the economy. MIT, when you look at them alumni, 30% of them, they have at least one or more patents. Almost 20-25% of them, they served on the many advisory boards, national or international companies. They have production, the companies are heavily production-oriented companies. So this is very critical. Anyone is investing on biomedical engineering departments, research institutes, or the ventures between university and industry, they have to pay attention for the translational research. This is University of Houston. I came here 2010 to build a department, and if you look at our building, we are right now we occupy the second floor of this building, and it's the first floor, and we have the clean rooms and also the core facilities. Second floor us, the third floor nuclear receptor center, and the fourth and fifth, if you look at them, more the biochemistry and the molecular the activities. Every floor there is two words are there: biology, medicine, or genetics. So this natural for the biomedical engineering because biology is extremely important. Yes, but it's the engineering is indeed helps to move biology and medicine to the next level. We have also clean room, as I mentioned, also for the characterization and the drug development in the first law. Our motto of our department is indeed really to create next generation leaders. Just not to people to just look for a job, but also play critical role, move the field. Just a couple of things. I just want to acknowledge that this slide is actually slightly modified from our dear colleague, Semahat Demir, and who used to be the program director at NSF and the president of the Culture University in Turkey. To me, it's the most important thing for the next generation engineers. It's very important to have strong analytical skills, and the creativity and ingenuity, professionalism are the must. And the independent thinking is the must to be creative, to be helpful, to move the needle. So take a challenge, because without challenge, to be honest with you, there will be not much in the innovations. And also the cross sections of the field, and it's extremely important. That's why we call the biomedical engineering, right? And the future is going to be on smart biomaterials. Future will be on data science, data engineering in healthcare. Future is going to be intelligent drug design and drug delivery. So those areas are really still need talent, and I think this is a tremendous opportunities for you in those areas. But the engineering brings another level on this engineering innovations. I think it's so the diversify your portfolio. The learning molecular biology, molecular genetics are very important, but adding computational and data science on it, it's going to be extremely valuable. Or adding to have micro nanotechnology, the background and the building platform, not for the molecular genetics, I think is extremely valuable. Our department currently focuses on the three major areas. One is the neural cognitive rehabilitation, covers more or less every areas from neural implants to all the way brain-computer interface. The second one is the bionanoscience, is the regenerative medicine. I believe that we hired, we may be one of the first or one of the departments, biomedical departments, hired the immunologist. Professor Chandra Mohan we hired, the PhD MD, and he's the PhD in immunology. So it's not that it's half of our departments, they have a different background. They have computer science background, they are medical doctors, or they have the molecular genetics. It's just to also the field is changing so much, and we need the people. They do not only focus in one area, but also they have much broader the perspective and the background, and also work with the other people. The third is the biomedical imaging and informatics. Soon we are going to change it to the data science and healthcare. This is Houston. My office is only 3 to 4 miles away from the Texas Medical Center. Texas Medical Center is the mecca for healthcare. As you can see, we have 37 hospitals in this area. It was part of one of the oil companies actually when they started at the beginning of 20th century or late the 19th century. And some of the names, I don't want to go one by one. For example, MD Anderson is one of the most respected research institutes for the cancer research, and is a part of the UT Health. University of Texas actually. Baylor College of Medicine and the both medical school and also the researchers in the Baylor in any areas from the neuroscience to all the way to cardiovascular. A member of the Herman, I'm sure that you have heard, is one of the also the healthcare the providers, Texas Heart Institute, Texas Rehabilitation Institute, and the Texas Children's Hospital, and the Methodist Hospital. On top of, so we have 3 medical schools in that area. Is Baylor College of Medicine, UT Health, and also Galveston is very close to here. But also the 3 the exceptional universities is the Texas A&M has some activities, although they are not the university is not fully located, but some of the biomedical engineering activities are in downtown. Rice University is a private university, and of course the University of Houston. University of Houston decided our visionary chancellor Remy Couture and also the dean Steve Spann, they decided to start a new medical school. It's called University of Houston College of Medicine, focus on more the community-based and more population-based, and to really solve the challenging problem, and it's in the area. So it's also involves heavily is innovation-based medical school. And medical school already had the 2 cohorts, and it's the incredible number of applications in the United States to go to the medical school. You have to have the college degree, and to my best knowledge, for the 35 spots and application somewhere from 4,000, 6,000, because the students' tuitions are paid by the fellowship. Something else happened in the Houston. Houston, of course, has been known, in my view, the best place, best healthcare delivery city in the world. And also some people, they also appreciate Houston is the mecca for the oil and gas companies, because almost 90% of the oil and gas companies headquarters are located in Houston. But it's 2010, the dear colleague, Dr. Robbins, who's a cardiac surgeon, he was a cardiac surgeon at Stanford University, decided to come to Houston to lead the Texas Medical Center as a CEO. He also initiated to have the TMX, the space for translational research and the space for the small and mid-sized companies. There are spin-off companies coming from these universities and also the medical centers, medical institutions, and hospitals. And also it's open all the nationwide and the globally for the small, mid-sized companies to have a hub. So they drastically changed the environment, and I'm going to talk in a minute about how many companies was built. It's surprising, because Houston, the challenge was, of course, there's a significant growth of the healthcare entrepreneurship in the Northeast area, especially in Boston. We visit, Professor Akay and I, we visit the Boston. Each time when we go, it's really surprising that is around Harvard, MIT, Boston University, there are much more small and mid-sized companies. There are always the spin-off companies and also the companies between industry and university. And also the California is the same. Of course, the California is in general, is especially Bay Area for the general, for technologies but lately you will see more and more biotech companies in San Francisco area. And I think San Diego is booming, and between LA and San Diego, you will see several hundred small, mid-sized companies. They're all spin-off companies, or they build jointly with the industry and university. The number is all on the screen, and I believe that we have more than 300 life science, medical device, and digital health companies in Houston. It's inside Texas Medical Center, located at is known as the TMX, we call, for the foundries for the companies. They are very mixed companies, and it's roughly is the 1/3 is medical device companies, 1/3 intelligent drug design, drug development companies, and the 1/3 digital health companies. And these companies are at every stage. I think it's the Johnson & Johnson has the very large footprint in that area, and they have the innovation center, one of the innovation centers, and I think they occupied is close to 35,000 square foot space. And of course, this is the early part of the 100,000 square foot space for the TMX. Is right now is, I believe that is about 200, and they will continue expanding. You will see a lot of cohorts, and from again medical device, drug design, digital health. Is AT&T has the foundry. In addition to TMX, the University of Houston, our university, again, I would like to acknowledge the incredible effort and incredible support by the Chancellor Qatar, and we have more than 30,000 square foot space just for companies. Okay, and we will continue to build, to expand. In my department, currently we have 18 faculty, and the yearly the research funding per faculty, we are well above $400,000 per year, and the incredible publication records at the Science, Nature, or other leading IEEE journals. And is our 4 faculty, they have their own companies, and we are going to, they have the new faculty we are hiring in the areas of the more digital health, and they are coming from industry background. Of course, we will continue have the more companies. In fact, one of the company was built the Professor Y.C. Zhang and his students, and they are receiving $2 million right now from the NIH for this. I think it's a warning for me, I guess, right, Ufuk? How are we doing time-wise? Can you hear me?
Yes, we can hear you.
Okay. Do I have another 5 minutes, or do you want me to stop and get the question?
Yeah, you have 5 minutes.
Okay. Let me talk about the one area, the brain engineering is I hope the videos will work. No, it's not going to work. I think it's I would like to bring your attention the areas of the neural, and there's a significant effort and funding available from the United States NIH or the DARPA or NSF in that field. I think it's going to really have immediate impact on people's life, and also the easy to translate, and it's going to be very important for the healthcare. And this is the young lady, as you can see, she lost the upper limbs, and instead of using brain-computer interface to control the neural prostheses, the brilliant ideas from the colleagues from the Case Western and also the Northwestern universities, they decided to use the body computers. So they are using EMG activities and to control the neural prostheses, because the EEG signals highly likely can be contaminated by noise, but EMG is sort of like enhance the brain signals coming through the brain, and as long as these neurons are innervating the muscles close to the surface, and this data, which we call the signals we call EMG, electromagnetic signals, can be detected, can be used to control the neural prostheses. Neural implants is also another key growth area. As you can see, this is very traditional, the silicon-based systems to place the electrodes on the surface of the brain to record the activities. And the neurons size can be different, axons can be different. It depends on their function, different part of the brain. Here that mainly we are talking about somatosensory cortex area. And we know that even by looking single neuron, we could get some idea. Of course, we cannot completely link the one neuron activity to the behavior. But this is the monkey, is the right-hand side, trying to reach the food. And we are also recording at the same time from single neuron in the brain, in the cortex. When the animal reaches the food, as you can see, the firing, neural firing is significantly increases. Of course, the system is old system, bulky system, but this new platform developed by the BrainGate, as you can see, fully the implantable and the wireless system, and we could get a very interesting the recording from that. I think it's let me stop here. I don't want to. If I have.
Professor Metinaka, you have still 3 minutes left, and
okay.
if you may, we can have a question.
Okay. Can you see it right now, screen? Okay. This is what we have done. This is a joint research. Professor Ohta. I'm sharing. You cannot see it, right?
No, we can see the file.
Window. Right now?
But yeah, we can see now.
Better?
Are you changing your slides?
I did not. I will try now. Can you see it? Changes?
No, we can't see any changes.
Okay. On the screen, do you see the implantable microimaging device?
Yes, I guess. But if you may, can move on with the question and answer parts.
No? No changes?
No changes.
Okay. I think maybe let me try one more time. There's one slide I really want you to guys see it because it's stop presentation. Let me do it one more time. Window. This one. Okay. You would do you see it?
Yes, we are seeing your presentation now.
Okay. I think it's if I go to the full mode and still.
And no, okay. When you go on full mode, the screen just is freezing.
Okay, so you can see the imaging device, right?
But yeah, we can see the device.
Okay, so let me tell you something about this. Normally, we record the neural activities from brain using multiple channels, and our group is a joint effort with Japan, Professor Yasumi Ohta, Jun Ohta, Professor Yasemin Akai, and I. We have been working with them. This developed technology is called the microimaging device, CMOS. We are entering the whole system into the addiction center of the brain, and we record from thousands of neurons at the same time. So the system has, as you can see, is an imaging area. The size we are talking about 0.3 millimeter to 0.6, almost 0.7 millimeter area. It's almost the size of the ventral tegmental area. We have also developed a new technology, and I'm unable to share with you right now. It's in the imaging corners areas. We can also record the electrophysiology, single unit activities of the neurons. At the same time, we can image them. So I think this is also the part of the innovations and our group we are working, and the Professor Akai and her team, they have developed the brain cancer chip and for GBM. This one is was the highlight actually of the IEEE, because our paper was ranked the number 2 out of 5.4 million papers in the summer of 2022. So the all credit goes to her, and Professor Akai is in the middle, Dr. Avcı is at the left-hand side, Dr. Shea is on the right-hand side. Let me stop here, answer any questions you may have. Thank you so much for your patience.
Thank you for your presentation. That was very fruitful. And here, actually, we have 2 questions. And due to limited time, we can only have 2 questions. And first comes from İdil Polat. She asked that at the beginning of your presentation, you said that you support women in these areas and that you care about the issue, which is really pleasing. I would like to ask, are there any companies, clubs, and foundations that you think will develop and support us, that a molecular biology and genetics student should communicate with in this context?
Of course, what we have is the industrial, the leadership, the summit. We are going to have another one. I highly encourage you to attend those. For the IEEE as a society, we are going to have the celebrate 70th years, and we are going to have Women in Engineering conferences, and we will also invite the CEOs from companies to interact with our students. And most of the companies in the United States, they are required, they have also the diversity and inclusion to do. So if she contacts and if she wants to get involved, and I will provide the names for her to get involved. So it's a major activity for our celebration this year, Women in Engineering Conference, and also they bring not only the professors, but also industry leaders to attend these activities, and they provide platform for open and interactive. And the molecular genetics are extremely important, and I mean, I'm sure that is many companies could be very interested, but always good to add, as I mentioned, something on top of, like the computation side, modeling side, or the building micro nanotechnologies for molecular genetics really puts ahead of the game. Thank you.
Yes, I think your answer was very, actually, how can I say, conspiring for women in this area that want to actually show themselves and have the support in like those areas, I guess.
I think the student can contact me. She can, or he can send email, and we will communicate. Okay. Second question.
Şeyda Ayacı,
Okay, Şeyda, go ahead.
What's your biggest motivation for chairing MBS, a community that connects students with science and organizes global events?
I think it's this is a team effort. Of course, I envisioned all these activities I put on the slides. And for this year's, we are going to have the first ever to have the biomanufacturing public forum. This will be led by the Professor Shankar Subramanian at the UC San Diego. The second event we are going to have, the first time for our society, we will have the engineering innovation in the practice. So we will have medical doctors are going to talk about the healthcare innovation needs and also the engineering medicine. I'm not talking about engineering in medicine. I'm talking about engineering medicine. So in the United States right now, 2 or 3 medical schools are looking for engineers to go to the medical school because you get engineering degree. These are medical schools are innovative based medical school. One of them is the Texas A&M, is in the Houston, and other one is also Urbana-Champaign Medical School, and also University of Houston College of Medicine. We are very healthcare innovation oriented levels. So the medical education is changing because they need more quote data science, they need more healthcare innovation. Okay.
Thank you for your answer,
Thank you so
Professor.
much. I think is the you should be very happy. My talk is over, and you will listen the best and brightest mind and the beautiful part of our family, Professor Yasemin Akay. Thank you so much for thank you.
So due to the overtime, we will be back in a minute. And for our viewers at YouTube, please stand by. We will be just back in a minute.
Thank you so much. Wonderful to see you all. Okay. Keep in touch.
And again, over and again, thank you very much for making room for us at this very limited calendar.
Thank you. It's my pleasure. Thank you. Bye.
ilaçlar bu beyin tümörlerinde. Bu çünkü bizim özellikle kendisi de bahsetmiştir. Özellikle üzerinde çalıştığımız bir konu. Bu yüzden ben de bunun başka bir tarafını, o teknolojisini anlattı. Ben de bu yöntemleri anlatmak istiyorum size. Evet, şimdi dur bakalım. Siz mi yaptınız, ben mi yaptım? Size next slide mı demeliyim, ne demeliyim? Siz açtınız.
Derseniz direkt geçerim.
Siz açarsanız. Peki. Şimdi bu brain tümörün bir tanımlanması tabii. Her zamanki gibi tümör, kanser, böyle korkulan bir hastalık ama özellikle beyinde olduğu zaman biraz daha korkulan galiba, Allah korusun. Yani bir anormal sel toplanması ve bu toplanma geometrik bir şekilde oluyor. Ve genelde bizim şeylerimizde neoplazm ya da lezyon deniyor bunlara. Yani tıp otoritelerinde. Sonraki lütfen. Şimdi ben bu Amerika'daki şeyini biliyorum tabii bu beyin tümörlerinin. 79.000'den fazla primer beyin tümörleri. Bu nasıl? Sadece beyinde oluyor. Yani bir yerde oluşup da sonra metastaz olmuş değil. Beyinde oluşan tümör. Teşhis edilecek bu sene. Ondan sonra bunun 4.800 tanesi çocuklarda ve 0'dan 19 yaşa kadar. Aslında 18 olması lazım. Tabii buradan 18 yaşındakiler bile pediatriste gidiyor. Yani 18 yaşına kadar çocuksunuz burada. Onun için çocuk diyor yani. Böyle üzerine kimse alınmasın. Yani bir de brain ve merkezi sinir sistemi tümörleri. Sadece beyin değil. Çok yaygın. 0 ile 14 arasında. Bunu büyük bir ihtimalle çevre kirliliğine bağlayanlar da var ki çok da haklılar. Çünkü bazen ne oluyor biliyor musunuz? Bir atıklar var. O atıkların üstü kapatılıyor. Endüstri atıkları, fabrika atıkları, özellikle çok endüstri olmuş yerlerde, New Jersey gibi öyle ve onların üstüne çocuk bahçesi yapılıyor. Onların üstüne çocuk bakım merkezi yapılıyor. Ne oluyor? Bütün o sanki atıklar hiç sızmıyor mu? Yağmur yağmıyor mu? Hiçbir yer o sulardan buharlaşıp o toprak yumuşadıkça çamurdan şundan bundan onlar bir şekilde dışarı çıkıyor. Yani eminim yani bu yüzden bu çevre kirlenmesine büyük ölçüde buna şey olduğuna eminim. Tabii bazı genetik faktörler de var. 700.000 kişi tümörle yaşıyor. Nasıl yaşıyor? Teşhis konduktan sonra bir tedavi edilme şeyi var. Tedavi süreci var. Tabii bu arada hayatınızı devam ettiriyorsunuz bir şekilde. Yani o tümörün izin verdiği bir şekilde hayatınızı devam ettirebiliyorsunuz. Bu arada tabii hemen ameliyat olursa o beyin tümörü, beyin düşünün yani her yeri fonksiyonel olan bir organ. Yani onun ne tarafını çıkarırsanız çıkarın etkilenmeyen bir fonksiyonunuzu düşünemezsiniz. O yüzden yani olabildiğince hayatını gene devam ettiriyor. 700.000 kişi şu anda Amerika'da. Ama şu an 17.000 kişi de bu sene bekleniyor hayatını kaybetmesi. Bazı tümörler çok daha ölümcül oluyor maalesef ve çok kötü huylu oluyor. Bunlar 100'den fazla tipi var beyin tümörlerinin. Sonraki lütfen. Bunlar nasıl tabii multiple insults dediği bu tümör oluşumu için birçok kötü etki. Çevreden olur, beslenme bozukluğundan olur, herhangi bir şeyden olur. Yani birçok kötü etki bu beyinde bu tümörlerin oluşmasına sebep olabiliyor. Ya da one big hit. Bu nasıl oluyor? Mesela bir radyasyon hücumu gibi diyeyim artık. Bir şey vardı. Sizin için çok yaşlı ama şey var mı? John Wayne'i hatırlar mı? Anneleriniz, babalarınız hatırlar. Cowboy filmlerinin vazgeçilmez artist şeyidir. Yani onlar bir zaman bir Amerika'da bir çölde şey yapıyorlar. Film çeviriyorlarmış. Orada da galiba bir nükleer denemeler yapılıyormuş tam orada. Tam ona çok yakın bile değil. Öyle biraz uzakta bir yerde ama o gün orada sette düşün John Wayne'le beraber ve ondan sonra o bütün çalışanlar edenler hepsi hayatlarının bir döneminde bir şekilde değişik bir kanserden ölüyor. Yani one big hit dedikleri bu şekilde bir şey olsa gerek. Öyle. Ya da böyle immün sistemi çok zayıf dediğiniz gibi o çevredeki insanlardan, o kötü etkilerden çok daha çabuk ve kolay etkilenebiliyor insanlar. Bir de bu cell to cell communications yani bir enflamasyonu bir haber veren, sinyal eden bir sistemler var, bir seller var. Ve bunlar bir şekilde bunlarda bir bozukluk olduğu zaman ve belki bu enflamasyonu tam olarak haber veremiyor. Ve bu da bir takım kötü etkilerin, bir takım kötü formasyonların daha pek fazla farkına varılmadan bir süre yaşamasını ve büyümesini sağlıyor. Böyle oluyor. Lütfen sonraki. Bunlar işte common sign'lar. Yani beyinde tümör olabileceğini, iyi huylu olabilir, kötü huylu olabilir ve hiç beklenmedik bir şekilde baş ağrıları, epileptik nöbetler gibi. Yani bir bellekte bir değişme, kişilikte bir davranışlarda bir değişme olabiliyor. Ama size söyleyeyim yani bunlar aslında bir stroke'un da habercisi. Onun için bir insanın bunu şey yaptığı an yani experience ettiği an bunu hemen doktora gitmesi çok daha iyi. Ya stroke da olabilir, çok daha ciddi bir şey de olabilir. Aynı şekilde bulanık görme, çift görme, bu da o yaşlandık, bu da yaşlılıktan bizim insanlarımızın genelde insanların bir şeyidir bu yani. Ve motor kontrol, hareketlerde bir bozukluk dediğim gibi bunlar yani tümörün yanında başka bozuklukların da olabileceğini beyinde ya da omurilikte gösterebilen işaretler. Tanıtın lütfen. Bu şimdi manyetik rezonans görüntüleme denilen bir alet çok sıklıkla kullanıyor. Öncelikle bu gold standart. Nasıl COVID-19'de teşhis için PCR, qPCR gold standart. Bu da manyetik rezonans görüntüleme de gold standart bunda. Ama ondan önce acil olarak sizi doktorun göndereceği şey kompüterize tomografi. Yani bu bir şeyi teşhis etmede gösteriyor. Yani teşhis etmede çok önemli bir olay, çok iyi bir yöntem. Bu eğer daha ciddi bir şeye işaret ediyorsa o zaman sizin MRI'ye gitmeniz gerekiyor. Yani Allah korusun yani hastanın MRI'ye gitmesi gerekiyor. Çevirin lütfen sonrası. Bir defa ya beyinde bir şey olduğunu fark ederlerse ya önce bir biyopsi almaya gidiyorlar beyinden, onu patolojiye gönderiyor, bir şey var mı yok mu beyinde diye ya da hemen surgery'e gidiyor. Hemen surgery'e gidiyor. Bu şekilde de olmayabilir. Hemen cerrahi gitmeyebilir. Biomarkerlar. Dokudaki biomarkerlar ve ayrıca body şeydeki mesela kandaki biomarkerlar bunu da bu böyle bir tümörün varlığına işaret edebiliyor. Bunlar bütün teşhis yöntemleri. Tabii surgery'ye hemen gitmeden bunu da düşünme şeyleri olabiliyor. Optionları olabiliyor. Tamam. Buna işte iki kategoride bir primer brain tümörleri ve beyinde oluşan bir de metastatik yani yayılan brain tümörleri var. Bu çok şey. Bunlar primerler iyi huylu veya kötü huylu olabiliyor. Ama metastatik her zaman kötü huylu olabiliyor brain tümörleri. Sonraki lütfen. Bunlar şimdi beyinde oluşan ve yayılan beyin tümörlerine bir şey. Bu beyinde oluşan bir tümör ve tabii şu ön tarafı, hasta bu tarafa bakıyor, bizden tarafa bakıyor ve onun için burada sağda değil aslında bu tümör solda. Bu da yayılan beyin tümörü, yayılan cinsinden beyin tümörüne bir ölçü. Sonraki lütfen. Bu yayılan beyin tümörleri, şimdi bir bakmak lazım. Bakın mesela ovarian cancer nereye geliyor? Karaciğere yayılıyor. Akciğer zarına yayılıyor. Kolorektal cancer kolonda oluşuyor, karaciğere yayılıyor. Sonra kemik, lung cancer kemiğe yayılıyor, beyinde metastaz yapıyor. Breast cancer ne yapıyor? Karaciğerde, akciğerde ve beyine ve kemiğe metastaz yapıyor. Yani dediğiniz gibi bu bence tartışılması gereken bir konu. Çünkü böyle bir şey olduğu zaman, yani beyinde böyle bir olay olduğu zaman bunlar kesinlikle kötü huylu da, yani acaba bedenin başka bir yerinden mi metastaz yaptı, yoksa sadece beyin mi? Öncelikle bunun kesinlikle tayin edilmesi lazım. Çünkü ona göre ya bir brain onkolojisti tedavisine gidecek, ya da bence bu esas sebebini, esas kökünü bulup önce onu tedavi etmek. Sonra o beyindeki metastazın şeyine gitmek, ya da ikisini birden. Bunlar çok çok critical konular. Tamam. Sonraki lütfen. Primerler işte, yani dediğimiz gibi ya oluyor, iyi huylu veya kötü huylu olabiliyor. Yani ve beynin her tarafında olabiliyor. Burada gördüğünüz gibi. Sonraki lütfen. Bunlar da tümörlerin dereceleri tabii. Bu International Grading World Health Organization tarafından. İşte birincisi en az malignant olanı, en yavaş büyüyeni. İkincisi yavaş büyüyen fakat yayılabilen ve tedaviden sonra biz tekrarlayabilen. Bu ise daha fazla. Üçüncüsü ve çok daha hızlı büyüyor. Kötü huylu ve genellikle tekrarlıyor. Ve maalesef dördüncüsü de en hızlısı ve en agresif olanı. Ve bir de işin enteresan tarafı, bu tümörün tek bir yapısında heterojenik bir yapı olabiliyor bu tümörlerde. Bunların her bir şeyinde değişik derecelerde hücreler olabiliyor. İşte bu beyin tümörlerinin zorluğu burada. Özellikle glioma ve glioblastoma dediğimiz brain hücresinin en zor, en kötü tarafı bu tedaviye geldiği zaman. Sonrasına bakalım. Evet, bunlar şimdi en görülen beyin tümörleri. Meningioma, bu meningeler biliyorsunuz beyin zarları. Onun beyin zarlarında görülen. Glioma, şu beyin zarının kapladığı, onun altındaki böyle daha bir yumuşak bir doku vardır beynin. Orada görülen tümörler. Ve bu pilositik astrocytoma en common bir şekilde çocuklarda. Bu brain tümörleri do not discriminate derken herkese gelir. Ne zengin, ne fakir, ne kadın, ne erkek, ne bir şey. Yani geldiği zaman hiçbir şekilde ayrımcılık yapmıyor. Bu çok kötü bir şey. Ama dediğim gibi insanların belki beslenme şeyleri, insanların daha az kirlenmiş bir çevrede yaşamaları, daha iyi bir bakım altında olmaları onların hayatlarını uzatabiliyor eğer tümör kötü huyluysa. Bu işte beyin zarının tümörü. Şimdi bu hasta şimdi bize yüzüyle bakıyor. Yani bu meningioma bu beyin zarında aslında sol tarafta olan bir şey ve gördüğünüz gibi burnu burada, gözleri burada. Bu da sol tarafta. Bu da şeyden görünüşü. Böyle tepeden görünüşü. Aynı hastalığın. Sonrası lütfen. Bu kötü huylu glioma, yani beyin zarının altındaki yumuşak dokuda oluşan beyin tümörü. Bunlar bazen oluyor, değişik tiplerini burada görüyorsunuz ama bunların hepsi birden karışmış bir şekilde de olabiliyor. Yani. Bu da öyle. Bu high grade, bu ya 3 derecesi ya 3 ya 4 olan bir şey. Genellikle dediğimiz gibi kötü huylu ve çabuk yayılabiliyor, çok agresif olabiliyor. Sonraki lütfen. Bu glioblastoma. Bizim üzerinde çalıştığımız bu. Glioblastoma multiform üstelik de. Nasıl multiform? Bu bir defa dediğiniz gibi en agresif ve en öldürücü olanı. Yani Allah korusun bu götüren bir şey. Yani ötekilerde belki biraz daha şansları var insanların. Bu çok zor. Yani bir senenin, 2 senenin üstüne çıkan oldukça az. Bu tedaviyle, şununla bununla. Olmuyor. Ve bu GBM dediğimiz glioblastoma multiform etrafında değişik grade'lerde seller olabiliyor. Bu aynı tümörün içinde hem böyle agresivitesi hem yaşam şeyi değişik olan hücreler oluyor. Bir de bunlar hep yuvarlak olarak diye düşünülür değil mi? Brain tümörleri. Bu böyle değil. Bazen böyle bir parmaklar falan çıkarıyor. Şeylerde görüyoruz. Ondan sonra o zaman işte bu cerrahları biraz şaşırtıyor diyeyim. Çünkü nasıl kesecek tümörü? Çünkü bunlar tümörü keserken bir de aynı etrafından biraz daha böyle dokuları almak zorundalar. Sağlıklı dokuları almak zorundalar ki yani o biraz bir şey etrafa kaçarsa onu da yakalasın diye. Ama bunda o bütün her tarafa böyle 5 parmak gibi yayıldıysa bunu nasıl yapacak? Buna çok daha önemli, sonra size göstereceğim bir navigasyon sistemi lazım cerrahlara. Bunları fark edebilmeleri için, bunları operasyonlarda kullanabilmeleri için. Sonraki lütfen. Senatör Ted Kennedy. Bu gene bayağı size göre yaşlı bir insan ama yani bu glioblastomaya teşhisi konuyor 2008'de ve epileptik seizure şeklinde. Bu önce epilepsi krizi diye gönderiliyor Cape Cod Hospital'da. Ama bu tabii hemen fark ediliyor. Mass General Hospital Boston'a gönderiliyor ve glioblastoma multiform. 15 ay. Yani eminim bir Ted Kennedy'nin düşünebiliyorsunuz. Yani aile yapısı, beslenmesi, yaşadığı çevre ve gördüğü bakım çok insandan çok daha iyi Amerika'da. Ama 15 ay. Sonraki lütfen. Bunlar şimdi yaşlara göre tümörler nasıl oluyor diye. Bir bakarsanız bunların hepsi bir küçük yaşlarda oluşabiliyor. 0-4 yaşlarında ama sonra bir azalma gösteriyor. Çok az veya çok bir azalma düşme gösteriyor 15-19 yaşlara göre bir şekilde. Belki immün sistemi daha bir güçleniyor insanların. Bunun bir nedeni var tabii. Bu en çok pituitary. Pituitary bir salgı bezidir biliyorsunuz. Tiroide çok yakın bir yerdedir ve bu salgı bezlerinde aslında oluşan şeyler çok kritiktir. Hemen müdahale edilmesi gerekir. Bakın 0-4 yaşta başlıyor ve artan sadece bu yaşla. 15-19 yaşlarında en şeye çıkıyor. Bu görünmesi 100.000 insanda görünme oranı 1,5 gibi bayağı yüksek bir oran yani. Sonraki lütfen. Şimdi bu da 19 yaştan sonrasını almış. Artık yetişkin. İnsanlar artık yetişkin olduğu 20-44 arası ile 75 yaş ve üstü diyor. Burada hemen hemen bakın normal bir seyir takip ediyor. Öyle çok fazla artmıyor değil mi? Bir kısmı. Yani burada gösteren fazla ne var? Glioblastoma biraz artıyor. Tabii 75 ve üstünde en yüksek yani orada maalesef insanlar tabii yaşlandıkça o immün sistem biraz etkileniyor herhalde onlar da daha bir artıyor. Sonra bu ötekilerde çok fazla artış yok yaşla. Çok şey yapan meningioma. Brain zarı tümörü. Bu da kötü huylu değil. Yani bu insanlar bu kötü huylu olmayan şeyle yaşayabiliyor. İşte ilacını alıyor, tekrarlarsa ne bileyim bir kötü olan şey, eğer ameliyat yaparlarsa beyinde hangi fonksiyonları etkiliyorsa yaşam kalitesi ona göre oluyor. En zor olan tarafı ama yaşıyorlar bir şekilde yani. Sonraki lütfen. Tamam. Brain tümörleri nasıl treat edilir? Bir surgery to remove tumor bulk, ama bunun da dediğim gibi çok şeyleri var, yani zorlukları var. Şimdi öncelikle tabii ameliyata gidiliyor. Öncelikle ameliyata gidiyor. Bu aslında çok doğru bir şey değil. Bence öncelikle kemoterapi ve şeyi küçültmek. Tümörü mümkün olduğu kadar küçültmek. Böyle olduğu zaman ameliyat yaptıkları zaman alacakları yer de daha küçük oluyor. Ve beyindeki fonksiyonel bölgelere de yapacakları zararları daha az oluyor bu ameliyatın. Ondan sonra da radyasyon tedavisi. Bence bu çok daha çünkü o da geri kalanını temizlemesi açısından bu klasik tedavi yöntemi. Sonraki lütfen. Benim bu size söylediğim bu işte. Nöronavigasyon dediğimiz yöntem. Şimdi nasıl, bakın hastanın hasta tabii full anesthetized bütün sistemlerle ve burada ya surgery yaparken ya da ne bileyim ben bir biyopsi alınırken bu sisteme ihtiyaç var. Bir taraftan da kamerada görüyorsunuz çok problar var. Bu görebiliyor musunuz şunları? O problar hani beynin neresine müdahale edildiğini gösteriyor. Laser pointer, harika. Ben onu kullanabiliyor muyum? Yok. O hep sizin tarafınızdan. Tamam, olsun. Teşekkürler. Tamam. Ne diyecektim? Bakın beynin tümörün dış sınırlarını yapmak açısından çok daha kolay. Bu nöronavigasyonla ameliyat yapmak ki, ama bu çok yeni bir teknoloji. Nerelerde, kim buna ulaşabiliyor? Hangi hastanelerde bu yapılabiliyor? Yani inşallah her tarafta ulaşabilir de, yani bu çok zor. Çünkü bir de böyle hem o ameliyat hem de böyle parmaklar şeklinde yayılmış tarafları varsa onu daha iyi görmek mümkün. Şeyin sınırlarını, etrafta kesilecek normal hücrelerin sınırlarını görebilmek böyle çok daha iyi. Bir de bu yaparken, radyasyon tedavisi yaparken böyle daha bir konsantre olabilmek şeyini veriyor. Onun tam yerini, tam koordinatlarını verdiği için daha bir konsantre radyasyonu daha yüksek miktarda buna verme olanağı tanıyor. Sonraki lütfen. Tamam, bunları gördüğünüz gibi. Bir de targeted, bu ikisini konuştuk, onun için yapmıyorum. Bu targeted ilaç tedavileri ne yapıyor? Sadece o tümörü hedef alan ilaçlar. Yani etraftaki dokuya, etraftaki sağlıklı dokuya zarar vermeyecek ilaçlar. Çünkü kemoterapinin çok kötü yan etkileri var. Çok kötü. Bazı veriyorlar şimdi. Şu ilaçları alın. Mide bulantısı olabilir, şöyle olur, böyle olur, başka yan etkileri olur. Onun için şu ilaçları alın diyor. Peki akıllı, eğitimli bir insan bunu alıyor. Peki cahil insan ne yapıyor? Bir defa bir cahil insan ilacı sevmez. Yabancı bir şeyleri sevmez. Yeni şeyleri sevmez, kabullenemez. Ne yapıyor? Almıyorum diyor. Ama sonra yan etkileri çok kötü oluyor. Yani bunu gördükleri için yapmaya çalışıyorlar sürekli. Repurpose drug. Siz bunu COVID-19'de gördünüz değil mi? Bazı doktorlar artık böyle, şimdi artık çok antiviral şeyler var. İlaçlar var mesela COVID'e değil mi? Ama daha önceden yoktu. Doktorlar ne yapıyor? Mesela diyor ya diyor şu ilacı biz kullanabilir miyiz acaba? Diye. Çünkü bazı hastalarda COVID için verilen ilaçlar çok kötü etki yapıyor. Çünkü o hastalar mesela onların komorbidite dediğimiz başka yanında da bir takım rahatsızlıkları var. O COVID için verdiği ilaçlar onlara çok zarar veriyor. O ilaçların etki göstermesini engelliyor öteki hastalıklarını. O yüzden doktorlar artık vazgeçmiş, repurpose demek yani başka şeylerde kullanılanları bunlara veriyorlar. İşte baş ağrısı şunu al, işte miden bulanırsa şunu al, işte bir de bağırsak sisteminde bir rahatsızlık olur, şunlar. Yani bu şekilde bir şey yapmaya çalışıyorlar. Yani sonra bir de yok öncekine gelin. Bir de düşük yoğunlukta alternatif elektrik alanı uygulanıyor. Bunu ne yapıyoruz? Hücre büyümesini engelliyor. Bir immün sistemi arttıran, kuvvetlendiren ilaçlar. Bir de hem diagnostik hem terapötik şekilde, kombine şekilde verilen ilaçlar. Bir de biomarkerlar. Bu konuda en çok denenen ilaçlar diyelim. Sonrası lütfen. Yani işte dediğimiz gibi iyi huylu tümör olduğu zaman hastalar yaşıyor. Bir de bunu predict etmek, önceden söyleyebilmek ki bunu hastayla olmasa bile her zaman hasta yakınlarıyla tartışabilmek. Yani tümör iyi huylu mu, kötü huylu mu, ne şekilde, ne kadar ömrü kaldı gibi bir şey olur her zaman, soru olur. Bu iyi huylu tümörlerde yani bu tümör çıkarılır, sonra gene olabilir ama bu tümörle yaşar bu insan. Ama tümörün getirdiği bir takım aksaklıklarla da maalesef eğer tümör önceden ameliyat edilirse kötü huylularda yani en büyük önceden belirleyici yaş 45 yaşın küçüğü genç olduğu zaman daha fazla şansı var. Bir de ne kadar tümör çıkarıldı, tümörün tipi ne, şey mi yani metastatik mi, sadece primer mi, biyolojik aktivitesi hastanın ne durumda? Bazı insanlar daha güçlü oluyor, bünyesi daha güçlü olduğu zaman, belki de bağışıklık sistemi daha güçlü olduğu zaman hastalıkla da daha güzel savaşabiliyor. Böyle işte. O yüzden glioblastomada bile, bu GBM'de bile hastalıkların verdikleri cevaplar çok değişik oluyor. Eğer hasta genelde çok sağlıklıysa, yani buna verdiği tepki ayrı, GBM'deki yaşama süreci ayrı. Bir de comorbidities dediğimiz başka hastalıkları varsa, bu GBM de yanına geldiği zaman çok daha bünye zayıf tabii. O zaman insanların yaşama şeyini etkileyebiliyor. Tanrısı lütfen. Yani şimdi bu gene yani aynı şeyler ama dediğim gibi yani bunlar şeyde de olabiliyor. Stroke gibi şeyler beyin rahatsızlıklarında da olabiliyor. Bu yüzden de bunların en ufak bu türlü hani bir normalden sapmış bir fonksiyonlarınızda gördüğünüz zaman Allah korusun yani hemen doktora gitmek, CT ondan sonra MRI çektirmek en iyisi. Sonrası lütfen. Bu iyi huylu tümörler, bunlarda bir şey, math reading challenges, bunlar önemli. Hani bu yorgunlukla baş ederseniz, short term memory, işte insan biraz yaşlıysa ayda sinir momentler çıkar. Ama yani şey yapmaz, bu şekilde etkiliyor. Eğer math reading günlük hayatımızda çok kullandığımız şeyler, en temel olanları bile, yani onda bir challenge olmaya başlarsa günlük hayatımızı etkiler. Bir de her gün normalde yaptığınız işinizi artık yapamıyor hale geliyor insanlar. Bir de yürürken dengesi, bu motor sistemindeki etkileşimleri yüzünden bir de insanların huyları değişiyor. Huyları değişiyor. Yani benign iyi huylu tümörlerde bile bunlar gerçekten bir şey zorluk yaratabiliyor. Sonraki lütfen. Şimdi bütün bu non-malignant iyi huylu olanlar bir daha büyük bir yüzdesini oluşturuyor bu şeyde. Öteki kötü huylular demiş ama gliomalar, bu özellikle kötü huylu olanlar çok bayağı büyük bir yüzdesini oluşturuyor kötü huylular arasında ve gliomalar arasında da en yaygın olanı maalesef glioblastoma. En agresif, en çabuk yayılan, yani Allah korusun en çabuk götüren diyeyim. Yani bu glioblastoma oluyor. Sonraki lütfen. Bir defa bu tedavide glioblastomaların tamamen ilerlemesi inanılmaz derecede hızlı. Görüyorsunuz değil mi? Yaşama şansı en başında %100 iken kaça iniyor? Ki yani 3 aydan sonra zaten hastada kesinlikle kendisini göstermek istiyor ve gösterme durumuna geliyor ve yani görüyorsunuz, yani buraya kadar da beklemiyor aslında. Evet. Sonraki lütfen. Şimdi hayatta kalma şeyi. Sadece yaparsanız ki bu çok daha düşük bir hayatta kalma şeyi ay olarak vermiş. Çünkü dediğim gibi yaptığınız zaman, ameliyat olduğu zaman bu etraftaki tümörleri etrafa kaçan tümörleri alamıyor. Çünkü hepsinin de böyle o hani state of the art nöronavigasyon şeyi yok. Radyoterapi evet bir şeye doğru bir şekilde hücreleri öldürdüğü için o da biraz daha fazla şansı var hastaların. Karmustin, radyoterapi ve karmustin. Karmustin ve lomustin, nitrozoürea türevi ilaçlar. Ve bunlar ya radyoterapiyle beraber veriliyor normal sistemik olarak ya da implant, yani böyle kontrollü salınım şeklinde bir yere bir implant konuluyor, kontrollü salınım şeklinde veriliyor ve gördüğünüz gibi arttırıyor. Temozolomide. Bu bayağı böyle golden standard bir şey. Hem radyoterapi hem temozolomide. Bunlar bir de temozolomidin yanında burada denedikleri statin türevi antikolesterolемik ilaçlar. Normalde insanlara kolesterolünü tedavi etmek için verdiği ilaçlar, bunlar bu kemoterapötik ilaçların şeyini arttırıyor. Yani etkisini arttırıyor. Onun için bunlar da bir arada veriliyor. Buradaki hastanelerde. Bu radyoterapi, temozolomide ve bevacizumab. Bu da ayrı bir şey ve bu da gördüğünüz gibi en yüksek hayatta kalma süresini veren bu türlü bir kombinasyon. Sonraki lütfen. Bu temozolomide şey gibi, golden standard, MRI gibi, qPCR gibi, golden standard. Şimdi radiation'la. Ne oluyor? Şimdi en başta yapmış. MRI ya da computed tomography. Böyle bir yaptığı zaman. Ondan sonra ya biyopsiye gidiliyor ya da hemen surgery ile, bu ameliyatla bu şey alınıyor. Ve ne oluyor bu parçalar sonra? Sonra patolojiye gidiyor. Patoloji bunu diyor. Evet, bu glioblastoma. Sonra ne oluyor? Tedaviye başlıyor. Bu radyoterapi ilk yapan first line terapi diye. Önce radyoterapi, sonra temozolomide ilaçları veriliyor. Ya da second line terapi, bu Avastin dedikleri bevacizumab. Ve nitrosoureas. Neydi bunlar? Carnostin ve lomustin. Bu ikisi veriliyor. Bu da ikincil bir terapi oluyor. Sonraki lütfen. Şimdi gördüğünüz gibi burada öncelikle bir basic research'ten başlıyor. Sonra bu pathway'ler ne kansere yol açıyor? Buna neyi target ederek, hangi bölgeyi, hangi molekülü target ederek, hangisini kullanarak, hangi yolla vererek bunları tedavi edebiliriz? Sonra ne oluyor? Bütün basic research'den sonra klinik çalışmalar başlıyor. Nerede kullanabiliriz? Hastaların şeyleri çok değişik. Onlara göre bir gruplandırıyor. Ne oluyor? Beyinlerinde tümör var. Beyinlerindeki tümörlerin hücre derecelerine göre hastalar gruplandırıyor. Mesela bir beyin tümörde bir grup hastanın 1, 2, 3 var mesela. Ötekinde 4 var, 2 var. Bunlar hep grupları ve buna göre nasıl bir tedavi uygulanacağı şeklinde. Hangi ilaçların buna daha etkili olacağı şeklinde bir gruplandırma oluyor. Label claims. Yani bu çok önemli bir olay. Şimdi bu kutular, reçeteler, içine prospektüsler çıkıyor değil mi? Yani bunların üzerinde hangi dozda alınacağı, işte son kullanma tarihi ve işte hamile olanların alınmaması gerekir. İşte 12 yaştan sonra daha büyüklere bu ilacı almaları gerekir. Yan etkileri şunlardır. İşte şu dozun üstüne çıkarsanız zehirlenme oluşur. Hemen arkasından zehirlenme kontrol merkezinin telefon numaraları falan. İşte bütün bunlar şeylerin kutularında, etiketlerinde olması gereken şeyler. Revenue potential. Bir de hani Big Pharma dedikleri ilaç firmalarının biz bundan ne kadar para kazanırız? Yani bir defa bu ilaçlar nezle ilacı gibi, grip ilacı gibi hani şey satmıyor. Bunlar çok daha düşük bir topluma hitap ediyor. Öyle olunca diyor ki ben bu ilaçla niye uğraşayım diyor. Yani bana ne kadar gelir getirir ona bakıyor bu insanlar. Sonraki lütfen. Şimdi hem challenge'lar var. Beni yeni terapide nasıl oluyor? Burada nasıl bir yeni bir şeyle, yeni bir terapi yapmaya değer mi? Bunda ne kadar biz bir iyileşme ya da ilerleme sağlarız? Ya da small population, size söylediğimiz gibi orphan drug potential. Yani bu ilaçlar satmazsa ne olur kendilerine göre? Bir de bu özellikle iyi huylu tümörlerde hani hastayı artırıyor. Bu tümör hastasının nüfusunu arttırıyor. Bunlar yaşıyor ya, tümörle beraber yaşıyor. Ameliyat olduğu zaman bile yaşıyor, arttırıyor. Bir de bunun şeyini nasıl? Genelde jenerik ilaçların fiyatları çok daha düşük. Ama daha önce bu ilacın patentini alan firmalar bunu engelleyebiliyor. Ki onların fiyatları çok daha pahalı çünkü. Ama jenerik ilaçlar satılmaya başlarsa bunlar kendi ilaçlarını satamaz. Sonra bir de kombinasyon terapi. Nasıl bir mekanizmalarla bir kombinasyon terapi uygulayabiliriz? Bunlar nasıl yan etkileri olur? Bunları nasıl etkileyebiliriz? Bu yan etkileri önlemek için de başka ilaçlar vermek zorunda kalır mıyız? Şey yapmıyorum. En büyük şeyler de blood-brain barrier. Bakın küçük moleküller çok daha favored bu tedavide. Bunlar ne yapıyor? Çünkü küçük moleküller blood-brain barrier'ı daha çok şey yapıyor. Daha çabuk geçiyor. Bu yüzden bunlar çok daha bir de tümörün heterojen olması. Glioblastoma multiform'de olduğu gibi. Bu en büyük challenge'lar bunlar. Evet, sonraki lütfen. Tamam. Şimdi bunlar hastada belli şeylerin, belli genlerin ya da belli reseptörlerin olup olmaması, aktif olup olmaması. Şimdi bu epithelial büyüme faktörü, reseptörü üçüncü varyantı. Tamam. Eğer bu hasta bunda pozitifse, ki %25'inde hastaların bu var, şanslı bir grup diyeyim, 23 ay oluyor hayatta kalma süresi. Negatifse, ki hastaların %75'inde 15 ay. Bir de MGMT gen var. Bunun iki şeyi var. Metillenmiş ve metillenmemiş. Eğer metillenmişi %45'inde hastaların 22 ay oluyor. Metillenmemiş olanı %55'inde maalesef bu sadece 13 ay. Yaşa gelince tabii 65 senior'dır. Yani artık 65 yaşına geldiğiniz zaman bütün senior indirimlerden faydalanırsınız. Yani her yerde bir başka türlü muamele görürsünüz. Yani ama ne yazık ki bu hastalıkta sizi biraz zorluyor, Allah korusun. Altında %50'si 65'in altında 15 ay. Ama 65'in üstünde gene %50'si yaşamı 9 ay. Tamam. Sonra lütfen. Tamam. Bunlar da klinik öncesi çalışmalar. Bu moleküler, scientific çalışmalar. Bir görüyorsunuz bütün bu kullanılan biyolojik small molecule immünoterapi, adı terapiye kullanılıyor. Bir şeyde ama 3. faza geldiği zaman biyolojikler bir şekilde buradan elimine edilmiş. Bu demektir ki çok fazla faydası görmemiş. Yani hep bir küçük moleküllü ilaçlar sanki daha bir şeydi. Bir de immünoterapi, terapötik ilaçlar daha bir şey. Evet, biliyorum ben zamanımı çok aştım. Çok az bir şey kaldı. Az bir şey kaldı. Bir sonrakini görebilir miyim? Tamam. Şimdi bakıyorsunuz bunlara. Bunlar çok değişik yeni approach'lar. Gene terapi, lighting, virüsler, nanoparticle stem, immünoterapiler çok şey. Active immunization. Şimdi bakın siz şeyi biliyorsunuz değil mi? Uğur Şahin'le Meltem Türeci, BioNTech'in sahipleri. Şimdi Meltem Türeci artık bu Covid'le şey yapmaktan vazgeçti. Kampanyayı bıraktı. Yeni bir center'ın başına geçti. Orada zaten daha önce kanser aşısında çalışıyorlarmış Covid'den önce. Şimdi o kanser aşısını tamamen kendi üstüne aldı. Bu center'ın üzerine aldı. Ve kanser için aşı, bu active immunization, bu inanılmaz bir şey. Yani kanser için herkesi aşılamak, risk grubu olan herkesi aşılamak, bu harika bir olay. İnşallah her şeyi şey yaparlar. Bir de monoklonal antibodies, bu biliyorsunuz çok yaygın olarak kullanılıyor. Covid'de yaygın olarak kullanılıyor. Kanserde de kullanılması düşünülüyor. Bir de small molecule kemoterapi, dediğiniz gibi bu blood-brain barrier issue yüzünden, yani bu small moleküller çok çok önemli. Ve ayrıca ve daha da yeni new options for glioblastoma patients. Yani sonraki lütfen. Çok teşekkür ediyorum. Sabrınız için, ilginiz için çok çok çok teşekkür ediyorum size. Onu alayım.
Hocam son dediğinizi duyamadık.
Sorularınız varsa dedim alayım.
Evet. Sorumuz birikti aslında ama zamanımız kısıtlı olduğu için biz ilk sorumuzu soracağız. Sizin için uygun mu?
Gayet iyi, gayet iyi. Tabii uygun.
Evet, çok teşekkür ederiz hocam. Bu ilham verici sunumunuz için, bize ayırdığınız vakit için ve bütün hazırladığınız her şey için. Gerçekten dinlemek çok mutlu etti bizi. Söylediğiniz her cümlede, her slaytta daha da ilham alarak dinledik sizleri. O yüzden bizim için, bize kattığınız her şey için çok teşekkür ederiz. Öncelikle ben de Yasemin İlayda Albayrak ve arkadaşım Gizem burada size sorularımızı yönelteceğiz. İlk sorumuz, aslında metastatik beyin tümörlerini teşhis ederken oldukça ince davranılması gerektiğini ve esas olduğunu öğrendik sunumumuzun başlarında. Peki bu konuda yapılan hatalarda geri dönüş sağlanabilir mi ve bu nasıl sağlanabilir?
Şimdi yani size bir şey söyleyeyim mi? Tıbbi hatalar hastanelerde çok yaygın. Bu yüzden de ölen hastalar da çok şey. Yani bu ortaya çıkmış bir şey. Ya hastanelerde ya insanlar enfeksiyon nedeniyle ölüyor ya da bu ameliyatlarda daha çok yapılan hatalardan ölebiliyor. Yani bu gerçekten çok üzerinde araştırılması gereken bir konu. Yani hata, şimdi iki taraflı. Hata olursa hatayı yapan insanla doktor onu düzeltmesi için fırsat mı verirsiniz? Yoksa başka doktora mı gidersiniz? Nasıl şey yaparsınız? Yani bir de onun üzerinde düşünmek lazım. Yani bu tamamen tıptaki arkadaşları düşünmesi gereken bir konu üzerinde. Yani ben doktor değilim, beyin cerrahı değilim. Yani ama bence başka insanlar daha en başında zaten. En başında hani second opinion derler ya, bence third opinion'da olmalı o. Yani bakacaksınız, ayrıca kendiniz de okuyacaksınız. Eğer kendinizde bir şey varsa, yani doktorun bilmesini istediğiniz bir şey varsa onu kesinlikle doktorunuzla tartışacaksınız. Bende böyle bir şey var. Mesela size MRI verir, manyetik rezonans görüntüleme, ama bakın sizde ileri derecede klostrofobi varsa o MRI'ye giremezsiniz. O bir tüp içine böyle sokuyor. O zaman ne der? Yani işte bende böyle bir şey var. Daha en başında düşün. Bir şey var, ben buna giremem. Bunu ancak beni biraz uyutarak alabilir misiniz? Onu aynı gün söyleyemezsiniz. Çünkü doktorun bunu yazması, bu ilacın gelmesi, özel bir ilaç, onu gelmesi ve size uygulanması lazım orada. Öyle kafadan size orada yapamaz. Kendisi o zaman lisansını kaybeder. O yüzden dediğim gibi böyle ufak tefek şeyler, hiç bilemediğimiz, hiç daha önceden farkına varamadığımız bir takım şeyleri böyle hata, dediğim gibi sonra herhalde inşallah düzeltilebilir. Ya da hiç böyle bir hata olmaz diyeyim artık.
Evet, çok haklısınız hocam. Yani bu yapılabilecek, karşılaşılabilecek bütün olaylarda aslında nasıl ilerlenmesi gerektiğini merak ederek sormuştuk bu soruyu. Verdiğiniz cevap için çok teşekkür ederiz.
Bir şey değil.
Hocam ben de başka bir soru soracağım şu an. Birçok çeşit beyin tümörü inceledik, resimleri gördük, karışık olarak bulunabiliyor dediniz beyinde. Yapılacak tedavilerde bu karışıklığın tespiti ve tedavisi nasıl etkileniyor?
Şimdi şöyle, ona baktığı zaman mesela kompüterize tomografide, ondan sonra MRI'de baktığı zaman onu görüyor zaten. Ya da böyle biyopsi alıyor. Bir şekilde öyle heterojen bir yapı gördüğü zaman, ki göstermesi lazım, ya da o nöronavigasyon şeyinde robotunda gördüğü zaman değişik yerlerden biyopsi alması lazım. Eğer hemen tümörü çıkarmayacaksa, bu özellikle GBM'de de çok önemli. Çünkü glioblastoma multiform demiş. Yani onun değişik grade'lerde her tarafında ya da iki değişik kanser bir şekilde bitişmiş. Onları öyle anlayabilecek ancak patoloji. Parça alacak, yani o parçayı alacak, biyopsiyi yapacak insanın çok dikkatli ve çok iyi olması lazım. O değişik parçaları patolojiye gönderecek. Onlar da bakacak. Yani.
Teşekkürler. Evet, verdiğiniz cevap için çok teşekkür ederiz hocam. Aslında benim bir sonraki soracağım ve son soracak olduğum soruyla da birazcık alakalı. Biz daha öncesinde sizin yaptığınız son araştırmaları ve yazdığınız son makaleleri de okuduk aslında. 3D virtual modelleme yaptığınızda ve geleceğe dair çok fazla umut vaat ettiğinden bahsetmişsiniz. Klinik çalışmalarda göreceğiniz sonuçların çok umut verici olduğunu söylemişsiniz. Bunun hakkında size soru sormak istedik aslında. Bunun ne çeşit güzellikler ve ne çeşit farklılıklar yaratacağını düşünüyorsunuz aslında?
Vallahi şöyle, ben öncelikle zaten eminim teknolojiyle bunu da yeneceğiz. Hayır, dediğiniz gibi, yani bunu çok istiyoruz. Bunu çok istiyoruz. Ama dediğim gibi öncelikle bir teşhis edilmesi. Öncelikle insanların kendi kendini bilmesi ve kendi kendine, yani bir değişiklik olduğu zaman bunu hemen erken teşhis deriz ya. Hep böyle göğüs kanserinde erken teşhis, her türlü şeyde. Yani kendinizde bir değişiklik hissetmeye başladığınız zaman bunu hemen bir doktora gitmek. Ve dediğim gibi bu akıllı sistemlere, akıllı ne diyeyim şey yapmak, yani iyi bir doktora gitmek ya da o iyi doktor size anlatabilir her şeyi, ama dediğimiz gibi başka bir doktora da danışmak. Öncelikle bu. Ne var bende diye şey yapacaksınız. Ondan sonra tedavi, ondan sonra ve tedavi opsiyonlarını da düşünürken çok dikkat edeceksiniz. Yaşam tarzınız nasıl? O sizin yaşam tarzınızı nasıl etkileyecek? Yani bütün bunları çok etraflı bir şekilde konuşmanız lazım doktorla. O teknoloji harika olur, evet. Her şeyin başı teknoloji tabii. Sizi çok ilerilere götürebilir, ama dediğim gibi onun oluşması için, onun yerine oturması için çok zamanın geçmesi de gerekebilir. Size hemen faydası olamayabilir. Çünkü benim mesela bizim şeylerimize, konferanslarımıza çok doktorlar da katılıyor. Ama doktorların ilk kafasında sorduğu şey, yeni bir teknoloji, ben bunu hastama nasıl taşıyacağım? Ne zaman taşıyabileceğim? Pardon, ya da bu eğer evde uygulanan bir şeyse ya da alınması gereken bir aletse nasıl olacak? Fiyatı ne kadar olacak? Yani onlar bu konuda, bunda hemen cevap bulamıyorlar. O yüzden de biraz canları sıkılıyor tabii. Yani onları da anlamak lazım. Bunları hani bedside'a, hastanın bedside'ına bunu nasıl taşıyacağım gibi düşünceleri var. Onun için her yeni yöntem yani bir yerine oturması lazım. Bir ilaç alacak, şöyle olacak. Yani onu hızlandırmaya çalışıyorlar şu anda. FDA'den. Bir de tabii Türkiye'de Sağlık Bakanlığı'ndan nasıl şey oluyor bilmiyorum. Yani evet.
Hocam verdiğiniz bu açıklayıcı cevap için çok teşekkür ederiz. Aslında sunumumuzun başından beri de gerçekten bizi en çok kafamıza oturan kısım erken teşhis oldu. Yani kendimizi dinlememiz gerektiği, doktorlarımızı dinlememiz gerektiği ve tamamen hem doktorlarımızın hem de kendi insanlarımızın bu oluşabilecek tüm durumlar hakkında bilgilenmesi gerektiği ve kendilerini ona göre bunlara hazırlayarak ilerlemesi gerektiğini fark ettik ve umarım bu seanslarımızda ve bu sunumumuzla birlikte herkes çok daha bilinçlenir. Ve
o çok önemli. Evet. Evet.
Kesinlikle. Özellikle katılımcılarımızdan çok fazla sorulan bir sorumuz daha var. Kıramadık, onu da iletmek istiyoruz sizlere. Vaktiniz varsa lütfen.
Benim vaktim var. Lütfen ben
teşekkür ederiz.
aldım çok.
Çok güzel.
Siz de burada olmaktan çok
kesinlikle.
Başka ne? Bir şey mi soracaktınız başka?
Söylüyorum şu an. Çeşitli kemoterapik ilaç salınım yollarıyla tümör bölgesi dışındaki çevresine saçılmış onkogenik hücrelerin tümör çevresine çekilmesi ve cerrahi müdahalenin kolaylaştırabilmesi mümkün müdür?
Tümör çevresine çekilmesi, tümörlerin tekrar, yani tümörü o kaçan hücreleri, ben böyle bir şey duymadım. Eğer böyle bir araştırma varsa olabilir ama ben hiç, yani zaten bütün dertleri onları yakalamak, onu önlemek, o kaçmaması yani. O yüzden tümörün etrafına şöyle şey yapıyorlar, böyle biraz daha sağlıklı bir doku, bir şey gibi, hani şu kalelerin önüne bir hendek kazarlar ya etrafına. O genişletiyorlar ki o şeyi o zaman kaçmasın. Bütün tümörü yakalayabilsinler. Ondan sonra kaçanları yani toplayamaz belki de, şeyden sonra radyasyon tedavisi belki onları öldürür. Yani nasıl toplar? Surgery'de böyle bir yöntem var mı? Ben onu bilmiyorum. Özür dilerim. Yani radyasyonda belki onu öldürür. Başka ben bilmiyorum.
Hocam kemoterapik ilaçlarla peki bu mümkün olabilir mi acaba? Etrafa saçılmış hani hücreleri öldürmek için ya da toplamak için.
Belki Sonra belki olabilir yani benim bildiğim bu şekilde pek olamaz. Zaten ilaçların önce yani ameliyattan önce verilmesi daha doğru olur sanki. O tümörü bir küçültsün, büyümeyi düşürsün, o tümörü küçültsün. Ondan sonra ameliyat olacak, ondan sonra radyasyon yapacak. Yani başka tedavi yöntemleri var mıdır?
Bu ameliyattan önce tümör küçülsün diye verdikleri hani ilaçlar bunu aslında sağlıyor mu? Hani sonuçta tümörü bir miktar küçültüyor sonuçta yanlış anlamadıysam.
Vallahi o da hastasına göre değişiyor. Bazı hasta daha iyi cevap veriyor. Bazı hasta o ilaca cevap vermiyor, başka ilaca daha iyi cevap veriyor. Yani
Ve büyük ihtimalle bahsettiğiniz seviyesine göre de hem verdikleri ilaç değişeceği için de o verdikleri cevaplar da değişecek. Buna bağlı olarak da doktorun bunu inceleyerek ilerlemesi gerekecektir.
evet yani onun için çok iyi eminim bütün doktorlarımız çok iyi yetişmiştir ama yani gerçekten alanında çok iyi yetişen bir doktoru bulmak ve onunla bu bütün bunları konuşmak ayrıca bir insanın eğer yani okuma yazma bilecek yaştaysa öyle çok küçük çocuk değilse yani kendi hastalığı konusunda okuması sürekli ve doktoruna sürekli sorular yönlendirmesi. Sürekli ve ondan sonra verdiği ilaçlar konusunda kendisinin bilgi sahibi olması, onların üzerine hastaların kendi kendini eğitmesi bu konuda çok önemli. Her zaman çok önemli. Bu bilinçli hastalar yani doktorların da şey yaptığı bilinçli hastalarla çalışması çok daha kolay. Çünkü insanlar diyor ki dediğim gibi o klostrofobi örneğini verdiğim gibi yani benim böyle bir şeyim var ya da bu MR'ya giremezsem seçimimiz ne olur? Ya da ailede böyle bir şey var. Yani bu tamam bu insan yaşıyor. Şeyle iyi huylu bir tümörle yaşıyor ama ileride böyle bir hastalık var. Bana bu hastalık gelirse ailemde gelen bir hastalık bana komorbidity yaparsa nasıl yaparız? Nasıl tedaviyi yönlendiririz? Bu yani hastaların öncelikle çok bilinçli olması lazım ve çok eğitimli bu kendi hastalıkları konusunda. Çünkü doktor da yani orada bir sürü hastayla uğraşıyor. Yani tamam bütün yaptığı sizin randevuya gelmeden önce sizin şeyinize bir göz atmak oluyor. Ne hastalıkları var, nesi var, şikayetleri neler, şunlar bunlar. Yani onun için hatta gerekli makaleleri okuması lazım insanların. Eğer biraz böyle bilimsel bir düzeyde bir eğitimi varsa hastaların kesinlikle bunu yapması lazım.
Kesinlikle hocam. Bahsettiğiniz son konular da gerçekten çok etkileyici ve çok farklı bir bakış açısıydı. Artık en başından itibaren bahsettiğiniz, bizlere verdiğiniz tüm bilgiler için, tüm katılımlar için, tüm uğraşlarınız için çok teşekkür ederiz. Gerçekten sizi dinlemek çok büyük bir zevkti. Her söylediğiniz kelimede kesinlikle tekrardan hem ne kadar çok bilinçlenmemiz gerektiğini hem de sizlerden daha ne kadar çok şeyler öğrenebileceğimizi daha da çok fark ettik ve çok mutlu olduk kesinlikle.
Ay ben de çok mutlu oldum. Sizi böyle hem daha böyle bir şey olarak bilinçli olarak gördüğüm için daha böyle ilgili olarak gördüğüm için ve bir de yüzünüz güldüğü için çocuklar inanamazsınız. O kadar az ki böyle güler yüzlü böyle aydınlık yüzlü insanlar yani insanlar biraz tabii çok zorluklar geçiyor böyle son 2 senedir. O yüzden yani ben çok az öğrencide gördüğüm kadar güler yüzlüsünüz ve aydınlık yüzlüsünüz. İnşallah her şeyiniz, yolunuz çok aydınlık olsun sizin de. Çok.
Size tanıştık. Çok
kesinlikle.
Çok sağ olun.
Çok sağ olun. Oldu. Peki hadi hepinize iyi akşamlar. Çok da zamanınızı aldım kusura bakmayın. Çok teşekkür
Estağfurullah biz çok teşekkür ederiz.
Görüşürüz hocam.
Görüşürüz hocam. Sağlıklı günler.
Size de. Bay bay.
Hello everyone. This was our last session and it was absolutely lovely, like all of our sessions. These 2 days, it's been so wonderful. It was so educated. I hope you guys get to feel the same thing as we do. I want to introduce my team. While we preparing this event, we were 30 people. These are my colleagues. We arrange all of it together. So I want to give them a word to finishing parts. Özlem, you can say.
Hello everyone again. I'm so happy for 2 days and I'm very thankful all of our participants for watching us in 2 days, and I want to thank you all my friends for this event. I'm so enjoy in the 2 days because I learned a lot of things which I don't know. I don't have anything about some topics and I really enjoyed.
Şevval?
First of all, thanks for all of our participations. It was honor to see you guys there, and thank you for not leaving us alone for these special days. We all enjoy, and I hope you guys enjoy it too.
Now we want to show our team to you guys.